becoming a world-class institution in research, sciences, and innovation to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, LIPI. LIPI has various research centers throughout Indonesia under the coordination of the chairman of LIPI, Deputy for Life Sciences, Deputy for Earth Sciences, Deputy for Engineering Sciences, Deputy for Social and Humanities Sciences, and Deputy for Scientific Services. LIPI has a pivotal role in drafting and establishing government regulations and policies related to economic, political, social, and cultural aspects. LIPI also manages scientific data and scientific considerations on biodiversity conservation, as well as the publication of global indexed scientific journals, and also appointed as a scientific authority in coaching national researchers and hosting the inauguration of Indonesian research professors. To support world-class research activities, LIPI managed 5 botanic gardens as a center for ex situ plant conservation, 3 museums that have the biggest collection of flora and fauna specimens in Southeast Asia, and regional level and Indonesian natural history, Barunajaya 8 research vessel, Indonesia Cultural Collection, which keeps the largest collection of microorganisms in Indonesia, and 2 international centers, namely Asia Pacific Center for Ecohydrology, APCE, and Regional Training and Research Center on Marine Biodiversity and Ecosystem Health, RTRC Marbest Center. LIPI has produced qualified research results that are beneficial to the society, including biodiversity, environment and maritime, food security, agriculture and farm, health and pharmacy, advanced material, energy, technology, information and communication, social dynamics, humanity and culture, technology innovation and utilization of science and technology, as well as scientific services, testing and development. LIPI's national scientific collaborations include collaborative research, technology licensing, development, application, and commercialization of research results, non-research collaborations, LIPI's expertise consulting, scientific coaching for youth, students, and teachers with various universities, research institutions, local governments, and industry. As for international collaborations, LIPI conducts joint research, joint laboratories, symposium, workshops, international conferences, exchanges for researchers and experts, as well as international scholarships, while being active as a member and a focal point in various international organizations. LIPI will keep continuing to strengthen national platforms by developing world-class researchers and building scientific collaborations to make great impacts on the nation's competitiveness. Every vision begins with a spark of inspiration, and every inspiration is driven by the passion to realize dreams. becoming a world-class institution in research, sciences, and innovation to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, LIPI. LIPI has various research centers throughout Indonesia under the coordination of the chairman of LIPI, Deputy for Life Sciences, Deputy for Earth Sciences, Deputy for Engineering Sciences, Deputy for Social and Humanities Sciences, and Deputy for Scientific Services. LIPI has a pivotal role in drafting and establishing government regulations and policies related to economic, political, social, and cultural aspects. LIPI also manages scientific data and scientific considerations on biodiversity conservation, as well as the publication of global indexed scientific journals, and also appointed as a scientific authority in coaching national researchers and hosting the inauguration of Indonesian research professors. To support world-class research activities, LIPI managed 5 botanic gardens as a center for ex situ plant conservation, 3 museums that have the biggest collection of flora and fauna specimens in Southeast Asia, and regional level and Indonesian natural history. Barunajaya 8 research vessel, Indonesia Cultural Collection, which keeps the largest collection of microorganisms in Indonesia, and 2 international centers, namely Asia Pacific Center for Ecohydrology, APCE, and Regional Training and Research Center on Marine Biodiversity and Ecosystem Health, RTRC Marbest Center. LIPI has produced qualified research results that are beneficial to the society, including biodiversity, environment and maritime, food security, agriculture and farm, health and pharmacy, advanced material, energy, technology, information and communication, social dynamics, humanity and culture, technology innovation and utilization of science and technology, as well as scientific services, testing and development. LIPI's national scientific collaborations include collaborative research, technology licensing, development, application, and commercialization of research results, non-research collaborations, LIPI's expertise consulting, scientific coaching for youth, students, and teachers with various universities, research institutions, local governments, and industry. As for international collaborations, LIPI conducts joint research, joint laboratories, symposium, workshops, international conferences, exchanges for researchers and experts, as well as international scholarships, while being active as a member and a focal point in various international organizations. LIPI will keep continuing to strengthen national platforms by developing world-class researchers and building scientific collaborations to make great impacts on the nation's competitiveness.
Ladies and gentlemen, may I have your attention? We would initiate the program. Bapak Ibu yang kami hormati, acara akan segera kami mulai. The Honorable Dr. Laksana Trihandoko, Chairman of the Indonesian Institute of Sciences, LIPI. Honorable Director of Research Centers and Head of Bureaus in LIPI, speakers and moderators, representatives from ministries, institutions, and universities, distinguished participants, ladies and gentlemen. Good morning. It is a great pleasure for me to welcome every one of you to the international webinar on Indonesian biodiversity, mainstreaming biodiversity conservation, bioprospection, and bioeconomy for sustainable livelihood. Wednesday, September 16, 2020. The webinar is a collaboration event between Indonesian Institute of Sciences, LIPI, with the Ministry of Science and Technology, National Research and Innovation Agency, as a part of the 25th years of HAKTEKNAS. This event will try to bring up issues from biodiversity conservation, the potential of biodiversity for bioprospection, and biodiversity valuation for bioeconomy. There are various speakers from Indonesian researchers, government, foreign research collaborators, and target participants from different backgrounds. So hopefully the biodiversity issue can reach as many sectors and stakeholders as possible. The webinar is conducted virtually and live from Zoom meeting room and official YouTube channel of LIPI. Ladies and gentlemen, the first agenda, National Anthem, Indonesia Raya. Ladies and gentlemen, we will start the international webinar with warm welcome remarks from the chairman of Indonesian Institute of Sciences and officially open the webinar. Dr. Laksana Trihandoko, the floor is yours.
Ya, terima kasih Mbak Renata. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera, salam sehat untuk kita semua. Salam, Om Swastiastu, Namo Buddhaya, salam kebajikan. Ladies and gentlemen, very good morning. First of all, the Honorable Minister for Research and Technology, the Chairman of National Agency for Research and Innovation, Professor Dr. Bambang Brojonegoro. He is supposed to attend the webinar at 11 o'clock, I think. Distinguished speakers, Professor Rosikon, as I can see, there is Professor Rosikon here, and also Professor Jimmy McGuire, Professor Campbell Oweb, and also Professor Kiria Boundy Mills. Welcome to this webinar, and thank you very much for joining this webinar. So first of all, it is a great pleasure for me to welcome and to greet all of you, all participants from across the globe through the Zoom meeting and also the LIPI YouTube channel. LIPI and the Ministry for Research and Technology, we are together organizing this international conference starting from today until tomorrow in the form of webinar related to biodiversity, yeah, either from terrestrial or maritime. So the conference is organized to actually celebrate the 75th of Indonesian's Independence Day and also the 25th of our National Technology Awakening Day. We call it as the HAKTEKNAS as well. Today's webinar is the webinar on biodiversity, webinar Biodiversitas Nusantara, yeah. In short, WEBINUSA, which covers the terrestrial biodiversity. While tomorrow, we are supposed to have the second webinar, International Symposium on Coastal and Marine Biodiversity. So tomorrow's webinar will be focused more on the marine biodiversity. So today's WEBINUSA is actually the third series of bi-monthly webinar of Indonesian biodiversity. So previously, it's already held in May and July, and now this webinar series actually aims to raise the awareness and mainstream the importance of biodiversity conservation, bioprospection, and also bioeconomy in any aspects of our life. Because we know that any biodiversity loss will raise serious implications, not only for the environment, but also for human well-being in many aspects, yeah, either in health, economics, culture, food, and also water security. Therefore, investigating and understanding the status of our biodiversity and the driving factors which might cause biodiversity loss are very, very important. This has become actually the global mission of the Convention on Biological Diversity, the CBD, yeah, actually the target number 1 of the strategic goal of IG target. So currently, the Indonesian Institute of Sciences, we are developing many activities, yeah, including the expedition, either for terrestrial and also ocean research, and also we are constructing many new infrastructures to support any activities for the conservation and sustainable use of resources, in line with our national research priority, and also number 14 and number 15, as I remember, of the SDGs, yeah, the Sustainable Development Goals. So Indonesian Institute of Sciences, LIPI, is currently also the coordinator of National Research Program for Biodiversity, yeah, in this country. As our endeavor in dealing with biodiversity loss, we undertake exploration to uncover biodiversity from all over Indonesia. Actually, from 71 to last year, we have already discovered 513 new species of fauna from Indonesia. Yeah, 513 fauna, and actually last year alone, there were 50 new species of fauna and 28 new species of flora. Yeah. So recently, a new species of what we call as the giant crustacean was discovered from the Sunda Strait, and we will hear more about the status of the Indonesian biodiversity later on in this morning session, I think, of today's webinar. Yeah, I think Professor Rosikon will deliver the talk. LIPI also the home for the largest natural history collections in Southeast Asia, yeah, since long time ago. In particular, the Herbarium Pokoriense already store almost 1 million flora collection since 1841. And on the other hand, the Museum Zoologicum Pokoriense already store more than 2,700,000 yeah, fauna collections, yeah, since 1894. So both museums are amongst the largest museum in the country and also in Southeast Asia region. So furthermore, the latest Indonesian culture collection, which already built in 2014, store nearly 5,000 collections of Indonesian microbes. Yeah. I think Dr. Atit was the head of the INACC, yeah, Indonesian Culture Collection. So starting from next year, I would like to inform you that starting from next year, so Indonesia, represented by LIPI, will have a national repository and depository for biodiversity. We call it as the National Scientific Repository, and which we plan to be accessible for everyone across the globe, yeah, and can be utilized by public. So I'm also proudly conveying the achievement of LIPI in the innovation from biodiversity and in particular bioprospection. Because recently we actively we have been involved, yeah, in what we call as the National Research Consortium on COVID-19, and through the consortium we have developed many innovations derived from many years of experience of our research in biodiversity. Just giving one example is the immunomodulator developed from our local biodiversity for COVID-19 patients with a mild condition. The immunomodulator, we have just finished the final clinical trial, yeah, last month, and we will hear more about this, yeah, and the experience from, I think, from Dr. Mandiri during the afternoon session in this webinar. So continuing the success of WEBINUSA series, I'm delighted to announce that right now, more than 2,000 participants from 12 countries are already registered for this webinar. Thank you very much for all participants. And even though the webinar is done virtually, I hope we can learn a lot from the speakers and obtain fruitful knowledge through this webinar. And finally, I would like to express my gratitude to our invited speakers and also the organizers, and your insights and also perspective, experience will be hopefully awakening our awareness and understanding on the importance of how we manage our biodiversity, how we develop the biodiversity through bioprospection, and hopefully generate something good as the bioeconomy. So further, I also expect that the speakers will stimulate fruitful discussions, yeah, during this webinar, because the participants are coming from various backgrounds. And I am very much looking forward to having rich discussion. And also this webinar will bring beneficial recommendations, not only scientifically, but also to the Indonesian government on how we should manage our biodiversity, how we conserve our biodiversity, and how we utilize it in a sustainable way for the human life. Yeah. So by saying Bismillahirrahmanirrahim, I would like to officially declare that this webinar is open. Have a nice and fruitful discussion. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Terima kasih banyak Pak Handoko. Thank you Pak Handoko for the remarks. Ladies and gentlemen, before continuing the session, let me inform you that today's webinar will be conducted in 3 sessions. The first session with the topic of mainstreaming biodiversity conservation, a multi-phased endeavor toward the improved status of biodiversity, which will be led by Dr. Ruliana Susanti from Research Center for Biology LIPI. The second session, entitled Mainstreaming Bioprospection, Harnessing Untapped Biodiversity for Novel Bioproduct, Challenge and Success Story. As the moderator for the second session will be Dr. Hoirul Himi Setiawan. And the third session would discuss related to mainstreaming bioeconomy, bio-based economy of sustainable biodiversity, and bioresource for better future. This time, Ms. Anggia Prasetyo Putri will be the moderator from 3:30 p.m. until 5:15 p.m. Ladies and gentlemen, in the next 2 hours, we will have the first session of presentation with the topic of mainstreaming biodiversity conservation, a multi-phased endeavor toward the improved status of biodiversity. But before we continue to the next session, I would like to read the webinar rules to the attendees. First, attendees must ensure their device meets the system requirements for Zoom and have stable internet of sufficient prepaid data package for smooth connection. The second, attendees are required to join Zoom webinar using proper ID indicating a real name and affiliation. Renaming feature in the Zoom webinar is enabled if needed. This is a webinar setup. Only panelists, speakers, moderators, and operators are seen and able to make audio-visual communication. Attendees can only see and listen to the panelists. 4, attendees are asked as much as possible to attend all the webinar sessions solemnly and attentively. 5, attendees are welcome to ask questions related to webinar talk or notify committee on technical difficulties via the Q&A and raise hand feature in Zoom or the live chat in YouTube. Attendees are not allowed to write opinion or information that are offensive, racist, or immoral in nature. Organizing committee have the full right to block and remove any attendee who breaks the above-mentioned rules from the Zoom webinar or YouTube live. The e-certificate of attendance will be given to attendees who fill the attendance register shared during the webinar. The e-certificates and PPT slides will be delivered to participants via email maximum 14 days after the webinar. And the webinar recording will be made available for general viewing on LIPI YouTube channel. Dan sebelum kita berlanjut ke sesi berikutnya, kami akan menampilkan sebuah video dari LIPI. Please enjoy.
Merayakan hari jadinya ke-53, Lembaga Ilmu Pengetahuan Indonesia mempersembahkan rekam jejak yang ditorehkan dalam Kaleidoskop LIPI 2019-2020. LIPI memberikan apresiasi kepada para peneliti Indonesia yang memiliki dedikasi tinggi dan telah bekerja keras dalam menghasilkan karya penelitian yang luar biasa. Para peneliti LIPI pun turut menorehkan segudang prestasi di negeri sendiri maupun kancah internasional. Mempertahankan prestasi yang sudah diraih dan meraih prestasi membanggakan lainnya, membuktikan bahwa LIPI konsisten dan terus berusaha sebaik mungkin dalam melakukan penelitian yang hasilnya dapat bermanfaat bagi masyarakat. Indonesia negeri yang kaya dalam keanekaragaman hayatinya. Namun, keanekaragaman hayati itu belum terungkap seluruhnya. Masih banyak keindahan flora dan keragaman fauna yang masih tersembunyi di balik tirai indah alam Indonesia. Para peneliti LIPI merekam temuan baru berbagai flora fauna di seluruh Indonesia. LIPI ada untuk masyarakat. Oleh karena itu, LIPI berusaha selalu mendekatkan diri pada masyarakat, menyebarluaskan hasil penelitian yang telah dilakukan pada masyarakat, dan meningkatkan kinerja menjadi lebih baik untuk meningkatkan bakti pada negeri. Dalam masa pandemi COVID-19 saat ini, LIPI terus melakukan berbagai macam riset untuk mendukung secara nyata percepatan penanganan COVID-19. Tidak hanya melakukan riset dalam bentuk kajian dan produk, LIPI juga mengembangkan pelatihan untuk pemenuhan SDM terlatih. Berbagai macam prestasi telah LIPI torehkan, melakukan riset yang secara nyata menghasilkan kebermanfaatan bagi masyarakat Indonesia. Tidak berpuas diri, LIPI akan terus melakukan berbagai riset dan menorehkan berbagai prestasi untuk negeri kita tercinta, Indonesia. Sampai jumpa di Kaleidoskop tahun 2020-2021.
Ladies and gentlemen, for the first session, may I kindly invite our moderator, Dr. Ruliana Susanti from Research Center for Biology LIPI. But before Dr. Ruliana start the session, let me introduce our moderator first. Dr. Ruliana Susanti is a researcher in the Research Center for Biology LIPI who specializes in plant ecology and forestry. She is also a former head of collaboration and information division in RCB LIPI. In biodiversity policy activities, Dr. Ruli actively involved as the Indonesian expert team for an intergovernmental conference and convention such as IPBES and BBNG. For the past 6 years, she has been assigned as the Indonesian delegate for the United Nations Convention on Biological Diversity, UNCBD, particularly SBSTTA and Nagoya Protocol issues. Dr. Ruli pursues bachelor and master degree from Bandung Institute of Technology and doctoral degree from Kagoshima University, Japan. Finally, Dr. Ruli, time is yours.
Okay. Thank you very much, Renata, for your kind introduction. Good morning, everybody, and also good evening for our presenter from the USA. Ladies and gentlemen, researchers, lecturers, and students from all around the world, welcome to this webinar. It's an honor for me to facilitate the first sessions in this international one-day webinar on Indonesian biodiversity. In this session, we will discuss about mainstreaming biodiversity conservation, a multifaceted endeavor toward the improved status of biodiversity. Sorry. For your information, there are 2,069 persons registered in this webinar who come from different backgrounds. There are researchers, teachers, lecturers, students, technicians, not only from Indonesia, but also from several other countries. And 60% of it registered for this session. So it is a big job for me also. Before we start our sessions, please allow me to remind all attendees again the rules for all of you. First, do not forget to renaming your feature for attendees who attend from the Zoom channel. And this is a webinar setup, so only panelists who will be seen and able to make audiovisual communications. Attendees are welcome to ask questions later related to this webinar, talk, and notify and later committee, and I will raise the question in the Q&A sessions. Please do not write your opinions or information that are offensive, racist, or immoral in nature. The e-certificate will be given later for all attendees who already filled the attendance form. And the e-certificate letter will be sent to you by the committee. Well, now it is time for me to introduce our panelists today. Our first speaker is Professor Rosikon Ubaidillah from Indonesian Institute of Sciences, followed by Professor Jimmy McGuire from University of California, Berkeley, followed by Dr. Kiria Bondi-Mills from University of California, Davis, and last but not least, my friend Dr. Campbell Owe from University of Alaska. All our speakers this morning will give presentation for about 25 minutes, and After all the presentation, we will have about 15 to 20 minutes discussion. So it depends on the time later. Now let's begin our first sessions. Our first speaker, Professor Rosikon Ubaidillah. Professor Rosikon graduated from Gajah Mada University. He pursued a master's degree from Imperial College London and doctoral degree from Ibaraki University, Japan. He started his career as an entomologist in 1985 and published many national and international publications. His expertise is recognized internationally in 2015 up to 2018. He was chosen as a member of multidisciplinary expert panel of intergovernmental platform for biodiversity and ecosystem. Professor Rosikon, are you ready? The time is
okay,
yours,
I'm
and
ready.
I will remind you if the time almost off. Thank you.
Okay, thank you, Bu Ruli. Terima kasih, Bu Ruli. Assalamualaikum warahmatullahi wabarakatuh. Distinguished participants, ladies and gentlemen, good morning, good afternoon, and good evening to where you are. My name is Rosikon Ubaidillah, researcher at Center Research on Biology. I'm pleased to have an opportunity to join with this webinar and to present this important issue on Indonesian biodiversity. Let me share my presentation, Bu Ruli.
Yes, sure, absolutely.
How can I share? Oh, really? Can the committee help that I have a presentation in the committee, but I cannot find here my presentation. Sorry.
Didi, can you share, Professor?
Oh, goodness. I'm sorry in the technical problem here, really, because my computer now is a little bit problem.
Didi?
That's you can find my presentation.
Wait a minute. Yes, we already open your presentation.
Okay.
Can you see it?
You find it?
Yes.
Okay. Okay. I will start. I will start that I would like to present the status and trend of biodiversity of Indonesia. I refer to the CBD 1992. We refer that the biodiversity is variable among living organisms from all sources, including inter alia, terrestrial, marine, and other aquatic ecosystem, and the ecological complexes of which they are part. This include diversity within species, between species, and ecosystem. I think everyone know that's about biodiversity. And there are three levels of biodiversity. If I say that ecosystem biodiversity, species biodiversity, and chain biodiversity, I will focus on the species biodiversity status of Indonesia. Indonesia is really rich in natural, culture, and biodiversity. We are in the strategic biogeographic between two Earths, geographical region, Indo-Malaya Ocean. And we have around 17,000 islands, 7.7 million meter square, and 5.8 million meter square in marine and coastal. 47 ecosystem milestone type vegetation. And we are in the top 5 top megadiversity countries in the world. We have about 29,375 species of vascular plant, around 7.5% of world's plant, and 12% of the world's mammal, around 773 species. ranking the first worldwide on the mammal, and around 755 species of reptile for trends, and 6.7% of the world's bird, around 1,711 species, and 30 species of primate, around fourth rank of the world, etc., etc. Really, we have a rich and cultural biodiversity, more than 400 ethnic groups, around 665 different languages and dialect in all around Indonesia. If we look at the detail of the major taxa on biodiversity in the comparison to the world's species, in mammal, as I said, that we have around 670 species, is around 10.5% of the world species. And unfortunately, around 191 is under threatened. And about 46 of that is about 46% is endemic. Fish also, we have a wide reach, around 4,700 species in Indonesia, and around 0.52% of the world. And quite unfortunate that 231 under threatened. Reptile and birds also, that's very rich, and but birds is around 15.5% of the world's species, and but 160 species is under threat. Endemism around 31. Reptile is quite high endemism, around 46 species, 46% are endemic, and around 11.3, around 6.7 is percent is world species, etc. We have data here in the major taxa. I will discuss more later. Presentation will be focused on status biodiversity in 7 geographic regions, because we have a really large area and we have many islands. So we have just concentrated on the 7 mainland: Sumatra, Kalimantan, Java, Sulawesi, Lesser Sunda, Moluccas, and Papua. And I will also focus on well-known species biodiversity. For plant biodiversity, it's quite high diversity here. That, for example, like biopids, we have around 2,723 species, around 13.28 are Indonesia's from the world's presentation. And also Pterodopid, 12,800 in the world. We have around 1,845 species, around 14.41% of the world. Plant diversity also, we have many distribution from Sumatra, Java, Kalimantan, etc. Java is quite still high in the diversity, I think, because of the more exploration here, because it's compared to the Papua and Moluccas, Java is still high in the diversity. In the group of taxa for the biodiversity from the Sumatra, Kalimantan, Java, we have Sumatra for vascular plant, we have around 5,600, Kalimantan 5,500, Java 6,000, still high, and Sulawesi 6,700, Lesser Sunda Island is 490, and Moluccas is quite high, and 2,275, and Papua 3,928. That is our status of the plant biodiversity in Indonesia in the 7 regions of geographic in Indonesia. If we just look at stress on the fixed plants, fixed species, Indonesia assumed to be the richest of fig in the world. According to the Rasmus, there are about 2,037 species distributed, and 100 species in Sumatra 82 species in Kalimantan, 92 species in Sulawesi, 7 species in Molucas, and 79 species in Papua, and 77 in Java. This fix is, of course, this is species of about species of bird and mammal, depending on that fit for food in the forest or in the terrestrial area others. If you look at this, the certain taxa on mammal itself, we have about 773 species of mammal in Indonesia, and about 46% are endemic, and about 12% of bird species. And Indonesia is known to be the richest of the mammal in the world. And if we look at the distribution of the mammal from Sumatra, Kalimantan, Java, and Sulawesi, Kalimantan is rich in mammal, and Sumatra as well, and Papua, and also the rich urban endemism is Papua is more endemic. That's around 49% of mammal of Indonesia is in Papua. However, the trend of mammal, according to the list of species index, is decreasing from 1992 to 2020. Look at the status of protection and government. At the moment, we have 137. Just last year, government produced the law to protect 137 species of mammal is protected, and we have 56 in Appendix 1 and 123 in Appendix 2 in CITES, and the IUCN Red List also noticed that 25 species are critical and 56 species is critical endangered. This is the situation of the mammal status and trend of the biodiversity of mammal. Birds also follow that the mammal. We have around 1,794 species of bird of Indonesia. About 400 species are migratory bird, and 527 are endemic for Indonesia. And the distribution is Papua is quite high in species, and Molucas also, and also Sumatra. Sumatra also high in species. Birds trends, we have around 33 species under threat. This is according to the LSI list species index. We have around 557 species are protected. That is just recently declared by government. And in the CITES status, we have 16 species under Appendix 1 and 241 under Appendix 2. In IUCN Red List species, we have 30 species under critical and 48 under endangered species. Follow the amphibians. There are about 365 species amphibians of Indonesia. Around 160 species are endemic for Indonesia. Around 50.6 species is very, very high in endemism of amphibian in Indonesia. And if we look at the data from the distribution, Papua and Kalimantan is very high in species richness, and followed by Sumatra. However, the endemism very high in Papua and in Sulawesi. Papua is around 52.3% amphibians from Indonesia are endemic for Papua. Trend of amphibians is also decreasing. of the trend 33 species under threat. And there are government law is only one is under protected. And there is so many, not so many information on IUCN, so they are still working on that. So 4 species have been recorded as endangered species. Reptile, just reptile is more critical than amphibian in that information. There are about 723 species of reptile in Indonesia, and about 333 species are endemic. Around 46% of the Indonesian reptile are endemic of Indonesia. About 40% of bird Varanus itself, are about 40% found in Papua and Maluka, occur in Indonesia. So it's quite rich in species of Varanus, including that Varanus cobordionesis, of course. And the distribution of the reptile from Sumatra, Java, and Kalimantan and Papua, the endemism is rich in Malukas and also in Lesser Sunda Island and in Sulawesi. There is 3 area, and also in Sumatra is very high endemism. How about the trend? We recorded 30 species under threat, and according the LSI list of species index, the reptile decreasing some species in Indonesia. And last time that government just produced the law that protected to 37 of reptile in Indonesia, and 9 species under Apedic 1 and 20 species under Apedic 2 in CITES. And in IUCN species red list, there are 7 species under critical endangered and 5 species under endangered species. Crustacean species, there are estimated about 1,635 species of crustacean, including marine on freshwater, of course, found in Indonesia, of which 207 species of crustacean freshwater are endemic, and 21 species freshwater crayfish all endemic, and about 755 species of marine ocean, around 443 species of crab, and 311 species of shrimp and lobster is found in Indonesian Ocean. If we look at the distribution of the crustacean, Sulawesi is very rich, and Sumatra followed by Papua and Sumatra. This is not necessary that this is the quite confirmed information, because there is still so many areas that have not been explored. I think that will be changed in the near future. If you look at the endemism, Papua is very high, around 57.8% found in crustacean found in Papua, and also Sulawesi, also 59.4% are found in Sulawesi. Crustacean trend of conservation is several crustacean species in 7 geographic area in Indonesia has decreases in the population, and there are about 50 species of crustacean recommended for protection. This is by LIPI and other team. Desi said that consisting of 17 species of marine crustacean recommended for protection, and 32 freshwater crustacean species are recommended for protection, for the full protection and limited protection. Sorry. Fishes, we have 4,782 fish, and around 40% are freshwater fish and 60% are marine fish. And if we look at the distribution, this is information from the fish-based database that Sumatra and Kalimantan is very high in species, in Papua especially. So that's the distribution of fish found in Indonesia. The trend, there are recorded about fish, 231 species are under threat. This is according to fish-based database, and 26 species are protected by government law, Indonesian government. I mean, that one species in Appendix 1, 17 species in Appendix 2 under CITES and IUCN Red List. Notice that 25 species critical and endangered, and 34 species in danger. So this is the trend of the fish. Mollusks. There are about estimated 4,000 species of mollusks, 1,000 species of bivalve, and 70 species of scaphoid, 100 species of cephalopod, etc., etc. This the number of species according to the Norcia Perscom, that the list of the species is Kalimantan is quite rich, mostly in, this is only in land snail. The land snail is Kalimantan is very rich in species. The trends, there are about 63 species of mollusks that are endangered, are recommended to for protection. And under the IUCN Red List, there are 26 species in critical and 13 species in endangered. And the government law protection is 5 species under the government law protection. Okay, let's go to the butterflies insect. There are around 1,900 species of butterfly. That's according to the Ibu Peggy, that 2,500 estimated occur in Indonesia. 2,500 species occur in Indonesia, and about 11 species in the world, and which 273 species are known to be endemic. This is the distribution of the butterfly. Sumatra is very high, and Kalimantan, and Papua is also followed by Papua and Sulawesi. But I think it's Papua. When we have more exploration, it will be rise up the species number of Papua butterfly. Trend of conservation. That's 26 species protected under government law, under Indonesian government law, and 26 species is Appendix 2 under CITES. And there are still working on red list species in IUCN that have not been reported, but recently they'll be reported how about the situation of conservation status of critical, endangered, or vulnerable. Special for honeybee, I will rise up. That's honeybee. There are about 7 species honeybee in the world, all found in Indonesia, and 2 species of which are endemic in Indonesia, namely Apis kosevnikovi and Apis nigrokinta. That is the distribution of honeybee. We have 7 that 2 species already, 1 species already extinct, and 1 species under threat. That is the richness of the honeybee. Even though we are rich in honeybee species, in fact, to meet national needs of the honey, we will still import 62% of honey every year, with a value about 13.68 million US dollar. It's quite big. This is our challenge to how to increase our product of honeybee, because we are rich in species, rich in flower, and we are in the equator, that every time there are flowering for food of bees. Microbe, the last is of a microbe status, the accession number of LIPI microbe collection. We have a bacteria of 1,352, accession number, actinomyces 529, and microalga 150, protozoa and yeast 1,627, and filamentous fungi 1,000 and 29 accession. That's according Seskiawan Presscom 2020. This is the situation of the INACC Center Research for Biology. So our biodiversity plays an important role in providing us with food, with water, with shelter, clean air, and medicine. But we have a very limited. Of course, in the future, we have to think. And very fragile. Our biodiversity is very fragile. We look at the loss of biodiversity. We look at the forest fire that have been destroy many biodiversity. It takes our full commitment to protect and maintain to ensure its sustainability. That is my statement here. And action must be taken to achieve our commitment. We have a two commitment: the Agenda Biodiversity Target, IG target, and we have a national target. So where we have to go, we must to decide where to go to maintain our biodiversity. Our challenge is many.
You have 2 more minutes.
Okay, Buruli, sorry, that's our challenge as a scientific community, as a researcher. We must take an opportunity to step and commit to protect biodiversity and use it as in a sustainable manner. As a researcher and scientific community, we must take the right step and commit to conduct better research direction in order to produce product that can be used for human welfare and maintain the health of nature. We also must take an opportunity to do research and developing and implementing more effective conservation and leading to the sustainable use of biodiversity across the Indonesia, including more effective greening of cities. Moving toward more effective multidiscipline, transdiscipline, and interdisciplinary research, and conducting mutually beneficial global research collaboration. We have to do global research collaboration to maintain our biodiversity. Our biodiversity is still poorly studied. The value is not being explored, and the utilization exists only based on direct use with no added value at all. So that the biodiversity survey, taxonomic study remain critically important, particularly in poorly inventoried area. And also the science and technology related to biodiversity accelerate too fast and need to be anticipated to get most benefit from the biodiversity. I've just briefly, Buruli, I just saw that the Indonesian fauna survey, that is many areas that have been explored, but still many also that have not been explored. Only birds and mammal and etc. That the data is more complete other than invertebrates, still very, very low, around less than 10% have been known to be have been identified. Well, where we have to go, where we have to find the funding. There are many challenges to get research collaboration. If we look at the data from the research collaboration of foreign research permit for Indonesia, sourced from the Ristek BRIN, every year around 700 proposals of foreign research to do research in Indonesia. And that's mostly as from Germany, Japan, United States, Australia, etc. If you look at the spending money of the 10 biggest research funding by countries, the German, United States, and Japan still dominate in the investment of the research on biodiversity. Look at that in total of the research. So if the total the research funding for the foreign research, around 48% are biology. But if we look at the fishery, agriculture, oceanography, ecology, and agriculture, there are also inside there is a biodiversity research. So if you look at in the total, I think there is around 75% of the budget research delivering to the Indonesia, doing research in Indonesia from foreign research is around 75% funding. So that is the investment. In closing, Bu Luli, I hope that with a discussion on the role of biodiversity and the role of biology in the conservation and sustainable use of biodiversity, including research, conservation, management strategy, as well as education and outreach, can produce significant and tangible products for our welfare. Thank you, Bu Ruli. Hello?
Thank you very much. Hello? Yes. Thank you
Yeah.
very much, Professor Sikon, for your
Sorry, I'm flowing, but I don't know whether there's something connection or not. Problem.
No, it's quite okay.
Okay, good.
It's okay.
Good. Thank you, Bu Ruli.
It's okay.
Thank you, everyone.
Well, thank you very much for your presentation, Professor Sikon, for your update on Indonesian status on biodiversity. I'm not going to resume your presentations. Everybody can make notes for what you were highlighting at the end of your presentations. Next, are you there, Jim? Before
I'm here, but I can't hear you, Bu Ruli.
start to listen to the presentation,
I'm okay to begin.
please allow me to introduce Professor Maguire. Professor Maguire. So Professor Jimmy Maguire was now working as a professor and also curator for herpetology in the Museum of Vertebrate Zoology, the University of California. Is there a CV PowerPoint? Okay. He has shown interest on herpetology since he wrote his master thesis back in 1990s. And since then, he published many publications in the field of phylogenetic, systematic, and evolutionary biology. For more than 20 years, Professor Maguire conducting research for Indonesian herpetofauna in collaboration with LIPI, ITB, and University of Indonesia. Professor, the time is yours. I will remind you again if the time is almost off. Thank you.
Okay, I think I need to share my screen.
Yes, please.
I don't know what you all see.
Yes, we already seen it, but you have to, yes.
Is there just one item on the screen?
Yes,
Just one.
just one.
Okay, thank you. Well, thank you so much for this opportunity to speak at this webinar, at this event. I'd like to thank the organizers for inviting me and giving me this opportunity. So today, I would like to talk about the work that I have been undertaking as part of a large collaborative team investigating the biodiversity of the mountains of Sulawesi. And I'm just going to start with an outline. I thought that before I jump in and talk about the research, that I should first say a few general words about Indonesian herpetological diversity, which I think is a nice follow-up to the information that Pak Rosikan just provided. And then I will talk a little bit about sort of my perspective on how the type of biodiversity research that I do can be informative for conservation efforts. And then finally, I'll do what I think the organizers were really hoping the main event would be, which is to talk a little bit about my experiences working in Indonesia as part of a collaborative team and what the challenges and rewards are for that sort of program. So I'm going to begin with just a little bit of information about biodiversity, herpetological diversity in Indonesia, starting with amphibians. So there are currently 8,224 recognized species of amphibians worldwide. And of these, a slightly updated number than the one that Pak Rosikan provided is that there are 378 that are native to Indonesia, so documented from Indonesia. And that includes 368 species of frogs, such as these two here, as well as 10 species of caecilians. But I can tell you right now that this number is a really massive underestimate. And there are clearly many, many more species of amphibians in Indonesia than have currently been documented. And one nice sort of bit of evidence that tells us this is just to look at the number of species of frogs that have been documented from Papua New Guinea. So Papua New Guinea, it composes the eastern half of New Guinea. Papua New Guinea has 401 documented species of frogs, and the entire amphibian fauna of Indonesia, which includes Papua province, the entire western half of New Guinea, is only 378 species. So it's very clear that the numbers are underestimated for Papua. And I think this is simply because many more researchers have been able to conduct fieldwork in Papua New Guinea than have been able to conduct fieldwork in Papua province. And in fact, if you look at the numbers that, Pak Rosikan indicated, there are 150 species of amphibians documented from Papua, which is far fewer than the 402 that have been documented for PNG. So very clear that there's many, many more species there. And the same logic also applies to Borneo, which is much better studied on the Malaysian Brunei third of the island than on the Indonesian 2/3, Kalimantan. So 378 species seems like a lot for an individual country, but the number must be more like 700 or 800 species. And this also doesn't even take into consideration that this is absolutely the age of discovery for amphibians and reptiles. And we discover new species on all of our fieldwork. There are new species everywhere in Indonesia to be discovered. It's not just this undersampling relative to adjacent areas in New Guinea and Borneo that that tell us that the fauna is much larger than we currently recognize. Now, the same thing applies to reptiles. There are currently 11,341 species of reptiles, non-bird reptiles in the world. And of these, 771 species are currently documented for Indonesia. But this number is almost certainly a dramatic underestimate, just as was the case for amphibians. So Indonesia is certainly one of the 2 or 3 richest countries in the world from a herpetology diversity standpoint. That probably applies to most terrestrial organisms, and Indonesia must be the most rich country in the world from a marine biodiversity standpoint. So the next thing that I would like to do is talk a little bit about our research program investigating vertebrate and arthropod diversity on mountains of Sulawesi. And this is a chance for me to talk a little bit about what a large collaborative effort, research effort, sort of looks like. And I'm going to show you a lot of pictures of field sites and sort of how we do things. I thought that might be a nice way to introduce expeditionary collaborative fieldwork to a large, diverse audience. And I'll talk a little bit about some of what we've learned when I get to the end. But I thought it'd be a little bit more fun to just show some pictures of our fieldwork and also to tell you a little bit about why we're conducting this work on Sulawesi. So this project that I'll be describing, it's funded by the United States National Science Foundation. And the program within the NSF that is supporting the project is called the Biodiversity Discovery and Analysis Program. And so that program is really motivated by the opportunities to discover additional diversity around the world. And so a main objective for our project is to try to do just that, go in the field and collect reptiles and amphibians and birds and mammals and spiders and other arthropods, and to document the diversity, to find and describe new species, and then to study that diversity. But before I get too far, I also just need to mention that Sulawesi has some special features that, that really caused us to organize our fieldwork in a particular way. And I'll tell you a little bit more about this in a moment, but Sulawesi has a bunch of regional, it has regional endemism. So there are a number of areas of endemism on the island. And so we were really motivated to work within those areas of endemism. This map here shows the areas of endemism as documented for monkeys. So you can see that there are little black bars here that separate the different areas of endemism. Each of these regions tends to have its own unique species relative to other parts of Sulawesi, which is really interesting and a fun thing to study. In fact, a lot of my prior research has been focused on trying to understand how speciation has happened across the island. And this has been more or less a two-dimensional study, a study where we were considering latitude, north-south, and longitude, east-west. And now this new project is thinking about a third dimension, the elevational dimension, and how diversification happens on mountains as well, because we know that that's important. And so one of our objectives is to perform elevational transect surveys to survey across the elevational gradient of mountains on Sulawesi, and we want to do it in each of the areas of endemism so that we capture both regional endemism and elevational stratification as well. Hopefully that will be obvious with the next few slides. So before I even do that, I do want to just provide a few sort of vital statistics for Sulawesi. I like to present these facts when talking to an audience that is from America or from some other place besides Indonesia. You all probably know everything that I'm about to tell you, but I thought I would go ahead and say it anyway, since we have 800 or 900 people watching this presentation. So Sulawesi is, of course, a tropical island. It resides between 2 degrees north and 6 degrees south latitude, so it spans the equator. It's a huge island. It's the 11th largest island in the world, larger than Java. 18 million people live on Sulawesi. It's a huge island, like a small continent. The island is special in many ways, and one way is that it's a highly mountainous, topographically complex island. So there are large areas, large swaths of Sulawesi that are over 1,000 meters in elevation. So it can give higher elevation than 1,000 meters. There are 15 summits on Sulawesi that are greater than 2,500 meters in elevation. There are 9 summits that are above 2,800 meters, and there are 6 summits that are above 3,000 meters. And that, together with the fact that Sulawesi has been isolated for its entire history of about 25 million years, everything on the island had to get there by overwater dispersal. That and this topographical relief sets up a lot of opportunity for speciation to occur along elevational gradients as well as regionally within the island. So we're really interested in that. Sulawesi is famous for having many endemic species found nowhere else. And these are some examples of famous endemics of Sulawesi, mammals and birds here, but the high degree of endemism applies to everything that's on Sulawesi. And evidently the fraction of birds that are endemic to Sulawesi, it's the highest fraction of any place on Earth, even more so than an older island like Madagascar. So Sulawesi is famous for its endemism. But it's also important to understand this regional diversification and these areas of endemism that I described a moment ago. So this is really well exemplified by the monkeys of Sulawesi. There are 7 species of macaque monkeys on Sulawesi. They're illustrated here. And each of their ranges is color-coded on this map. And so you can see that each little sector of Sulawesi here has its own species. And this turns out to be a common pattern among the biota of Sulawesi. We see this in many other taxa. The boundaries between species might be in slightly different places, but this pattern of regional endemism is quite strong and general for the island. So consequently, for our mountain study, it's really important to us that we sample mountains in each of these areas of endemism so that we can capture both the regional endemism and also we hope to also capture the elevational endemism as well. So these are the mountains that we have already sampled during the first 5 years of our project. So we've been to 6 mountains so far. We're still hoping we can do 2 mountains on the northern peninsula and potentially revisit Gunung Mekongo, which has already been studied by a large team of LIPI researchers along with researchers from UC Davis, but we would still like to go back there if we can wrangle the money to do it. And if any of us ever gets to go in the field again. These are some photos of some of the mountains that we've already visited. It includes the largest mountain on Sulawesi. That mountain is actually Gunung Rantomario. We visited Gunung Latimojong, which is adjacent to Gunung Rantomario, which is 3,440 meters, a very large mountain. But all the mountains that we've been visiting have been quite large. The last mountain that we visited was a smaller mountain, but which is called, it doesn't even have a name. but it's near the village of Iliomata, but that one I'm not going to talk about today. So when we go to these mountains, we have a team that's composed of mammalogists, people who study mammals, herpetologists, ornithologists, ichthyologists, entomologists, and parasitologists, all of which are working together out of field camps on these mountain slopes with the goal of sampling across a large elevational gradient. And so I wanted to show you some pictures of what it looks like to organize a trip like this, mostly because I thought that it's fun and hopefully everybody will be interested. The teams are very large. This doesn't even really capture the scope of it. On our Gunung Torempupu expedition, we had 42 people in our field camp. So, these are large teams composed of mostly Indonesian researchers and local assistants, but also, a fairly large number of Western scientists as well. And provisioning these field camps is a big task. We have to bring a lot of gear up the mountain. It's always carried up in rice sacks. These are some pictures showing how this works. We have a base of operation in the nearest city, and where we can buy a lot of the provisions that we need, and then we load up a big truck and we drive it to the village that's closest to the mountain, and then we have to try to get the gear up the mountain. Sometimes that starts with walking, sometimes that starts with motorcycles if there's some trails, but ultimately we wind up having to walk that gear to the final field campsites, and all of us have to carry what we can up the mountain, although there's far too much gear for our team to carry on our own. So we always hire local people who are much stronger, faster, better than us, who carry the gear up the mountain. And we build a field camp. And that field camp essentially becomes a small laboratory in the field where we're able to conduct our research over the course of anywhere from 18 to 25 days, usually, at one of these sites. And we have a generator, and so we can have some electricity, and it'll basically allows us to make a lab in the field. This particular lab here on Gunung Torempupu was 6 hours hike from the nearest village and across a pretty large river. So pretty amazing that we could get all this gear up the mountain to establish our field camp. This is a higher elevation field camp. This is our field kitchen. And by establishing camps like this, it has allowed us to go to visit these high elevation forests that are otherwise very difficult to reach. Many of them are not protected in any way, but there's still pristine habitat available just because of distance, because it's hard for people to get all the way up to those high elevation sites. They're often utilized by local people, but not usually for farming. And so there's still forest in place on many of the big mountains of Sulawesi. This is an example of mossy forest or cloud forest. And I'll just show you a few pictures showing this spectacular forest that we're able to work in. So fortunate to be able to work in it. And then as we get higher on the mountain, the vegetation changes again. And if so we get near the summits, we end up with this, with trees that are smaller and that have smaller leaves with waxy coatings that allow them to be more desiccation resistant. And at these elevations, it can be much colder and the vegetation is much lower. And of course, these changes in vegetation also result in there being different species of animals in these places as well, which is the reason why we're trying to get to these elevations. I thought I'd tell you just a little bit about some of the preliminary results from our work thus far. I'll tell you a little bit more about the amphibian and reptile work. And I just wanted to be able to present a list of some of the key players that have been participating in the fieldwork to show you the sort of scale and scope of the research team that's involved in the fieldwork. So here we see the list of people who have been conducting the mammal-focused work. And the mammal research has resulted in collections of large numbers of specimens, discoveries of many new species, including new genera of mammals. And of course, the ultimate goal is to also produce, in addition to species descriptions, more synthetic papers trying to understand how speciation has happened across these elevational gradients on Sulawesi. And that work is ongoing. This is a slide showcasing the amphibian and reptile team, my team. You can see that there are a number of participants who are involved in the fieldwork and in the research that's following the fieldwork. and we've produced a fair number of papers. Excuse me. I just want to tell you a little bit about some of what we're discovering in it, and it's actually really relevant to this discussion of underestimated diversity on Sulawesi as well. So for example, we have large studies now that we're undertaking where we're studying the flying lizards of Sulawesi. And the flying lizards don't show any elevational diversification on mountains, but they do show a lot of regional variation, including cryptic speciation. And so for example, we believe there are at least 9 species of flying lizards on Sulawesi proper, and another 6 on islands immediately adjacent to Sulawesi. Only 3 of those Sulawesi species have names at this point, but there are definitely more species than are currently recognized. This is an example of a fanged frog. Fanged frogs are extremely interesting. They exhibit both regional and elevational diversification. There are currently 5 species of fanged frogs recognized, but we believe that there are potentially as many as 40 species of fanged frogs on Sulawesi alone. So on Pakrosikan's list, the current knowledge is 40 amphibian species total on Sulawesi, but there may be nearly 40 undescribed limnonectes on Sulawesi. And that doesn't even include other, all the other amphibians that are also present on Sulawesi. For example, Oreophrine, a microhylid frog shown here, this is a group that shows some really interesting elevational diversification on Sulawesi. Most mountains have 2 or even 3 species of Oreophrine, and they sort themselves elevationally in bands on the mountain. But there's also regional diversification. We don't even know how many species of Oreophrine there might be, but only 3 of them have been named so far. So, there might be 20 species of Oreophrine on Sulawesi. 3 have been named. This is the kind of pattern that we're going to see all over, not just Sulawesi, but other parts of Indonesia. So we know that the fauna is really dramatically, in the terms of the numbers of species, is dramatically underestimated. We've been doing bird work, of course. This is an example of the team of researchers that have been involved in the bird field work. We're unlikely to find new species of birds because birds are the one group of organisms that seem to be almost entirely known for the whole world, even very remote places like Indonesia. But there are likely cryptic species to be found on Sulawesi, and we are developing these comparative phylogeographic data sets for birds that will tell us whether or not that happens to be the case for Sulawesi, where we don't actually know whether or not cryptic speciation has been an important process. And we have also arthropod teams making collections. And with arthropods, all bets are off. I mean, we collect thousands of specimens. There are thousands of species. It'll probably take years to sort it all out. We have a graduate student who's studying the spiders, who is more likely to be able to sort out all the species that she and the other researchers have been collecting. But with respect to all the other arthropods, it's just overwhelming diversity on Sulawesi. And we've also been collecting parasite data for all of the vertebrate groups, as well as collecting chytrid fungus data for the frogs. And if anybody have questions about, if you know about chytrid and want to ask me questions about it, I'm happy to answer. It's good news from the chytrid standpoint when it comes to the Sulawesi amphibian fauna. So that's the sort of covering the field work in that project. And I'd like to spend just a moment talking about conservation and what sort of I believe this kind of biodiversity research can do to inform conservation planning and hopefully conservation protection. I think that we all know that it's difficult to protect diversity if you don't know that it exists. So it's really important for teams of biodiversity researchers like our teams to go out in the field and identify the diversity and fully document it. And I think that that research has the opportunity to potentially identify areas that are particularly important for protection, areas that have particularly high endemism or particularly large numbers of species, and also areas that are underprotected at the moment. And so I'd like to just make 2 points here in that regard. On the island of Sulawesi, we have these areas of endemism, and we know from our biogeography work that these areas of endemism exist. Many of the areas of endemism have protected areas on them. They have national parks or they have chagar alam, but a few of them don't. And these would seem to be areas that should be prioritized for conservation planning by government agencies that are engaged in that sort of effort. So for example, the Eastern Peninsula doesn't have any large conservation areas. I mean, there's Morowali, I suppose, but it's right at the interface, the biogeographical boundary. The Northern Peninsula, the southern third of it, also has, you know, a few areas, chagar alam in this area, but is not as well protected as it might be. And we now know from our elevational diversification work that we also need to be thinking about protecting both high and low elevation habitats. And so, for example, Gunung Bawah Kereng on the Southwest Peninsula here, Bawah Kereng and Lompo Batang both are their sister peaks on one massif. None of the high elevation habitat is protected, but we know there's tons of endemic diversity there. So Bantimurung National Park is present in the lowlands, but none of the high elevations are protected. It would be tremendous to see what's left of the forest on Lompo Batang, which is really, or sorry, on Bawah Kereng, which is really only above 1,400 meters, to receive some attention in terms of protection. So the last thing I'll do is talk a little bit about collaboration. And I fear talking about the challenges of collaboration, but I will go ahead and do it because I think it's important. And then I'll talk about the rewards, which I don't fear talking about at all. From a challenges standpoint, I would say that the biggest challenge for foreign researchers is with the permitting process. And I would first give a shout out to RISTEC, because I find that RISTEC, one, they've really dramatically improved the situation in recent years by developing their online system for the permit application process, which helps a lot. And they're always, you know, very professional and helpful. But we have an ongoing problem dealing with research visas, which, since I think I'm running out of time, I don't want to spend a huge amount of time talking about, but it has become a problem in recent years. I'm happy to talk about that in the panel discussion if people would like to hear about it. And the other thing that I thought I would mention is just that the way that the permits are managed, this is the Boule perspective, by the way, is that you apply for your permit, and then when your permit has been approved, you show up in Indonesia, and then you have to spend 1 to 2 weeks walking paperwork around to various government offices, which is expensive because you have to be based in Jakarta when you're doing this. If you have a large team, everybody has to be holed up in a hotel, an expensive hotel in Jakarta or in Bogor. And all of that makes it expensive, and it also makes it impossible to really contemplate short-term fieldwork in Indonesia unless you live in Indonesia. And so, you know, for many researchers, they can't really get away and spend more than, you know, 2 or 3 weeks doing fieldwork. And you really can't even contemplate having a program in Indonesia if you're a foreign researcher and that's your situation. You really have to be committed to 5 or 6 weeks or 2 months of fieldwork because of the way that the process is organized. And it would be so much better if that process could be streamlined as it is in many other countries. I do want to talk about the rewards because, you know, I've spent 22 years working in Indonesia, and I'd like to spend the rest of my life working in Indonesia. So clearly the rewards for me dramatically outweigh the challenges. The opportunity to work in this incredibly biodiverse country, the most interesting in the world, in my opinion, from a biodiversity standpoint, and especially from a biogeography speciation standpoint, I mean, it's just tremendous. It's worth whatever obstacles I have to overcome. I also feel so fortunate to have been able to work with so many Indonesian scientists. This recent 5-year project that involves people working on not just amphibians and reptiles, but so many other taxa, has allowed me to engage with so many just amazing people in Indonesia that, I mean, it's really just been fantastic. It'll be a real shame if I'm not able to continue to do that kind of really large scale collaborative research in the future. We'll have to, you know, write more grants to be able to continue doing that kind of work. Of course, playing any role in biodiversity discovery, and especially the possibility of assisting with long-term protection of that biodiversity in Indonesia by providing information would be, you know, basically a dream come true for me. And finally, I'll just say that this opportunity to work with so many Indonesian researchers and additional Western researchers working on other focal groups in seeing them working in the field in their element, the techniques they use for their field work, which are often dramatically different than the techniques we use for herpetology, has just been a joy. I mean, it's really been fabulous. And so when I first sort of proposed this large collaborative effort, I feared that I would somehow during the process that I would upset all my Indonesian colleagues or just wear them out, that it would just become too frustrating for whatever reason to sort of conduct this large scope research. And I don't think that's happened, which I'm grateful for. And I'm just hoping that we'll be able to do even more of this kind of research in the future. So I'll just finish with another picture. This is from the last day at Iliomata on our last expedition in 2019. A lot of happy researchers who've just come off the mountain. Thank you very much for your attention.
Thank you very much, Pak Jim. I was mesmerized by your presentation. I forget to remind you about the time. My mistake.
Yeah, I'm sorry. I'm sure
it's okay. It's okay. It is indeed a very interesting presentations. I guess I believe there are many questions from the audience for our attendees, and I would like to remind you all, everybody, you are welcome to use the question and answer feature in Zoom or live chat in our YouTube channel to write your questions. As 80% of the attendees is from Indonesians, you can also write your questions in Bahasa and I will try to translate it. I hope Pak Rosikon and Pak Jim has more time and can directly answer the questions also. Please, I welcome you. Thank you very much again, Pak Jim. Now we are going to listen our next speaker, Dr. Kyria Bondimils. Ibu Kyria, are you there?
Yes, I'm right here. Can you hear me?
Yes, of course, clear. Okay, I will introduce Ibu Kyria. So Dr. Bondimils has been the curator of Puff Yeast Culture Collection in University of California since 2001. She is one of the executive board of the World Federation for Culture Collection and the steering committee for the U.S. Culture Collection Network. Her current research project includes studies of yeast, insect ecology, and screening yeast for commercial useful properties. Ibu Kyria, you have 25 minutes. Please, the time is yours. Thank you.
Yeah, thank you for that. I want to thank the meeting organizers for inviting me to present at this wonderful meeting. It's great to touch base with what's going on in Indonesia and revisit a project that I have completed and hopefully be able to set up some new collaborations. There's a lot going on here. So I'll be talking about a project that was funded, and the funding ran out several years ago, but some of the activities are continuing in various forms. So this was a project funded by a program called ICBG, and I'll describe that program. There's more information about this project at the UC Davis website, icbg.ucdavis.edu. So ICBG stands for International Cooperative Biodiversity Groups, and that is a program that was funded by the National Institutes of Health in the United States, but also was co-funded by 3 other agencies, the U.S. Department of Agriculture, the Department of Energy, and the National Science Foundation. And because the money came from these 4 different agencies, that shaped how our project was performed. Of course, our projects are often shaped by the source of the money. So the ICBG program was funded projects that performed by 14 different groups in 20 different countries. And they ranged from 1993. The last projects were funded in 2014. So this program no longer is funding new projects. The aims of these ICBG projects The most important aim, of course, is to foster partnerships between scientists in the United States and those in developing nations. These projects all performed various types of biodiversity surveys and used the biodiversity information to inform local conservation policies. And now, because some of the funding came from the US Department of Agriculture, a lot of these projects had either, and also funding from the Department of Energy, these projects either had an agriculture component or a bioenergy component. So, and that was due to the source of the funding. And also a major component of these projects, because it was funded by the National Institutes of Health, a major component of these projects was to screen various natural products to identify novel therapeutic activities. And these projects were required to screen for several specific activities. One was anti-cancer activity, also a disease affecting the host nation, and something also related to central nervous system disorders. So we had to accomplish a lot of many different tasks in this project, but they were all connected to biodiversity. And that was the reason that the UC Davis researchers that were the lead on this project chose to work with Indonesia. That was the most important reason, is the high biodiversity and the high endemism that is present in Indonesia in general and Sulawesi specifically, as you heard from the previous couple of talks. So we were searching for an area that had high biodiversity and endemism, had intact habitats, either primary or secondary forests, areas that were accessible, and also areas that were threatened by climate change or development or other factors. And that the reason for that is we wanted to be able to use the biodiversity data to help shape conservation efforts. Also, there were logistic reasons to work in Sulawesi and Indonesia in general. UC Davis researchers already had some existing collaborations with researchers at LIPI, which helped to jumpstart the project. And also researchers at LIPI already had some expertise in the areas that we needed. So this diagram shows the structure of this project and the 4 different teams that were all involved that interacted with each other. The team, what I'll call team number 1, was the biodiversity team. So the biodiversity researchers selected the field sites. They performed the surveys of plants, insects, and vertebrates. And then they also worked with the microbes team to identify plant and insect hosts of the microbes that we isolated. And the biodiversity team also prepared plant materials that was used for team 3, the therapeutic screening. And they also provided biodiversity data to team number 4 that was developing the conservation and outreach programs. And then team number 2, which I was the lead of, was focusing on microbes and isolating and identifying a broad range of microbes. We were looking at bacteria, yeasts, and filamentous fungi. So it was quite a diverse project. We expanded the culture collections at LIPI and also the Ministry of Forestry and at UC Davis. And we screened the microbes for the targets that were important to the funding agencies, which were bioenergy. And also we cultured some microbes that were used in the therapeutic screening. Then team number 3 was the therapeutic screening. So they took the extracts, they prepared extracts from the plant materials that were gathered by the biodiversity team. They prepared extracts of the microbes prepared by the microbe team, and they screened them for bioactives in the categories that are listed here: anti-cancer, things affecting the immune system, neurological activity, and then identified the active compounds when they found some hits. So all of these interacting components of this project contributed new knowledge that was used by the conservation and outreach team. And this team took that new knowledge and presented it to stakeholders, to decision makers, and helped with training locals in conservation, and also reaching out to decision makers to promote conservation, because this project demonstrated not just the biodiversity, but also the utility, the importance, the potential for new therapeutics, for new biofuels that are connected to and relies on the biodiversity in this region. So the scientific team was a pretty large team. We had representatives from 4 different biological collections at UC Davis: the yeast collection, insects, the herbarium, and the Museum of Wildlife and Fish Biology. And we worked with equivalents, our counterparts from collections both at LIPI and at the Ministry of Forestry. And then we also had the therapeutic screening work that was done at 3 different University of California campuses, partnering with the researchers at LIPI Chemistry, LIPI Biology, and ITB. So one of the most challenging parts of this project was before we could do any field work, we had to have our agreements in place. So this project started in 2009. Our first field trip was in 2009 in the summer. And it took over a year for these agreements to be signed and put in place. The first 2 agreements were the most important. That was the Memorandum of Understanding, or MOU, and the Material Transfer Agreement. And this, I want to point out, this was before the Nagoya Protocol. So we did not have the framework that was developed later that now helps guide these types of agreements. But we were able to put together agreements that were agreed by both sides to make sure that we had everyone had the same understanding of what was going to happen with this project, what was the fate of the materials, and so there was a lot of thought that went into those agreements. The aims of the biodiversity survey were to conduct surveys of plants, insects, and vertebrates. So we had people with a lot of different types of expertise out in the field together. And we worked at, I believe it was 5 different elevations. And our research site was the Makanga Mountains, which was mentioned in the previous talk, which was in Southeast Sulawesi. The biodiversity team developed and performed these surveys, and they also provided data for the conservation planning that was done by Team 4. This just shows a couple of the groups that were, as they were heading out on a couple of the expeditions, and some of the leadership of these teams. So this Team 1, the biodiversity team, was very successful especially in publications, but also in just the number and variety of specimens they collected. So there were about 6 expeditions to the Makanga mountain range, 1 and a couple to some other sites in Southeast Sulawesi. The specimens that were collected and preserved were deposited in the LIPI collection and the UC Davis collection. And there's over 1,000 plant specimens, roughly 1 million insect specimens, about 3,000 vertebrates, and also some vertebrate gut contents were preserved. And so in the course of this work, so a huge number of specimens were collected. There's quite a few publications. This showcases just some of the many publications that came out of this work. Some of these were directly by this team. Some of these publications were came out after this particular project was completed, but used the specimens that were isolated by this team. So this work continues. There's still work going on with describing new species. I was just told there's potentially a new species of owl, which apparently new species of birds, as you just heard, are not very common. They're pretty well known already, but there may be a new species of owl. And also there's work going on a treatise on the birds of Sulawesi in collaboration with somebody at UC Berkeley. I'll jump ahead to team number 3, the therapeutic screening. This team prepared extracts from the microbes and the plants that were prepared by the other teams and screened them for various activities against cancer, immune enhancing activity, neurological activity, and ability to combat tuberculosis. And this team identified the active compounds that were responsible for some of these activities. And in this picture, the top picture, those huge bins are full of dried plant material that was collected in the field by the botanists and laid out to dry out in the field and then brought into the lab. So that quite a lot of plant material was brought in for this therapeutic screening work. And this shows some of the team members that were involved in this work. Len Yeldenus is the tall guy in the center in that top photo. He retired shortly after this project finished in 2014. And so this is some researchers at LIPI Chemistry. And so there were also researchers at UC San Francisco, UC Davis, UC Santa Cruz. So it was quite a broad project. It was a lot of work to do all of this screening against all of the different targets that we were required to screen based on the requirements from the funding agency. All right, team 4 was the conservation outreach and education team, and their job was to spread the word about this project. They developed a lot of different workshops, training sessions. They also held information sessions for stakeholders, policymakers. So they were very good at taking all of this information developed by the other teams and sharing it with the general public and with the politicians. So this shows some of the people that were involved in the conservation and outreach portion. One of the most exciting developments out of this was that the Makonga mountain range has been declared, I believe, as a protected forest. And I would love to hear an update on what the progress is towards preserving this site that we've spent a lot of time documenting the biodiversity. Hopefully somebody at LIPI can fill in some of the gaps in my knowledge of what the status of this research site is. All right, now I'll focus on the microbe work. So I was the leader of the microbe work. I am the curator of the Pfaff Yeast Culture Collection at University of California, Davis. So my area of expertise is yeast. I have worked in the past with bacteria, with filamentous fungi. So I was able to refresh some of my knowledge about those types of organisms for this project. Our co-leaders were August and Atit from LIPI, and also Dr. Satepu, who became my postdoc. And also we had quite a few students, technicians from UC Davis and Florida and LIPI. And also one key member of this team was Nathan Schiff. He was an entomologist with USDA Forest Service in Mississippi. And he was very knowledgeable about wood feeding beetles and how to identify, how to find and how to identify certain beetle species. Now, why were we involving beetles in a microbe survey? Well, it turns out that 2 of our targets were connected to beetles. So because part of the funding for this program came from the Department of Energy, one of their targets that we were looking for was looking for new cellulase enzymes. So cellulases are enzymes that break down plant material. So that is one of the more costly steps in converting plant material, such as agricultural waste, forest residues, etc., converting that to biofuels, such as ethanol and other alcohols. And so one of our targets was to isolate new cellulase enzymes. If you want to find a new cellulase enzyme, the best place to look in nature is a habitat where cellulose is being degraded. And these wood-feeding beetle larvae spend most of their childhood eating wood. And specific species will spend months, weeks or months, just inside wood, chewing up the wood, and that is their main source of nutrition. So our theory was that these larvae would have microbes in their gut that had strong cellulases. So we were able to, we found hundreds of these various types of wood-feeding beetle larvae. We dissected them, plated the guts onto plates, and isolated the yeasts and the filamentous fungi, and also some bacteria. And this plate shows a simple plating assay. These blue speckles, the little blue particles, are xylan that's been conjugated to a dye. If we plate a microbe on the surface of that agar that can break down xylan, meaning it produces a xylanase, the blue dye will be released into the agar. So these solid blue agar portions of this plate means that microbe produced a lot of xylanase. So we isolated organisms from wood-feeding beetle larvae and decaying wood in order to isolate new cellulases. And also we targeted specific habitats also for the therapeutics. Actinobacteria are known to produce some of the classes of bioactives we were looking for the therapeutic screening. So we isolated actinobacteria and also filamentous fungi from soil and from leaf litter. Our third target that we were searching for was yeasts that have high amounts of lipids inside the cell. Before I started this project, before we visited the field, I did some reviewing of the literature to learn more about high oil yeasts. And it turns out that many of the 2,000 known yeast species, about 50 of them were known to be high in oil. And of those, many of those originated from either the surfaces of leaves or from soil. So we isolated yeasts from leaf surfaces and from soil. So we targeted specific habitats, and we had a rubric of how many of each type of organism that we wanted to isolate from each type of habitat to optimize our chances of finding specific organisms with specific properties that we were looking for. So we had 4 expeditions total, and we isolated and identified and preserved about 1,800 microbes. And of those, we had about 500 or 600 bacteria, just a few dozen actinobacteria, close to 500 yeasts, and about 400 filamentous fungi. Of the 500 yeast strains, we found about 60 of those are likely to be new species of yeasts. So this was a very rich and very novel type of survey. I found a lot of new species. So there's a lot of, I could spend the rest of my career collaborating with Indonesians, my counterparts, to publish these new species descriptions. That's a lot of new species. So these microbes were deposited in collections at 3 different organizations. And that is a fairly common practice for microbe collections. The same strain is deposited in multiple collections for long-term security reasons. So I described briefly some of the bioenergy work that we did. We found quite a few yeasts and filamentous fungi that were able to break down xylan. And then we used PCR to amplify and sequence the xylanase genes. And we discovered that there's some new homologs of these new classes of xylanase genes. So that was pretty exciting. We also were looking at high oil yeasts, as I mentioned. So I mentioned that some of the known species, some of the yeasts that were already previously known to be high in oil were isolated from leaf surfaces. But there's one extra twist I want to mention. So some of these yeasts that are high in oil, they form what's called ballistospores. So a ballistospore is a spore that that forms on top of a stalk, and then when it's mature, it shoots off, and it can shoot up to 1 centimeter. So there's this special method to isolate these yeasts. So the picture on the top shows how we did this. This is from a publication in the 1940s. It's not a new process, but it's a really cool one. So we would take the lid of a Petri plate, smear some Vaseline on it, put sections of leaf on that inside the lid, and then put the agar plate above it. If there's a yeast on that leaf surface that can shoot a spore onto the agar, it will form a colony on the agar. So it's a really simple way of all of the molds, bacteria, and yeast that you would find on a leaf surface. Only a few of those will be able to shoot a spore onto the agar. So it was a great way to select for the types of yeast that we were looking for. So we found quite a few strains of yeast that were high in oil. This photomicrograph shows a yeast cell that is stained with Nile red, which is a fluorescent dye that binds to oils. And these glowing balls are lipid bodies. So these yeasts are really high in oil. They can be up to 65% oil by dry weight. This oil in the future may be, if we can bring down the production cost, it may be a new oil to replace petroleum or vegetable oils for biofuels.
Ibu Kiria, sorry to remind you, you have 2 or 3 minutes left.
Okay, thank you. I'm almost done. Okay, so this project has completed. The money was spent long ago, but there is, especially in the biodiversity work, the vertebrate surveys, there's still some work, still some collaborations that have continued. There are quite a few microbes that are preserved in these 3 institutions. Like I mentioned, there's about 60 potential new yeast species that we could describe. Also, these microbes could be used by third parties for a lot of different research areas. I do not have any of these microbes in my public collection because our material transfer agreement does not allow me to It was a really positive experience, one of the most exciting and projects that I've done. And so of course, we required a lot of people from a lot of different areas. So I want to thank all of the folks in Indonesia that helped make this project possible, especially the permitting process. That was a ditto to, that is a challenging part of any of these projects. So I will be happy to answer any questions in the question session.
Okay, thank you very much, Ibu Kiria, for sharing your project experience. I guess we need to extend our research for future collaborations also. I assume that we will not have a question and answer because of the limitations of time. Our ministry will give a speech at 11 sharp. I hope he will come on time. So I welcome again Professor Rosikon, Professor McGuire, and Ibu Kiria to answer the questions in the Q&A feature at Zoom channel, and also the later maybe in the YouTube channel. For all attendees, I remind you to, if you need the certificate, then please fill the attendance form that already distributed by the committee. Okay, shall we move to our last speaker today, Dr. Campbell Oweb? Pak Kem, are you there?
Hi there. Yep.
Oh, thank you. Let me introduce you first. Campbell, Dr. Campbell Webb, or we usually call him Cam Webb, is currently a researcher at the University of Alaska. I can say that he's falling in love in Indonesia and pour out his passion of ecology, phylogenetic and molecular systematic, taxonomy, forest conservation, and biodiversity informatics of Indonesian flora since early 1990 and up to now. Pak Cam can fluently speak Bahasa, so everybody are welcome to give questions to him. And now still give some advice for Kalimantan healthcare project, healthcare and conservation project focused on the Gunung Palung National Park. Welcome, the time is yours. You have 25 minutes. I will remind you 3 minutes before the time off. Thank you.
Okay, thanks. Can you see my screen?
Yes,
Right.
yes.
Oke, terima kasih Bu Ruli. Dan selamat pagi kepada semua peserta yang di Indonesia dari Alaska di sini. Terima kasih juga ke Bu Ayu dan Kapus P2B Bu Ati atas undangan bicara hari ini. Sebetulnya saya senang sekali untuk terima undangan tersebut, oleh karena saya memang sangat rindu Indonesia. Dan webinar ini merupakan kesempatan bertemu teman-teman saya dan juga ingat banyak hal tentang Indonesia hari ini. Tumbuhannya, mitra penelitian, maupun masakan, dan juga suhunya, oleh karena di sini di Alaska sering agak lebih dingin dari Indonesia. Nah, sekarang baik saya ganti bahasa. Jarang saya bisa memakai bahasa Indonesia di sini dan ya kemungkinan saya makin agak kurang lancar sekarang. So I'll switch to English, but please do send any questions in Indonesian if you'd like to. So let's talk about the magnificent flora of Indonesia. Many species, as Pat Rosikon already mentioned. And it's interesting and useful to start right at the beginning and go back in history and think about why there are so many species in Indonesia and so many plant species. And one of the reasons is that the area that is now Indonesia has been more or less in the same place in a tropical warm zone for many tens of millions of years. And different groups of plants have arrived in different waves over time. The first wave back in the late Cretaceous, and then a second wave when India crashed into Asia at about 45 million years ago. And then recently, as Australia has approached Asia, and for many of the islands of Indonesia, there's been Australian plants arriving too. And then you add to that the many islands that break up species, and you get a very rich background for speciation and for species. You add to that contemporary climatic variation, as you all know, in the West and in the north of Indonesia, it's a moist climate, and then in the southeast and much of the south is a much drier climate. there's also many mountains. So we have wet forest and dry forest and montane forest, grasslands and mangroves. So all of this adds together to create a very rich number of species. The estimates are around 30,000, and that's based on estimates of the larger Malaysia, which includes parts of Malaysia and the whole island of New Guinea, of about 41,000. But these are still estimates. There are many species still to find, and many that have not yet been documented. High endemism, estimates of about 46% of plant species on Papua are endemic. They only occur on the island of New Guinea. It has the largest forest, tropical forest, the tallest tropical forest in the world, really quite wonderful. On the island of Borneo, some dipterocarps will reach nearly 80 meters. So for botanists, it's a total dream. And for the people of Indonesia, it's a great resource. But we're still a long way from having a checklist. Flora Malaysiana, which is the flora documentation project started in the middle of the last century, is still working through, and many, in fact, most of the big families still have yet to be addressed. And so people have estimated, Marco Roos has estimated that it probably, at the current pace, we're looking at more than 100 years to finish the documentation. I always like to throw in here that lists of species are not necessarily needed for forest protection, but they are definitely useful. So my contribution to the study of the flora is sort of 25 years of mainly making forest plots. Many of you have done that as students and as researchers. And I've been fortunate to be able to make permanent plots in Borneo at 5 different locations: Flores, Sulawesi, Seram, and Papua. And I should acknowledge the funding here came often from the National Science Foundation, the Annal Arboretum of Harvard University, the National Geographic Society, and all the permissions that were needed were much appreciated, coming from Minristek, LIPI, Ministry of Forestry, and KASDA. And I'd also like to acknowledge here my most recent co-PI on an Indonesian NSF grant, Sarah Matthews. So we found over the years a bunch of different things. I'm just going to highlight two things here. There's a lot of fun things to get through, and not a great deal of time. Way back when I started, I was mainly focused on forest ecology and on the regeneration of seedlings under the larger trees. And at Gunung Palung in West Kalimantan, we found a density-dependent pattern in the mortality of seedlings. So the more abundant taxa, the more abundant species, actually had higher mortality of seedlings or lower survival. And that leads to a pattern of a population turnover that can help stabilize biodiversity. And this is a pattern that's been found elsewhere, but some other forests also don't find it. So it was an interesting thing to find here. More recently, especially when I was able to travel to some other different islands, we've been studying forest floristics and also forest phyllofloristics with phylogenetic information. And here's a little graphic of the composition of the top 10 families across different sites on Borneo, Flores, Seram, Sulawesi, and Waigeo in Papua. And you can see that overall, there's a lot of sharing of the different families between islands, but the abundant families and definitely the dominant species are very different on different islands. And this is a interesting phenomenon. Just to note that Malaysia does have fairly similar forests. We don't have a Wallace's line for plants, but at the same time, there's great variation in the details of the common species at each site. When I look back and think what will be most useful to people who are working on the flora, it's probably going to be not so much these patterns, but it's more the specimens that we have collected, and more recently, the great number of photographs and the other metadata that we have collected for these different plants at these different sites. So we, in the most recent study across these different islands, we took over 30,000 images, carefully documenting the fresh material at each site. We have DNA barcodes for most of the taxa. We have wood density estimates, specific leaf area, and also some ethnobotanical data. And all of this has become, is wrapped up, and I would say fairly well organized, and I've become very interested and engaged in the area of biodiversity informatics. And that's how we can share and standardize data so that other people can use it. So I hope these data will be useful to taxonomists working on different taxa in the future. Now, I'm here in Alaska now, and actually one of the things I'm doing is working on a flora of Alaska. But Alaska is a very different situation taxonomically than Indonesia. So for instance, Alaska is 2.3 times the area of Borneo Island, but we only have 25 tree species, as opposed to somewhere near 5,000 tree species for Borneo. And most of the taxonomy up here is done. There are still new species being found, but very rarely. And people have looked carefully at most of the specimens. So this is a very different situation from the vast number of taxa and the unnamed specimens that exist in herbaria in Indonesia. And it's likely that many Indonesian plant taxa simply will not get worked on in the traditional way that has been possible for less diverse areas. So I think there's a pressing need for a new model of taxonomy, not that will be better, but that just will be faster. And we're all working towards this. None of us really know how this is going to work. But I think elements of it will be that collections, we're not going to make a lot of new collections anymore. So we have to mine the herbaria for the specimens that exist there, and digitizing them is really important. And then also we can use the public who now are armed with smartphones can make fantastic observations. And we call these citizen observations. And this is also really important. Much of the work of taxonomy is matching different plants together in piles and saying this is a taxon, this is a taxon. And it's likely that we need to rely to some extent now on algorithm-based similarity measures, for instance, images and DNA data. And then we can still link those classes back if we have the right informatics approaches to literature in the past. And if any of you haven't discovered the Biodiversity Heritage Library, it's a tremendous resource for taxonomy. What this means, though, is that data curation becomes almost as vital as specimen curation. And just as an example, the AI, artificial intelligence recognition of taxa is just improving all the time. And there's a fantastic paper on the identification of herbarium specimens by Carranza Rojos just a few years ago. What this means, I think, is that there needs to be investment in biodiversity informatics for those interested in the field of taxonomy. And just looking through some of the LIPI initiatives that are already existing, there are some great programs. There's IBIS, the Indonesian Biodiversity Information System, INABIF, the Indonesian Biodiversity Information Facility, and a data sharing and publication sharing portal called the Dataverse, which I was just looking at. It's fantastic. I have to admit, though, that one of the greatest resources that would help taxonomists, plant taxonomists, is the complete digitization and databasing of the Bogo herbarium. And I know the history of this, and I just want to encourage anyone working on it that this is, when it comes online and can be shared, this will be a tremendous resource for the world and for Indonesian taxonomists. I've also heard that there's a DNA barcoding database in the works, and we will be the first customers to deposit our data there. I've worked through the programs that the last project with some fantastic young programmers, and so I know the skills are there throughout Indonesia in software design, and bringing the software engineers together with the taxonomists, I think is a very fertile area. And one of the projects which I just found out about, which I'm very excited to see, is the biodiversity program. And this is an effort to take the many university scriptsies, many of which look at rare sites and have collected important biodiversity information, bring them together with species lists and share them in a common portal. And that's a great project which I haven't seen anywhere else in the world. So that's wonderful. At the same time, it's really important that we don't go all the way to the tech, and that basic taxonomy training and understanding what taxa are is just as fundamental. And so young students, I think it would be great to bring these two areas together to help develop skills for taxonomy in Indonesia. Now, when I was asked to speak today, I was also asked to reflect a little bit on the process of collaboration with Indonesian institutions and scientists. So here goes. The first step, of course, is getting research permissions, and has already been discussed by Jimmy. This does take some time and patience for researchers. I was lucky to be able to live for extended periods in Indonesia, so I had a lot more time. But it is a lengthy process. But I can, at the same time, see the structural reasons for the many letters and office visits that exist. And I won't comment any more on this. I also can't really speak to the most recent issues, since I haven't requested a research permit in 5 years now. And in truth, I don't even really want to complain about that, because I was never once denied a research permit and was able to visit Indonesia again and again during 25 years. And I was able to live more or less continuously as a researcher there for 10 years, from 2005 to 2015. And indeed, the process of obtaining permits has been remarkably stable, even during the transition from LIPI managing permits to RISTEC managing permits. And I do miss the visits to the strange and wonderful Widyagraha building on the bottom left there, with its strange interior and special smell of Kretek cigarettes. And thanks to Pak Ruben Siletonga, who was the last staff member I dealt with at the LIPI Kerjasama office there in 2007. Stepping back, it's easy to see that Indonesian authorities are always trying to balance the needs of the nation with benefits of an open scientific process. On the one hand are the sovereign rights to control biodiversity and biodiversity data for the benefits of the people of Indonesia. And also there are numerous experiences of having been taken advantage of. Now, on the other hand, though, there are scientific opportunities to learn, teach, and share, and science works best, without a doubt, when information and materials can flow easily. But getting this balance is not easy for any country or for any institution. And as I think back, I'm very aware now of the various mistakes that I have made, and particularly of the tolerance of my counterparts and the permit issuing authorities. My reports were often late and a little bit slim. I'm still working on some papers that I owe all the parties involved. And my plans sometimes changed and were not in accord with my proposal. So I'm personally very grateful for the generous benefit of the doubt that my counterparts offered me over the years. And I do apologize for the problems I might have caused. All I can do is urge Indonesian scientific authorities to keep this openness going into the future, and to offer and expect goodwill from researchers. One of the things that was very helpful towards the end was when I came into a position where I could negotiate and work on MOUs, and these have after having an MOU, many of the opportunities for misunderstanding are reduced. Now, thinking about this issue of collaboration, and as I was thinking back on my time in Indonesia, there's no question that the social dimension of the research, the people who I got to work with, were almost as important, if not more important, than studying the plants. And I've made many friends over the years, and there are many people whose contributions I absolutely could not have done without. And so I thought I would actually put together a list of names in the different classes of people who've helped me so much over the years. And this is it's a little risky because I'm sure I'm going to forget someone. And if you are forgotten on the list, on this list, I do ask you to forgive me. But I just wanted to indicate the different kinds of people who have made my time in Indonesia so important. So I thought I'd start with those who I might call my senior, people such as Pak Kuswata and Pak Dedi Donedi. Pat Dedi and I worked together on putting together a large conference with a large other staff as well, the ATBC, Association of Tropical Biology and Conservation Conference in 2010. And that was a very testing but also wonderful experience in the end. Other friends, Pa Ishmael, who's helped me learn the plants of Indonesia for so many years, Pa Uwai, Pa Eko, and then many other people. And there are going to be so many people I forget, but there are people who I've known when I arrived as a 21-year-old. These were the people I looked up to. And then working over the years with so many staff at LIPI, at P2 Bay, just a huge long list here, especially at the herbarium. That became a second home to me, and I miss it. I miss it a lot. I miss the collections. I miss the people. I miss the canteen. This last project, I had particular counterparts, and they've been so helpful, and they got through. They solved so many problems, and I've got to mention Pat Arif Hidayat and Pat Teguh Triyono, who were the counterparts for this last project we had, and then many other people who helped with aspects. And I'm running out of time, so I'm not going to name them all. I also had collaborations with Universitas Gunadharma, with Pat Madewiyana, and they helped develop our informatics side, and I worked with some very gifted young programmers there. Two people I have to name are my friends and field assistants for many years, Pak Andra Setiawan and Pak Achun Herianto. And they went to all the different sites with me and managed many of the logistics and took most of the photos that I mentioned before. At each site, we had local institutional counterparts who would come along, mainly from KSDR, but some other local universities. And without the tremendous number of village members, none of this would have been possible, and especially for the tree climbers who willingly and apparently enjoyably climb so many tall trees to help us get our specimens. I also noticed that over the years I've been able to give talks at a lot of universities, and I added them up. There's quite a lot here, and these students have always inspired me over the years. And then finally, a group of people who I've come to know recently, just fairly recently, but these are the amateur botanists. And in one case in particular is amazing. There's a Facebook group called Plants Community, started by some amateur botanists in and now professional botanists in Kalimantan Barat. And they have uploaded over 2,500 images, high quality images from different taxa, and they offer nearly instantaneous determinations, and they're extremely skilled, and it's been a wonder to learn them and to work with them. And so coming back out of the people who have been so important, I just want to end by saying that this role of citizen science is becoming more and more vital and more capable. Looking back nearly 15 years ago, there was an Indonesian citizen science group called FOBI. Unfortunately, the website seems to be down, but that was an inspiration. Then, as I've mentioned, there's the Facebook Plants Community, and then the #1 platform that exists right now in the world. There were many, but there's one that's just gone ahead, is iNaturalist. And just before this presentation, I just looked, and there are over 170,000 observations of organisms from over 13,000 species in Indonesia. And this is going up all the time, and it's over 5,000 observers. And the fantastic thing about iNaturalist is it feeds data straight into GBIF, and where it can be mixed with the specimens in museums and create a much richer picture of the distribution of organisms. So just a review and just to leave you with a few parting thoughts. It would be wonderful, it will be wonderful to have a complete flora of Indonesia. How do we get there? Well, I think it's really important that we all still keep collaborating nationally and internationally. And that depends so much on sharing and on communication and goodwill. And I have received so much from my Indonesian collaborators. Flora Malaysiana is a vital project. It's the backbone for a flora of Indonesia. It would be extracted from Flora Malaysiana. So that's so important to keep going. And then building informatics capacity is going to be important as technology plays a larger role, including the citizen science element. How we do this new taxonomy, we don't know, nobody knows in the world, but I think the ground is rich for Indonesians to play an important part in that. So thank you so much for listening. And again, thank you to all my friends in Indonesia. Terima kasih.
Thank you, Pak Kem, for managing the time the time. And also thank you very much for your very interesting presentations on Indonesian flora and also the use of informatics in the field of taxonomy and ecology. It is very interesting. Well, thank you again. Listening to all of our speakers, we can agree together that there's still many work to be done to reveal more information regarding our biodiversity and its functions. And also there are homework also for our Ministry of Respect, right? Regarding the perizinan. Okay, I'm really sorry for all our audience that we don't have enough time for the question and answer sessions. Because of our minister is already here. Selamat datang, Pak Menteri. Please allow me to close this session first before we start for the keynote speaker. Regarding that, I hope our speaker can spend more time to respond directly to our attendees' questions in the Q&A feature in the Zoom channel. Thank you very much for all of our speakers, Professor Rosikon, Professor Maguire, Dr. Kiria, Dr. Kem. Thank you for sharing your knowledge and enlightening us about Indonesian biodiversity. Thank you also for all of our audience who already stay until the end of these sessions. Do not forget that we still have 2 more sessions today after the keynote speaker. Be healthy, stay safe, and spread positive vibes. I'll give the mic to Renata. Thank you.
Thank you, Dr. Liana Susanti and the speakers of the session 1 for the informative session. And I would like to say welcome to Professor Bambang Permadi Sumantri Brojonegoro, Minister of the Ministry of Science and Technology, National Research and Innovation Agency. Welcome to the international webinar on Indonesian biodiversity, mainstreaming biodiversity conservation, bioprospection, and bioeconomy for sustainable livelihood. Wednesday, September 16, 2020. Before we step up to the next agenda, let me read the CV of Professor Bambang. Professor Bambang Brojonegoro started his career as an academia in Universitas Indonesia as a lecturer, researcher, faculty dean. The opportunities to contribute to this country as the Minister of Research and Technology, Minister of National Development Planning, and Minister of Finance have established Professor Brojonegoro's career firmly in integrating Indonesian research, technology, innovation, development planning, financing, and economic stabilization. Ladies and gentlemen, next agenda, keynote speech by the Minister of Research and Technology, National Research and Innovation Agency, Professor Bambang Permadi Sumantri Brojonegoro, PhD. The floor is yours.
Thank you, Renata, for introduction. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to all of you, Pak Handoko and all the LIPI management, researchers, staffs, and also distinguished speakers from both from Indonesia as well as from abroad. I believe there are some from USA, and distinguished participants, ladies and gentlemen. First of all, I would like to thank LIPI for conducting this international webinar on biodiversity as part of Hari Kebangkitan Teknologi Nasional, or National Technology Day, that was already held in August 10. So talking about biodiversity, if you can, I can share the presentation. We know that biodiversity is part of our life. There is no doubt about that, because biodiversity will play a critical role, like in food, providing a source of food, source of energy, source of clean water, as well as providing quality environment, spiritual fulfillment, as well as preservation of cultural heritage, mental and physical well-being. So basically the whole aspect of our life actually related to the biodiversity. So clearly biodiversity is an asset for long-term sustainable development. Indonesia is very well known for its biodiversity. On the next page. In terms of terrestrial biodiversity, we are ranked second after Brazil. But when we include the marine biodiversity, Indonesia clearly at the top. So it means for my colleagues at LIPI, our next responsibility, our next job would be how to study, learn, and explore this marine biodiversity. And don't forget, we have Banda Sea as one of the best laboratory for marine diversity. And LIPI already has the special research center for ocean, for deep sea ocean at Banda, and we hope we can optimize that center. But in terms of terrestrial biodiversity, as we can see, Indonesia is a home for more than 10% of world mammals, 8% reptile, and 50% of insects. And I think that could be more, because I believe there are still some more species not yet discovered or founded in Indonesia. So we have now, basically we are blessed with our richness in biodiversity. And of course, we have to use the biodiversity sustainably. On the next page, one of the important things for us is bioproduct through bioprospection. And one of the example is immunomodulator and also traditional herbal. I believe Indonesia has a potential to be one of the main players in pharmaceutical industry in the future. Why we have that potential? Because we have this biodiversity. We have the materials for producing, traditional medicine as well as immunomodulator. We remember that China gains the reputation in medicine. In the past, we used to call it Chinese medicine or obat Cina, simply because they can really optimize their potential in biodiversity as well, combined with the technology. So at the end, they become one of the main players in pharmaceutical industry. Now, Indonesia still depends so much on the import of pharmaceutical material, especially for material for the medicine, because we don't have strong chemical industry to support this kind of material for the medicine. But we have this biodiversity that could be labeled as the medicine of the future. In fact, if we include marine biodiversity, the potential is even bigger, because many people already think or predict that marine biodiversity will also be the medicine of the future. So in order to really optimize this medicine of the future from our biodiversity and make the terminology Indonesian medicine as a globalized world, then of course we need to do more research and development on this bioproduct, including traditional herbal, traditional medicine, yeah, and also the phytopharmaca and immunomodulator. There are many potential can be created from our biodiversity. Aside from that, we also have the potential of bioeconomy in ecosystem services and wildlife trade. So we already witnessed that 40 million Indonesians, so quite substantially, living in rural areas rely on biodiversity for their subsistence needs. Of course, mostly for food and as well as energy. So it means we agree that biodiversity itself is very valuable. And of course, it becomes more and more valuable if we can create value added from that biodiversity. However, there are future challenges for our biodiversity. First, of course, there is potential of biodiversity loss that will have direct and indirect impact on our life, as well as less than full disclosure of our potential of biodiversity. As I mentioned, we try very hard, especially our colleagues in LIPI researchers, try to find as much as possible our potential of biodiversity. But of course, it will take time, it will take a lot of energy, it will take a lot of budget as well. So, you know, the progress must be not as fast as we expect. But more importantly, we need to keep working on disclosing more potential of our biodiversity. So at the end, when we create the value added will be even bigger when we discover more of our potential in biodiversity. So public awareness and cooperation from every aspect of stakeholder is necessary in order to do conservation as well as sustainable bioprospection and bioeconomy. I believe mainstreaming in biodiversity becomes very important. So far, as we are aware, biodiversity is not yet part of our mainstream, both in economic development as well as in the public policy. So our challenge now, our task, and we can learn also from other countries' experience, how can we mainstream the biodiversity, both in economic development programs, as well as in public policy mainstream. I believe this is quite challenging, but I believe with the awareness of richness of our biodiversity, people will understand more. People will be more aware about the importance of biodiversity in the policy as well as in economic development program. And the objective would be to reduce negative impacts of human activity by acknowledging the contribution of biodiversity to human well-being. I believe this is kind of campaign that we need to carry out in order to gain public support in biodiversity. So for example, we always have the issue of the forest fire, you know, every year, especially during the dry season, government trying to deploy as much as possible the forces in order to reduce the forest fire and the impact of the forest fire. But I think we need to analyze more why forest fire happens. There could be some natural cause of the forest fire, but what I want to focus is the forest fire caused by the human activities. People basically need land, need a piece of land, some hectares of land, in order to do some economic activities, especially plantation or agriculture. So we need to do a campaign. How can, for example, our tropical forests be beneficial for people who live in the surrounding area? I think that will be more important than just saying that you cannot, you know, destroy these forests, tropical forests, because of the biodiversity reason, because of many other reasons. I think the best campaign would be how we can translate the benefit of tropical forests to the people in the surrounding area. So it means they can still do the economic activities without destroying or without disturbing the tropical forest. That would be one important campaign that we need to carry out. And I think colleagues of LIPI researchers can give inputs on how we can do this kind of campaign, especially to attract the awareness of the people who live around the tropical forest area. So again, mainstreaming of biodiversity will be a collective effort to integrate actions related to conservation and sustainable biodiversity at every stage of policy, plan, program, and project cycle. So it's quite clear that mainstreaming will have to start even from the policy level up to the action level. And that has to be understood by every part of the community. Again, we need to focus our campaign on people who live surrounding tropical forests or any other areas that has the richness of our biodiversity. So, accommodative and conducive policies in state level are prerequisite for biodiversity mainstreaming in a country. And adoption on Convention on Biological Diversity strategic plans, its IT target and SDG into national planning for biodiversity and sustainable development are the essential first step in realizing mainstreaming. We have to start really from the upstream, and the upstream would be long-term development goals. And in this case, globally, we have the SDGs. And SDGs can be a platform, to introduce the importance and the benefit of biodiversity for human life. So, what happened with our strategy and action plan post-2020? Integrating biodiversity into state development model, as I mentioned, can be a challenging task. Some of the reason could come from inside the bureaucracy itself, as we understand, and also people at LIPI who already very well informed about the bureaucratic structure of our government. Institutional barrier always there. Whenever we have government structure, organization, automatically it will create kind of institutional barrier. And sometimes institutional barrier can be really a challenging or a constraint for mainstreaming the biodiversity. And then usually it can be worsened by the poor data or lack of data on the status of our national biodiversity. If people don't understand about the richness of and variety of our biodiversity, usually they are not attracted to really involve in the policies regarding the biodiversity itself. And another part of a challenge within government Indonesia is inadequate communication to support mainstreaming in cross-sectoral and national plans. So this kind of bureaucratic constraint has to be anticipated whenever we are trying to mainstream. So the best way to mainstream, of course, you need to go to the 30 years development plan or the long-term development plan, and then translate it into 5 years development plan. Although it's already in the 5 years development plan, there is no guarantee. Since I'm former Minister of National Development Planning, there is no kind of assurance or guarantee that you already have program for 5 years, it will be fully implemented in each budget year. So you have to anticipate some, sometimes less than necessary budget available to do the mainstreaming of biodiversity itself. So the need of strong leadership who can initiate the mainstreaming, pace the infrastructure accommodation, and provides multitude of motivation and incentive that follow or sustain the effort is paramount. So again, strong leadership. But of course, we understand in a democratic situation like Indonesia, the word strong leadership sometimes can be misleading. So we need actually not only strong leadership, but also solid management, solid government management. Because at the end of the day, if the bureaucracy left as it is, then it will be hard to make sure biodiversity is part of our economic development program or the public policy issue. So we need to keep active in pursuing the agenda of biodiversity into mainstream of development, and at the same time, we have solid management to implement that kind of program in the field. So in the long run, successful mainstreaming endeavor will be reflected in the collective paradigm shift and better attitude toward biodiversity, as well as biodiversity status and human well-being. Well, in this case, we can also learn from developed country. I think we have some distinguished panelists from US, for example. Since I spent almost 6 years studying in US for my graduate study, I can see the difference, for example, between US people attitude and Indonesian people attitude towards environment and towards biodiversity. Of course, the difference could come from different economic status. For the country like US, in which the GDP per capita is already high and then categorized as a developed country or negara maju, of course, the people who already so-called settle with their life, they don't feel that they are in the difficult economic situation, they can respect more, they can give more appreciation to their biodiversity. And there will be effort to secure or to maintain, to make sure the sustainability of the biodiversity itself. Of course, when we are talking about Indonesia, Indonesia was still upper middle income country. In fact, in the very beginning of upper middle income country, with GDP per capita, I think currently around $4,000 per year, then of course, the needs for economic life is still very much needed. So people still look for economic opportunities before thinking about other things. So with this kind of community, of course, we cannot expect people will give more attention to the issue of biodiversity, to the issue of, let's say, maintaining the environment. Instead, they will still look at biodiversity or environment or natural resources as a way, to fulfill their economic needs or their income needs. So with this kind of difference, then of course, our job as a government is to make sure people can maintain the balance between their economic needs and the needs to preserve the environment and the needs to preserve the biodiversity. So that's the government job. In the case of, as I mentioned, the developed country like US or countries in the Western Europe, for example, then government doesn't have to do much. In fact, they just maintain the property, the property related to the environment and biodiversity. And the people already have the awareness about the importance of biodiversity itself. So again, this is very critical, but we need to, gradually, create the awareness to the people, since we have the aspiration, let's say in 2045, 100 years of our independence, Indonesia can be a developed country with a relatively high income per capita, because we will graduate from upper middle income to the high income country, and hopefully by that time, people already have strong awareness about the importance of biodiversity and the appreciation that biodiversity is part of their life and biodiversity can give the life to the Indonesians in general. I think that will be my short keynote, and of course, I hope that the one-day webinar on Indonesian biodiversity will not only create the information or knowledge about biodiversity, but more importantly, how we should work on our biodiversity richness for the future. And I think talking about the Indonesian medicine, the phytopharmaca is one of the great things. The other one would be the energy. I believe we need also to look at our biodiversity as an alternative source of renewable energy, aside from other type of renewable energy that we already know very well. And one last thing, I really appreciate researchers at LIPI that now trying to focus on the circular economy, because now some of them trying to trying to utilize the waste from everything into something else, into source of energy like biomass, or into, for example, another product like paper, for example. So instead of cutting the trees for the papers, researchers at LIPI can use the waste to produce paper that has similar quality with the paper from the woods. So I believe this is my expectation from LIPI active participation in refilling our biodiversity as well as optimizing our biodiversity. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you so much, Prof. Bambang, for the keynotes and joining us to today's webinar. Next, I would like to invite Chairman of Indonesian Institute of Sciences, LIPI, Dr. Laksana Trihandoko, some lines as the appreciation of the attendance to our minister today and some conclusion about the Indonesian biodiversity. Pak Handoko, waktu dan tempat kami silahkan. Yes, Pak Handoko, floor is yours. Pak Handoko, yeah, I would like to invite Chairman of LIPI, Dr. Laksana Trihandoko, to give some lines as the appreciation to our minister today and some conclusion maybe about the Indonesian biodiversity.
Yeah,
to Pak Handoko, floor is yours.
Yes, thank you very much. On behalf of the Indonesian Institute of Sciences, LIPI, I would like to thank to Professor Dr. Bambang Brojonegoro for a very inspiring talk. And we know that the biodiversity and how we conserve our biodiversity, how we utilize and our biodiversity through bioprospection is very, very important for our future and also for the development of our country. Terima kasih Pak Bambang, Pak Menristek. I pass back the floor to Mbak Renata. Sorry.
Thank you so much once again to Prof. Bambang Brojonegoro and to Dr. Laksana Trihandoko for the opening. We hope that our webinar will be fruitful and going well. Thank you once again. Thank you. Ladies and gentlemen, before
Terima kasih Pak Menteri.
Ya, terima kasih Pak Noko.
Yeah.
Ladies and gentlemen, before continuing the next session, we will have an interactive quiz. There will be merchandises from LIPI and books from LIPI Press for 10 lucky participants. You ready? Mr. Amal, please lead the session. Ya, sekali lagi kami sampaikan terima kasih kepada Pak Menteri Ristek Brin atas bergabungnya pada hari ini dan memberikan sesi keynote speaker. Then, to the next session, kita akan mengadakan interaktif quiz kepada Mas Akmal. Silahkan untuk memimpin sesinya.
Baik, terima kasih Mbak Renata. Hello, ladies and gentlemen. Now, time to break the first session. We will relax for a few minutes to play the quiz. The committee will display the procedures for playing the quiz. All participants, please click the quiz link in the Zoom chat feature. Once again, all attendees, please click the quiz link in the Zoom chat to go to the game arena. Afterwards, please write down the names of the players according to the rules listed in the Zoom chat. Once again, please write down the names of the players according to the rules listed in the Zoom chat. Please pay attention. I will read the rules of the game. Procedures for playing quiz. First, this quiz is only followed by WEBINUSA participants on the Zoom channel. Second, the committees are not allowed to take the quiz. Third, before the quiz begins, all participants must provide the identity of the quiz player according to the rules. If there are participants who do not comply with the rules, they will be disqualified. Fourth, the format for the identity of the participation in this quiz is as follows. Name, 1 or 2 syllables registered in the webinar registration, dash, origin of the institution. The name is up to 25 characters long. First name or second name could be abbreviated. Look at the examples. M. Nikmatullah, dash, LIPI.
Ladies and gentlemen, we are very sorry for their technical problem here. about the signal, I just want to reminder that we have the next session at 12:30. The second session about mainstreaming bioprospection, harnessing untapped biodiversity for novel bioproduct, challenge and success story, that will be led by Dr. Khairul Ilmi Setiawan from LIPI. So for participants who wish to break, we have time until 12:30, and please join again to this webinar, and then we will have the second session. I think Mr. Akmal has already joined again to the Zoom meeting. Mr. Akmal, can you hear me?
Ya, bisa. Oke, sudah bisa.
Okay. Mr. Akmal, please, time is yours.
Mohon maaf, ada trouble sedikit. Saya akan lanjutkan kembali membacakan peraturan kuisnya. And the last, the quiz will be guided by the MC, and the time for the quiz to end is up to the MC's decision. From all participants who take the quiz, we will choose 5 winners. After getting the winners, the MC will close the quiz by giving a signal. And the following are the books giveaway for all the winners. May you be among the lucky ones to get the prize for the book. Okay, now I want to check the number of participants who have entered. Please. The committee maybe can share the quiz room, please. Udah share screen belum? Coba share screen. Untuk para peserta mungkin bisa dimasukkan kodenya. Saya akan sebutkan 1614339. 1614339. I would reply 161439. For code of the game 161439. Okay. 20 participants. We are still waiting for the other participants. Please join the researchers, the educators, teachers, undergraduate students. Please join graduate students. Please click the link, please. Please click the link of the quiz on the chat Zoom. Mungkin panitia bisa ulang kembali memberikan linknya di Zoom chat. Untuk aturan tata cara nama pemain, memasukkan nama pemain, nama, strip, instansi. Jangan sampai salah. Nama, strip, instansi. 1 minute again. 314 attendees now. If I say go, the committee can start pressing the start button. Oke, peserta terus bertambah. Please join, ladies and gentlemen, for this quiz. Okay. Maybe we are waiting up to 400 participants. Please join with the correct username or player name. $364 now. Sebentar lagi kita akan memulai kuis. $381. Okay. One again. Okay, let's begin. Ready, set, go. Okay, the quiz game has started. All the quiz participants, please answer 10 questions quickly and accurately, not only fast, but also precise. So, who is in the first place now? Let's see. Fajrin again, wow, from LIPI. Okay, Akang Fajrin, Mr. Fajrin from LIPI. In the second position, oh, Mr. Ganjar Cahyadi, Mas Ganjar Cahyadi from SITH, ITB. Can Mr. Fajrin stay his position? Let's see. Mantap. Mr. Fajrin stay in first position. Wow. It seems Mr. Ganjar has slumped. I just see the first position. The second, Isa dari LIPI juga. the first, Tania A dari Mas Ganjar, back to the second position. Let's see. Fajrin masih mempertahankan singgasananya. It's great. It's so great, incredible. So Fajrin. Mas Oni in 5th position. Mas Oni Marwayana from LIPI too. Oh, now in 6th position. Mungkin Mas Ganjar menyalip Pak Fajrin. Okay. Now the first position is still Mr. Fajrin, second Mr. Ganjar, and third Mas Hendricks from here. Kalau sudah didapatkan 10 menang, all right, we get the players of, we get the winners, I think. Oh, congratulations. Posisi pertama akhirnya jatuh pada Muhammad Ihya. Nomor 2 Fajrin. And third Mr. Ganjar. We will 5 winners. Congratulations for all the winners. Congratulations for all the winners. Each winner will get a LIPI merchandise and 1 book of LIPI Press. The organizers please take screenshots as proof of the names of the winners. We shall contact the winners. Once again, congratulations to the winners. Jangan lupa nanti ada kuis sesi selanjutnya, kira-kira jam 2:30 setelah sesi kedua. So stay tuned. Congratulations for all the winners. Thank you. Mrs. Renata, time is yours.
Okay, thank you, Mr. Okay, thank you, Mr. Akmal, and congratulations for the lucky participants. Dan seperti yang tadi dibilang, nanti akan ada sesi kedua untuk kuisnya, jadi tetap di webinar internasional ini. Ladies and gentlemen, once again, I want to remind you that we will start the next session, Mainstreaming Bioprospection, Harnessing Untapped Biodiversity for Novel Bioproduct, Challenge and Success Story at 12:30. As the moderator for the second session will be Dr. Khairul Himistiawan from LIPI. So stay tuned. Thank you and see ya. Now we will play a movie created by LIPI entitled The Zoologist. Please enjoy.
Terus terang LIPI itu aku nggak tahu apa-apa kecuali dulu nonton kuis Galileo. Artinya setelah aku masuk di LIPI berubah gitu, paradigma peneliti yang kaku dan lain-lain itu ternyata nggak kayak gitu waktu di LIPI.
Ketika kita menemukan sesuatu yang katakanlah aneh, langka, jenis baru, tentunya itu suatu hal yang sangat menarik. Biasanya kalau sebelum saya berangkat itu saya harus minta doa restu atau izin dari bapak atau ibu atau orang tua kita atau orang yang kita cintai di rumah.
Tujuan kamu adalah mencari ilmu, dan kalau kamu mempunyai ilmu, manfaatkan pada orang lain. Pesan kami hanya seperti itu.
Apa? Coba dibaca. Ini.
Amfibi dan reptil.
Amfibi dan reptil.
Awalnya sih terus terang LIPI itu aku nggak tahu apa-apa kecuali dulu nonton kuis Galileo. Jadi kan ada terima kasih LIPI. Itu aja sebenarnya aku taunya dari situ. Atau waktu pas kuliah biologi itu masalah CITES. LIPI yang menentukan kuota-kuota itu, jadi yang tahu LIPI itu aku waktu itu cuma itu. Jadi waktu daftar itu benar-benar blank, LIPI itu apa tuh nggak ngerti. Kan dulu bayanganku peneliti itu kan kaku, kayak profesor, kayak apa gitu. Cuma dulu tuh memang senangnya emang ke kayak kegiatan outdoor gitu ya. Lebih senang gitu waktu pas kuliah di biologi, dikenalin hal-hal yang kayak gitu. Enjoy juga sih, ya maksudnya artinya setelah aku masuk di LIPI berubah gitu, paradigma peneliti yang kaku dan lain-lain itu ternyata nggak kayak gitu waktu di LIPI.
Kesenangan saya terhadap hewan itu memang sejak kecil. Apapun itu, baik burung, ular, kata, saya senang hewan sejak kecil. Hal yang paling mendasar yang men-encourage saya menjadi seorang peneliti itu pada saat saya masuk Fakultas Biologi. Di situ saya tergabung di dalam kelompok studi herpetologi, di mana di situ mulai mengenalkan atau mempelajari, mengenal sebetulnya amfibi dan reptil. Justru itu challenge bagi saya karena belum banyak orang atau belum banyak peneliti yang menggeluti bidang itu. Sehingga karena belum banyak orang yang mengetahui dan karena saya juga senang dari itu, maka saya menggeluti bidang itu sampai sekarang.
In the world of crocodiles, size is everything. And this one is an absolute monster. At 6 meters long,
pandangan kami di biologi, besok kalau kamu itu lulus, dia ya bilangnya ya udah toh Bu, nanti kan ya ke depan wong ilmu itu ya insya Allah ada manfaatnya kan begitu. Oh iya ya, anak itu kalau sudah dewasa dia menentukan seperti itu mesti punya angan-angan dan cita-cita tersendiri yang saya belum bisa mengetahui kan begitu. Saya menyadari itu semuanya, maka saya pun ya hanya mendoakan begitu saja. Dan kuncinya kamu di mana saja, kapan saja, kamu di hutan, kamu di mana saja, jangan sampai meninggalkan sholat. Inilah pesan kami. Doa, mohon, semua keselamatan itu hanya di tangan Allah. Semuanya kamu jadi peneliti, jadi ini semuanya kehendaknya Allah. Allah kalau tidak menghendaki tidak jadi seperti itu kamu. Tujuan kamu adalah mencari ilmu. Dan kalau kamu mempunyai ilmu, manfaatkan pada orang lain. Pesan kami hanya seperti itu.
Background saya S1 saya dari budidaya, hewan budidaya, kemudian saya mulai berkenalan dengan hewan liar, berkenalan dengan kelelawar, tikus, kemudian juga sesudah lulus terpikir saya ingin sesuatu yang lain gitu. Kalau gitu mungkin salah satu tempat yang kira-kira pas untuk ini ya LIPI. Hewan yang saya pertama kali kenal adalah ya kelelawar. Kenapa saya pilih kelelawar? Karena pada saat itu adalah ketika saya lihat kok cukup banyak ya kelelawar, suatu yang menarik sebab bayangan saya kelelawar itu hanya ya pemakan buah ya, codot. Kemudian yang saya pikir pada saat itu adalah kelelawar itu semua rambutnya berwarna gelap abu-abu. Ternyata ketika saya mulai
bekerja, mulai berkenalan dengan kelelawar, mulai menangkap dengan jaring, rupanya sangat bervariasi.
Kebetulan bukan ekspedisi besar, jadi pergi hanya bertiga, aku dengan teman peneliti asing dan dengan satu teman dari kantor. Kita itu negosiasi, kita mulai dari jam 4 sore, berakhir jam 2 pagi. Itu hanya penentuan harga dan dengan bahasa yang kita nggak tahu, saya butuh translator dan lain-lain, itu yang benar-benar menguras energi. Kemudian masih harus mengikuti upacara adat ketika masuk sebuah kawasan. Saya nggak tahu ya, kita menghormati kan setiap daerah itu adatnya seperti apa. Cuma itu benar-benar menguras emosi dan lain hal gitu, karena upacara adat itu nggak sederhana. Upacara adatnya sederhana, tapi proses menuju itunya itu nggak sederhana. Harus mengumpulkan semua tetua adat yang tersebar di mana-mana gitu, semua harus berkumpul. Terus kita harus mengunyah sirih dan pinang, kemudian memberikan kayak koin gitu untuk diberi sebagai persembahan dan lain-lain. Nah, kan dari perwakilan adat itu ada yang ikut kita ke atas mendampingi jika terjadi sesuatu menurut mereka. Kita tuh sebenarnya belum sampai pada batas yang kita sepakati di bawah, jadi masih kurang sekitar 200 meter lagi lah ketinggiannya. Ada angin sebenarnya, angin waktu itu kan gelombang memang tinggi, jadi ada angin musim itu dari laut itu angin besar, sampai kita di atas itu angin besar kan. Nah di situ udah yang orang dari adat yang ikut kita itu mulai berdoa dan lain-lain, dan akhirnya kita tuh seperti dibilang ini karena kita melanggar aturan adat. Cuma saya pikir karena mereka sebetulnya takut di hutan itu gitu. Nah terus mereka pergi ke atas pagi harinya, nah dia bongkar semua trap kita sampai di bawah. Ya itulah yang benar-benar menguras emosi jiwa gitu.
Intinya ketika kita datang ke suatu wilayah, jangan sampai meninggalkan masalah gitu. Jadi harus kita meninggalkan juga harus dalam kondisi peaceful juga. Jadi apapun itu, pride di harga diri itu tidak perlu ditonjolkan terlalu berlebihan sehingga membuat orang lain tidak respect ke kita. Jadi ya sebisa mungkin bernegosiasi untuk menyelesaikan masalah. Itu yang terpenting. Dan ingat kita pendatang gitu. Jadi hormati aturan yang ada, walaupun kita pikir itu nggak fair gitu ya, tapi memang aturannya seperti itu di sini. Memang dia yang punya aturan seperti itu, ya udah hormati dulu. Paling men-challenge itu ketika kita menghandle ular yang cukup besar. Jadi ketika kita menemukan kayak misalnya saya dulu pernah pengalaman di situ ada piton karpet sepanjang 3,5 meter. Dan saat itu ular itu berada di atas pohon yang di bawahnya adalah air atau teluk ya pada saat, jadi di atas air laut. Kemudian dia tidur di situ. Hampir seminggu ular itu diam di situ. Karena di pulau itu belum pernah ada record ular jenis itu, maka saya harus mendokumentasikan menjadi sebuah spesimen di museum yang harus kita studi, harus kita cek apakah itu jenis yang sama atau jenis yang beda. Mau nggak mau saya harus mengkoleksinya. Ketika kita survei di lapangan itu ada sesuatu yang baru terus. Jadi bukan hanya pada saat penemuan jenis baru, tapi pada saat kita menghadapi atau menghandle hewan itu kita butuh kesabaran. Kita akan mempelajari hewan itu ketika kita menghandlenya, kita mengkoleksinya, kemudian kita mencatat perilakunya, itu hal-hal yang baru. Jadi ada memang yang sama dengan pengalaman-pengalaman sebelumnya, tapi ketika kita menemukan jenis yang belum pernah kita lihat, itu biasanya kita akan mencatat kembali. Ada informasi baru di situ. Inilah yang menariknya. Jadi informasi sedikit pun tetap akan tercatat, dan itu sangat berharga bagi sains, bagi dunia ilmu pengetahuan.
RSS spesial sama khusus 2 orang ini, like
200 meter.
Mantap. Asal jangan hujan. Hujan bubar ya. Saya tahun ini genap saya 30 tahun bekerja di LIPI. Saya mulai kerja Januari 1986, jadi pas Januari kemarin ada saya 30 tahun kerja di LIPI. Banyak hal yang menarik, terutama ketika kita menemukan sesuatu yang katakanlah aneh, langka, jenis baru, tentunya itu suatu hal yang sangat menarik ya. Saya sempat mengalami saraf saya terjepit tulang belakang di tulang belakang yang keempat kelima itu karena akumulasi dari jatuh. Itu saya pikir cukup lama, 3 bulan saya nggak bisa apa-apa, lumpuh. Lumpuhnya bukan karena tidak bisa bergerak, tapi tidak bisa bergerak karena tidak tahan sakitnya. Nah itu 3 bulan, sudah beberapa
rename ini sudah masuk tapi masih yes. Dan
Terus terang LIPI itu aku nggak tahu apa-apa kecuali dulu nonton kuis Galileo. Artinya setelah aku masuk di LIPI berubah gitu, paradigma peneliti yang kaku dan lain-lain itu ternyata nggak kayak gitu waktu di LIPI.
Ketika kita menemukan sesuatu yang katakanlah aneh, langka, jenis baru, tentunya itu suatu hal yang sangat menarik.
Biasanya kalau sebelum saya berangkat itu saya harus minta doa restu atau izin dari bapak atau ibu atau orang tua kita atau orang yang kita cintai di rumah.
Tujuan kamu adalah mencari ilmu. Dan kalau kamu mempunyai ilmu, manfaatkan pada orang lain. Pesan kami hanya seperti itu.
Apa? Coba dibaca. Ini.
Amfibi dan reptil.
Amfibi dan reptil.
Awalnya sih terus terang LIPI itu aku nggak tahu apa-apa kecuali dulu nonton kuis Galileo. Jadi kan ada terima kasih LIPI. Itu aja sebenarnya aku taunya dari situ. Atau waktu pas kuliah biologi itu masalah science. LIPI yang menentukan kuota-kuota itu, jadi yang tahu LIPI itu aku waktu itu cuma itu. Jadi waktu daftar tuh benar-benar blank, LIPI itu apa tuh nggak ngerti. Kan dulu bayanganku peneliti itu kan kaku, kayak profesor, kayak apa gitu. Cuma dulu tuh memang senangnya emang ke kayak kegiatan outdoor gitu ya. Lebih senang gitu waktu pas kuliah di biologi, dikenalin hal-hal yang kayak gitu. Enjoy juga sih, ya maksudnya artinya setelah aku masuk di LIPI berubah gitu, paradigma peneliti yang kaku dan lain-lain itu ternyata nggak kayak gitu waktu di LIPI.
Kesenangan saya terhadap hewan itu memang sejak kecil. Apapun itu, baik burung, ular, kata, saya senang hewan sejak kecil. Hal yang paling mendasar yang men-encourage saya menjadi seorang peneliti itu pada saat saya masuk Fakultas Biologi. Di situ saya tergabung di dalam kelompok studi herpetologi, di mana di situ mulai mengenalkan atau mempelajari, mengenal sebetulnya amfibi dan reptil. Justru itu challenge bagi saya karena belum banyak orang atau belum banyak peneliti yang menggeluti bidang itu. Sehingga karena belum banyak orang yang mengetahui dan karena saya juga senang dari itu, maka saya menggeluti bidang itu sampai sekarang.
In the world of crocodiles, size is everything. And this one is an absolute monster. At 6 meters long,
Pandangan kami di biologi, besok kalau kamu itu lulus, dia ya bilangnya ya, udah toh Bu, nanti kan ya ke depan wong ilmu itu ya, insya Allah ada manfaatnya kan begitu. Oh iya ya, anak itu kalau sudah dewasa, dia menentukan seperti itu mesti punya angan-angan dan cita-cita tersendiri yang saya belum bisa mengetahui kan begitu. Saya menyadari itu semuanya, maka saya pun ya hanya mendoakan begitu saja. Dan kuncinya kamu di mana saja, kapan saja kamu di hutan, kamu di mana saja, jangan sampai meninggalkan sholat. Inilah pesan kami. Doa, mohon, semua keselamatan itu hanya di tangan Allah. Semuanya kamu jadi peneliti, jadi ini semuanya kehendaknya Allah. Allah kalau tidak menghendaki tidak jadi seperti itu kamu. Tujuan kamu adalah mencari ilmu. Dan kalau kamu mempunyai ilmu, manfaatkan pada orang lain. Pesan kami hanya seperti itu.
Background saya S1 saya dari budidaya, hewan budidaya, kemudian saya mulai berkenalan dengan hewan liar, berkenalan dengan kelelawar, tikus, kemudian juga sesudah lulus terpikir saya ingin sesuatu yang lain gitu. kalau gitu mungkin salah satu tempat yang kira-kira pas untuk ini ya LIPI. Hewan yang saya pertama kali kenal adalah ya kelelawar. Kenapa saya pilih kelelawar? Karena pada saat itu adalah ketika saya lihat kok cukup banyak ya kelelawar, suatu yang menarik sebab bayangan saya kelelawar itu hanya ya pemakan buah ya, codot. Kemudian yang saya pikir pada saat itu adalah kelelawar itu semua rambutnya berwarna gelap abu-abu. Ternyata ketika saya mulai kerja, mulai berkenalan dengan kelelawar, mulai menangkap dengan jaring, rupanya sangat bervariasi.
Bukan ekspedisi besar, jadi pergi hanya bertiga, aku dengan teman peneliti asing dan dengan satu teman dari kantor. Kita itu negosiasi, kita mulai dari jam 4 sore, berakhir jam 2 pagi. Itu hanya penentuan harga dan dengan bahasa yang kita nggak tahu, saya butuh translator dan lain-lain, itu yang benar-benar menguras energi. Kemudian masih harus mengikuti upacara adat ketika masuk sebuah kawasan. Saya nggak tahu ya, kita menghormati kan setiap daerah itu adatnya seperti apa, cuma itu benar-benar menguras emosi dan lain hal gitu, karena upacara adat itu nggak sederhana. Upacara adatnya sederhana, tapi proses menuju itunya itu nggak sederhana. Harus mengumpulkan semua tetua adat yang tersebar di mana-mana gitu, semua harus berkumpul. Terus kita harus mengunyah sirih dan pinang, kemudian memberikan kayak koin gitu untuk diberi sebagai persembahan dan lain-lain. Nah, kan dari perwakilan adat itu ada yang ikut kita ke atas mendampingi jika terjadi sesuatu menurut mereka. Kita tuh sebenarnya belum sampai pada batas yang kita sepakati di bawah, jadi masih kurang sekitar 200 meter lagi lah ketinggiannya. Ada angin sebenarnya, angin waktu itu kan gelombang memang tinggi, jadi ada angin musim itu dari laut itu angin besar, sampai kita di atas itu angin besar kan. Nah di situ udah yang orang dari adat yang ikut kita itu mulai berdoa dan lain-lain, dan akhirnya kita tuh seperti dibilang ini karena kita melanggar aturan adat. Coba saya pikir karena mereka sebetulnya takut di hutan itu gitu. Nah terus mereka pergi ke atas pagi harinya, nah dia bongkar semua trap kita sampai di bawah. Ya itulah yang benar-benar menguras emosi jiwa gitu.
Intinya ketika kita datang ke suatu wilayah, jangan sampai meninggalkan masalah gitu. Jadi harus kita meninggalkan juga harus dalam kondisi peaceful juga. Jadi apapun itu, pride di harga diri itu tidak perlu ditonjolkan terlalu berlebihan sehingga membuat orang lain tidak respect ke kita. Jadi ya sebisa mungkin bernegosiasi untuk menyelesaikan masalah. Itu yang terpenting. Dan ingat kita pendatang gitu. Jadi hormati aturan yang ada, walaupun kita pikir itu nggak fair gitu ya, tapi memang aturannya seperti itu di sini. Memang dia yang punya aturan seperti itu, ya udah hormati dulu. Paling men-challenge itu ketika kita menghandle ular yang cukup besar. Menemukan kayak misalnya di situ ada piton karpet sepanjang 3,5 meter. Dan saat itu ular itu berada di atas pohon yang di bawahnya adalah air atau teluk ya pada saat, jadi di atas air laut. Kemudian dia tidur di situ. Hampir seminggu ular itu diam di situ. Karena di pulau itu belum pernah ada record ular jenis itu, maka saya harus mendokumentasikan menjadi sebuah spesimen di museum yang harus kita studi, harus kita cek apakah itu jenis yang sama atau jenis yang beda. Mau nggak mau saya harus mengkoleksinya. Ketika kita survei di lapangan itu ada sesuatu yang baru terus. Jadi bukan hanya pada saat penemuan jenis baru, tapi pada saat kita menghadapi atau menghandle hewan itu kita akan mempelajari hewan itu ketika kita menghandlenya, kita mengkoleksinya, kemudian kita mencatat perilakunya, itu hal-hal yang baru. Jadi ada memang yang sama dengan pengalaman-pengalaman sebelumnya, tapi ketika kita menemukan jenis yang belum pernah kita lihat, itu biasanya kita akan mencatat kembali. Ada informasi baru di situ. Inilah yang menariknya. Jadi informasi sedikit pun tetap akan tercatat, dan itu sangat berharga bagi sains, bagi dunia ilmu pengetahuan.
RSS spesial sama khusus 2 orang ini, like. Kerja Januari 1986, jadi pas Januari kemarin ada saya 30 tahun kerja di LIPI. Banyak hal yang menarik, terutama ketika kita menemukan sesuatu yang katakanlah aneh, langka. Saya sempat dalam belakang 4 kali itu saraf udah dukanya itu ya, kadang-kadang kita harus naik gunung cukup tinggi, turun lagi, kemudian lewat padang yang sangat panas. Ini sebab kalau tidak, pikirnya ngapain jauh, enggak, lebih baik enak tidur di rumah dan sebagainya. Tapi saya menikmati pekerjaan ini. Kemudian juga ekspedisi, nantinya kita kerjakan di lab, hasilnya kita periksa lebih lanjut, kita bikin karya ilmiah, karya tulis yang tentunya kita berharap yaitu bermanfaat bagi kita semua, bagi kesejahteraan, bagi masyarakat dan kesejahteraan bangsa.
Ketika kita ke lapangan, kita bisa fresh, betul-betul fokus pada riset kita yang kita senangi. Jadi kita benar-benar merasa senang atau bahagia karena kita bisa melakukan yang kita senangi, yaitu hobi riset tadi, hobi survei segala macam. Kemudian sukanya lagi kita lebih dekat dengan teman-teman tim. Satu, karena kita di lapangan, kita tahu kondisi mereka pada saat mereka down, kita lebih dekat secara emosional. Kita bisa berkomunikasi, makan bareng, ke sungai bareng, mencuci bareng segala macam.
Ketika kita bekerja, kadang-kadang aku nggak inget siapa-siapa. Aku hanya inget, maksudnya ya nggak tahu, mungkin terlalu fokus atau apa, terkadang nggak inget. Cuma yang kalau di lapangan, yang kalau kita langsung inget keluarga dan lain-lain itu ketika kita dapat apa ya, tawa itu baru kita ingat. Kalau dalam
nomor yang Anda tuju tidak ada. Nomor yang Anda tuju tidak menjawab.
Ibu, nih ditumper. Halo. Ibu lagi nih. Tak telepon lah diangkat ya. Alhamdulillah sehat kabeh. Bapak Ibu sehat? Lancar, alhamdulillah. Ya sing ngono sik tak ngubungi Ibu sik karo Bapak ya. Biasanya kalau sebelum saya berangkat itu saya harus minta doa restu atau izin dari Bapak atau Ibu atau orang tua kita atau orang yang kita cintai di rumah. Kemudian setiap ada sinyal atau setiap ada kesempatan itu, walaupun SMS, walaupun belum bisa telepon, tapi kita ngabari bahwa kondisi kita baik-baik, posisi kita ada di sini. Kemudian begitu kita pulang ke kota, itu kita juga harus memberi informasi. Intinya setiap ada kesempatan untuk memberi kabar ke rumah, ya berilah kabar ke rumah, karena tentu saja orang-orang di rumah yang mencintai kita pun juga mengharapkan hal yang sama.
Ada tapi katanya di lagi umur lagi.
Nah, kalau yang ini kuda itu jelas ada. Nah, yang saya butuhkan sebenarnya kan informasi kantong-kantong di mana sih peternak yang punya kuda-kuda banyak. Nah, itu sebenarnya yang saya butuhkan informasi itu. Kita udah negosiasi, udah oke, sosial gitu. Jadi ya disabotase, kita gagal. Ya itu sih. Danau aja yuk.
Danau, oke. Okay. First walking.
Yuk.
By foot.
By car.
Salah satunya kita tidak berkomunikasi dengan mereka sehingga mereka merasa dilangkai. Itu yang paling banyak terjadi. Yang kedua, ada beberapa yang memang pengen mengambil keuntungan dari kedatangan kita. Nah, ini sebetulnya kita nggak perlu strict mengeksklud mereka, dirangkul aja, diajak ngobrol, dia pendekatan yang persuasi. Saya pikir itu akan cair. Yang kuda kemarin di sini, itu pemilik kuda misalnya oke, kemudian ada satu provokator yang memang, jadi bisa saja kalau kita menjelaskan ke mereka bahwa kedatangan kita itu sebagai peneliti yang resmi, yang juga ditugaskan oleh bupati untuk datang mengungkap potensi di sini, saya pikir dia juga nggak akan banyak menghalangi kita.
Ya karena ini kudanya hidup, jadi kita memutuskan biar ya dengan cara yang non destruktif lah ya. Jadi kita ngambil rambut aja dari kuda untuk kita analisa lebih lanjut nanti di lab.
Kemarin tuh kan ada satu kita bengkok, dia berontak di keinjek. Tuh, gerak terus dia.
Baik, kita buka lagi. Nih, kita buka lagi. Apa? Coba dibaca. Yang ini.
Itu fauna.
Terus bawahnya? Ini.
Amfibi dan reptil.
Amfibi dan reptil. Terus? Berarti herpetofauna artinya apa?
Binatang.
Yang?
Bukan burung walet itu.
Binatang yang melata. Kita akan menggunakan baik siang maupun malam. Siang itu target kita adalah skiing atau kadal yang aktif di siang hari. Itu kita menggunakan trapping, jadi dalam hal ini menggunakan glue trap untuk menangkap atau mengkoleksi jenis-jenis tersebut. Kemudian untuk malam hari targetnya adalah koleksi katak dan ular. Karakter yang paling bisa dilihat dari jenis-jenis ular berbisa itu pasti dia mempunyai gigi taring ya, gigi taring. Ini gigi taringnya, jadi kalau kita bisa melihat ini sudah kan, nih. Ini gigi taring. Oke. Dan gigi taringnya untuk grup marga Crotalidae ini, suku Crotalidae ini bisa ditarik ke belakang. Jadi kalau dia nggak menggigit, posisinya terlipat ke belakang. Ketika dia menggigit ke depan seperti ini, nancap terus dia bisa tarik ke belakang. Itu namanya Solenoglypha. Di sini ada satu jenis ular hijau namanya Trimeresurus insularis. Di Indonesia kasus gigitan ular ini paling banyak, sehingga dari riset ini saya ingin mengkoleksi kualitas fenom dari jenis ular tersebut. Nah, kualitas fenom dari jenis ular ini bisa digunakan untuk aspek bioprospeksi, guna pemanfaatan untuk reduksi kanker, kemudian beberapa bahan obat lainnya, yaitu dari fenom ular tersebut.
Pas waktu awal jadi peneliti, tega nggak tega ya. Sekarang sih biasa saja, dalam arti bukan aku ingin membantai ya, tapi makanya kita mengambil spesimen itu ya secukupnya aja, nggak usah terlalu banyak, karena kan niatnya hanya untuk pengetahuan kan. Jadi ya gitu, kalau sekarang sih sudah lebih tahan ya. Kalau awal-awal sih memang kayak nggak tega gitu, apalagi kalau hewannya sudah lebih besar dari tikus dan kelelawar gitu. Itu kadang tuh mata mereka seperti berbicara.
Tapi ada etikanya gitu, bagaimana cara membunuh hewan agar hewan itu merasa tidak disakiti, kemudian menjadikan spesimen itu jangan sampai rusak. Ini sangat berharga, sampai kapanpun tetap harus terawetkan, sehingga ada standarnya bahwa spesimen itu benar-benar harus terawetkan dengan benar, sehingga tidak nanti kalau kita kembali lagi ke sini sudah tidak mengkoleksi lagi hewan-hewan yang sama.
Perasaan bersalah atau tidak itu sebetulnya saya kembalikan lagi ke diri saya, sebab ini semua untuk ilmu pengetahuan dan juga untuk konservasi, untuk pelestarian hewan itu sendiri. Intinya yang kami lakukan adalah menangkap, kemudian beberapa ekor kami ambil sebagai sampel untuk dipelajari lebih jauh lagi, dan itu adalah tujuannya untuk mempelajari aspek-aspek biologinya. Jadi dengan mengambil sampel yang terbatas sebagai cuplikan, kadang cukup kita juga ketika merasa jumlahnya sudah cukup, kita catat saja jumlahnya, berapa ekor, jenisnya apa, kita lepaskan lagi. Nah, intinya adalah dengan mengungkapkan hal seperti itu, tentunya kita bisa menyampaikan rekomendasi apa yang sebaiknya dilakukan agar keanekaragaman hayat itu tetap terjamin, lestari, dan tentunya tidak hanya itu, pemanfaatannya bagaimana itu yang lebih penting lagi, sebab itu untuk kesejahteraan bangsa.
Ini apa lagi?
Kehidupan peneliti apa ya?
Wes, Mas punya pacar nggak gitu?
Mas punya pacar nggak?
Serius ditanya.
Aku serius lah. Direkam loh Yunus.
Kita bisa lihat ekornya, telinga besar, ekor panjang.
Waktu saat saya lihat itu matanya dan telinganya yang kelihatan.
Kira-kira adakah kira-kira yang menyerupai ini?
Ceritain dong Pak, masak apa nih?
Ini masak jamur saus padang. Saus itu ala Mandiri bersama Chef Tri.
Jamurnya jamur apa?
Jamur tiram.
Apa di mana?
Hutan Elvin.
Kalau perasaan saya sih begitu ikut ekspedisi ini kayak dapat beasiswa. Bener. Karena semua temen LIPI, BIT, dan lain-lain itu seperti guru buat saya. Gitu, ketika ketemu di lapangan ternyata wah asik juga, banyak nambah ilmu gitu kan. Bayangan saya tadinya ketika bareng-bareng para ilmuwan ini pasti kerjanya kaku. Ternyata seru semuanya.
Wabah pneumonia misterius tengah menggemparkan
Wuhan karena dianggap sebagai awal penyebaran virus corona.
Terus meningkat, menyentuh angka lebih dari 5.500 pasien.
Akhir tahun 2019 lalu, dunia dikejutkan dengan munculnya virus baru ketika beberapa pedagang dari pasar grosir makanan laut Huanan, kota Wuhan, Provinsi Hubei, China, positif terpapar Coronavirus Disease 2019 atau COVID-19. Virus ini sangat mirip dengan virus corona yang ada pada kelelawar bat SARS-CoV. Walaupun belum ada bukti bahwa asal virus COVID-19 ini bersumber dari kelelawar, tapi nggak ada salahnya kita mengenal apa itu kelelawar. Kelelawar merupakan satu-satunya mamalia yang dapat terbang sempurna yang berasal dari ordo Chiroptera. Satwa ini memiliki lebih dari 1.400 spesies di dunia dan 239 spesies diantaranya berada di Indonesia. Kelelawar memiliki habitat alami di gua, hutan, bahkan di lingkungan sekitar kita. Kalau di perkotaan, biasanya kelelawar akan hinggap di loteng atau atap bangunan. Kelelawar disebut sebagai hewan nokturnal, yang artinya hewan ini tidur di siang hari dan berburu makanan di malam hari. Karena itu, kelelawar memiliki kemampuan observasi yang sangat baik dalam kondisi gelap. Kelelawar pemakan buah memiliki penglihatan yang tajam dan indera penciuman yang sangat baik. Sedangkan kelelawar pemakan serangga memiliki kemampuan ekolokasi dengan menggunakan gelombang ultrasonik yang berasal dari hidung dan mulutnya untuk mencari mangsa. Yang unik, kelelawar memiliki cara tidur dengan menggantung terbalik menggunakan kedua kakinya. Karena warnanya yang hitam atau abu-abu, kelelawar sering disebut hewan yang kotor. Ternyata sebaliknya, kelelawar termasuk hewan yang bersih karena rajin membersihkan tubuhnya secara teratur. Kelelawar diduga merupakan salah satu reservoir alami atau tempat berkembang biak agen penyakit yang dapat berpindah ke manusia. Hal ini dapat dijelaskan melalui istilah zoonosis. Zoonosis atau penyakit zoonotik adalah penyakit yang secara alami dapat menular dari hewan ke manusia atau sebaliknya. Zoonosis disebabkan oleh patogen seperti bakteri, virus, fungi, serta parasit seperti protozoa dan cacing. Flu burung adalah salah satu dari sekian penyakit yang ditularkan dari hewan ke manusia. Walaupun kelelawar memiliki agen penyakit yang dibawanya, namun jika kita tidak berinteraksi secara langsung dan mengganggu atau merusak habitatnya, maka potensi untuk tertular penyakit dari hewan tersebut sangat kecil. Selain itu, kelelawar juga memiliki manfaat bagi kehidupan. Kelelawar dapat menjaga keseimbangan ekosistem, membantu membasmi hama pertanian bagi para petani, mengendalikan populasi serangga yang berpotensi sebagai vektor penyakit, dan membantu penyerbukan bunga berbagai tanaman buah. Dengan adanya virus corona yang berdampak ke seluruh dunia ini, dapat menjadi salah satu alasan untuk menerapkan regulasi yang jelas tentang pasar hewan dan perdagangan satwa liar, meningkatkan kebersihan dan keamanan di peternakan, meminimalkan kontak dengan hewan liar, dan menghentikan perambahan hutan serta perusakan lingkungan. Kelelawar merupakan bagian dari rantai kehidupan manusia, maka diperlukan kesadaran bersama semua pihak untuk melestarikannya demi kehidupan yang lebih baik. Dengan menjaga alam, berarti kita juga menjaga kehidupan.
Selamat siang.
Selamat siang Bapak Ibu.
Selamat siang. Welcome back to the international webinar on Indonesian Biodiversity, mainstreaming biodiversity conservation, bioprospection, and bioeconomy for sustainable livelihood. Wednesday, September 16, 2020. Next session would be a presentation about mainstreaming bioprospection. As the moderator for the session would be Dr. Khairul Ilmi Setiawan from LIPI. A little bit short brief about Dr. Khairul Ilmi. He is a researcher in Research Center for Biomaterials, Indonesian Institute of Sciences. He has graduated from Kyoto University for his master degree and doctoral degree. And right now, he is Executive Director, the International Society for Sustainable Future of Human Security, and Country Representative Indonesia for Pacific Rim Termite Research Group, PRTRG. Langsung saja to our moderator, Dr. Khairul. Time is yours to lead the session. Thank you.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, ladies and gentlemen. Thank you very much for the committee to introduce me as the moderator of session 2. Let me confirm whether my voice can be clearly heard by all participants. And for this session, we will discuss a very important topics about harnessing untapped biodiversity for novel bioproducts. So we will talk about the challenge and success story. For those who are not familiar with the term of bioprospection, so bioprospection is the exploration of biodiversity resources for genetics or biochemicals information that could be developed into commercially valuable bioproducts. So for this session, we will have 3 prominent scientists. The first speaker will be Dr. Masteria Yunofil Saputra. So let me introduce him a bit. So Dr. Masteria is a researcher at the Research Center for Biotechnology, Indonesian Institute of Sciences, LIPI. And for the second speaker, we will have Dr. Dede Heri Yulianto. He is a researcher from Research Center for Biomaterials, Indonesian Institute of Sciences, LIPI. And for the third speaker, we will have Professor Taifo Mahmud. So he is a professor at Oregon State University. So ladies and gentlemen, for the first speaker, I may invite Dr. Masteria Yunofil Saputra. So let me read a bit about his curriculum vitae. So Dr. Masteria graduated from Università Politecnica delle Marche, Ancona, Italy, for his master and PhD. And his research interest is on medicinal chemistry and marine biotechnology. So he is currently one of the frontline researchers that working on the immunomodulator for COVID-19 research group from LIPI. So I think he will talk about the very interesting topics for the current session. So let's invite Dr. Masteria, and the floor is yours, Ms. Dr. Masteria, please.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Dr. Khairul Ilmi, for such nice introductions. First of all, I would like to thanks to all the organizing committee to invite me as presenter today. So then I would like to thank you also to Bu Atit and Bu Lilis, also helped me to present my presentations today. And today also it's a little bit nervous for me because I am in the same stage with my teacher, a great professor, Professor Taifo Mahmud. I think this today is midnight in US, in Oregon. So thank you very much, Prof. Taifo. So today my presentations about Indonesian, sorry, I will share my screen. Okay, today's my presentation about Indonesian biodiversity for drug discovery and development. So yeah, my presentations may be not much more focused about immunomodulator than what we did in COVID-19, because I think that's today we are talking about biodiversity. So I would like to talking about the biodiversity in terms of general, but I also put some of 6 slides about what we did for the clinical trials in Wisma Atlet for immunomodulatory activity for COVID-19. So yeah, as we know that I'm sorry to mention again that Indonesia is the Indonesia is the second position for the mega biodiversity in the earth. Maybe if we combine between land and oceans, maybe we are in the first one right now in terms of the biodiversity. So that makes Indonesians really advantage for in terms of bioprospections. So in particular for drug discovery. So I see that if we're talking about the maritime area, we can see that Indonesia is the world's most diversity of marine organisms. That's because we are consist of most of 70,000 islands, and we have the 81,000 of the coastlands. So this archipelagics makes Indonesians have much more diversity in terms of the coral reefs, so spawns, soft corals, and ascidians. So how could scientists utilize the biodiversity? So if we talk or we discuss with biologists or ecologists, we always talk about biodiversity about plants, animals, and environments. If we discuss with the chemists or pharmacists, so they look in different area, because the biodiversity, they're thinking about natural products or biological active compounds that can be utilized for pharmaceutical or new drug discovery developments. As you can see, that's okay. If we look in the ocean, I'm like a chemist or pharmacist, so I'm thinking the oceans have like coral reef, have sponge, have the soft coral, have ascidian, have microbes. So I'm thinking that they are consists of natural products, or they consist of bioactive compounds that really benefits to us to looking the pharmaceutical development or new drug discovery developments. So if we talk about drug discovery, that's the long story. It takes a lot of time. I think maybe Prof. Taifo maybe we can explain later. So we need takes 50 or 20 years to develop one new drugs. So it means that it takes long time and takes several phases to start in drug discovery. So you see that's in my presentations right now. From around 10,000 or 20,000 candidate drugs or natural products, maybe only one drug's approval for the markets. And we have to follow 5 steps for approval of the drugs in the market. For the first step, like a discovery screening, discovery takes with collection, the sample, taxonomies, extraction, then we do the screening, such as anti-cancer, antibacterial, and so on and so on. Then after that, we got the compounds, we got the screening good result, then we looking the mechanism of actions like lead optimization, like combinatorial chemistry or structural based drug designs. And discuss the 3 steps with talking about absorption, distribution, metabolism, accretion, toxicity studies such as in vivo study. And the step 4, clinical trials. And the clinical trials, that's long process, because in one drug we have to make 3 clinical trials: phase 1, phase 2, and phase 3. And after phase 3, so we get the approvals. So that's why the clinical trial, that's why the drugs takes a long time to develop. So as we see that Indonesians, we have the most of the, I think we have the second plants biodiversity in the world. So our ancestors, as you know, that we have always used the traditional medicines or medicinal plants. We call it jamu. So in BPOM, we have three traditional medicines into two groups. So first of traditional jamu, that's based on the empirical experience, and the second is standardized herbal medicine that we call it obat herbal terstandar in BPOM. So that's need the clinical study. And the third group is phytopharmaca. So the phytopharmaca is little bit complicated because we have to do clinical study and the clinical studies. So that's three group that classifies by BPOM in Indonesia. So if we look the medicinal plants in industry, so we can see that medicinal plants in Indonesia have high economics and health values in both indigenous and modern community. So from 96 to 2002, we are jumping very much for the market value of the traditional medicines. So you can see 12.4 million to 130 million. That's because the increasing the traditional medicine manufacture. In 96, only 570x industry. Now almost 810 industries and 87 manufacturers considered large-scale industry. So in 2011 and 2015, Indonesian medicinal plants community represents strong competitiveness in the global markets. So we can compare that Indonesian local jamu or obat herbal terstandar or phytopharmaca. We can compare with Campo medicines in Japan or traditional Chinese medicine in China. So that's really good news for us right now. So if you look the production per year, so you can see that most of them like ginger, galagal, lesser galagal, turmeric, and Japanese turmeric. So I believe that's production this year is increasing because the COVID-19 case right now is increasing, and the people use the right now back to the Chinese traditional herbal medicine to prevent themselves from the COVID-19. So you see that's the value also and volume of Indonesian medicinal plants exports so increasing. So then I hope still be increasing also in this year in Indonesia. So from the example of natural products, we can see that medicinal plants, there is 4 drugs right now that's in the markets that we see. First, we can now see artemisinin. So from Artemisia annula, then gatamine from Galanthus woronis. So this use drug ecnobornatical and first isolation from Galanthus. Then anti-cancer, silvesterol. So this is use approvals from FDA approved. Then very most popular in Indonesia is curcumins. So they use for antiviral drugs such as hepatitis B, C, HIV, and RCF. So what we're doing right now in our research groups, we did the clinical trials for the immunomodulator in emergency hospital in Wisma Atlet for the patients mild symptom of COVID-19. So we finished the clinical trials in the last in the middle of August. So right now we have to make a report and submit to BPOM all of the results of our clinical trial, and keep finger crossed that will be good result later. We get good, maybe I hope we can get a good news from BPOM. So we have used 2 commodities for the clinical trial. First, the Immunoglobulin Anthropragis paniculata, then Zingiber officinale Jayamera, then Philanthus neuruli, and Blumia basimira. Then we working also with the Cordyceps militaris. So these are two commodities that we use for the clinical trial in Wisma Atlet. So that's why we choose this commodity for the immunomodulator. So we have several data from the immunomodulator, for example, that's red ginger. So in the clinical studies, so red ginger have the clinical study for the COVID for the immunomodulator. So they have also in vivo study for the significant reduced DNA alpha, interleukin 6 and CV1, and that's really important also for the immunomodulator. And for the in vitro, there is, we can note that Sogal 10-gingero from red ginger have significant reduced NO release from macrophage, and ginger extract increase also EFN1's response. So for example, also we call the grip with Philanthus neuroi. So they have also the clinical study also with this commodity for the immunomodulatory, and they also an in vivo and in vitro. Then we also using the anthropophagous also for the clinical study. We have the also the data. That's why we use this for the immunomodulator COVID-19. So what we did in COVID-19 patients in a clinical trial. So we called our clinical trial ImmunoCov. So we designed with randomized experimental double blinds and placebo control. Then set the emergency hospital for COVID-19 in Wisma Atlet. Then we have a lot of collaborations like LIPI, University of Gadjah Mada, and PDPOCI, Perhimpunan Dokter Obat Tradisional dan Jamu, and PDPI, Perhimpunan Dokter Perhimpunan Dokter Paru in Indonesia, dan BNPB and Puskes TNI. So all of them involved in our clinical trial. So subject COVID-19 with positive patient with myopia and period June to August 2019. So this is our study designs for the clinical trial. So as I mentioned before, randomized experimental double-blind placebo control. So we have 40 days interventions with 19 subjects. So we have group 1, 2, and 3 with standard therapy with intervention once. With maybe we use the we don't know in group 2 and group 3. So but because we have a randomized experimental, so yeah, once maybe the combination herbal with red ginger and Philanthus neuroi and so on, and group 2 with standard therapy intervention with cordyceps militaris, and group 3 is standard therapy with placebo. So this is the gold standard for the clinical trials because we use the randomized experimentals, double blinds, and placebo control. Then, yeah, the inclusion criteria, yeah, the patient that we did 18 and 50 years old, and yeah, should be COVID-19 positive and confirmed with RT-PCR, mild pneumonia, then onset maybe 7 days, less than 7 days, and no comorbid. So exclusion for the outpatient is currently joining no other clinical trials, pregnancy or breastfeeding, and taking other immunomodulator, and they don't have the history TB, leukosis, collagenoses, HIV, and autoimmune. That's very important. So right now we're talking about the marine natural products. So we know that the oceans cover more than 70% of Indonesian territory, represent a virtual untapped resource for discovery potential new drugs. So as literature we did right now, we have more than 100 marine compounds, new compounds, where I said marine compounds that have been isolated from Indonesian marine organisms, such as ponds, soft coral, tunicate, algae, and reported more than 70 publications. So most of the marine compounds have the biological activities such as anti-cancers, antibacterial, and antivirals. So if we see in the markets right now, there is several approved drugs from the marine natural product or derivative from marine natural product, such as the first famous drugs is trabectedin, or we call it famous name Yondelis for anti-cancer. This is the first drug approval from the marine natural products. And the others is cytarabine and vidarabine. So as we can see that cytarabine and vidarabine the market value is $93 million in 2007. And we have also Indonesia like a Conus magus, itu we can set Conus snails, so we can get Indonesia very easy. So they have the name of compound name conotoxins. So we can see that this use with the drugs markets name Prialt using the analgesic treatment or pains. So we can see the revenue from this Prialt reaches $6.1 million in 2019, and that's also interesting for us because still lack of researcher or still lack of industry working with the marine area. That's why we are focusing in marine natural products in our research groups. So we were talking about the SARS-CoV-2. That is very interesting also from the marine natural products. So we call it, we have the one compounds named plitidepsins, we call it aplidin, from Aplidium albicans. So Aplidium albicans, if we can go, if we can diving, we can see in all of the territory in Indonesia. So Aplidium albican. So one of our college from PharmaMars and also from Boeing Pharmaceuticals in Korea, they did in vitro studies from these compounds. So from the in vitro study of these compounds, they saw that this plitidepsis more antiviral activity than remdesivir, and also have the active times higher activity than remdesivir in human lung cells models. So in the coronavirus, so this will be one of the candidate for drugs later in the market for the SARS-CoV-2, and PharmaMars right now did the clinical trial phase 1, and maybe the next months they will do the phase 2 clinical trial, and we hope we can bring these compounds or bring this product to Indonesia also to do the clinical trial for the COVID-19. Then if you can see, that's Indonesia, that's my paper in 2016. Most of Indonesians marine natural products, we're looking for the anti-cancer. So for example, almost 34 papers mention about anti-cancer or cytotoxic activity from Indonesian marine natural products. But unfortunately, most of the marine natural products from Indonesia, most of them conducted for foreign researcher or in collaboration with Indonesian researcher. So just a little 1 or 3 publication on the Indonesian researcher in one publications, and how we hope that will be increasing for the next time in Indonesians. Then we can see that's the global case of marine natural product industry. So in 2002, for the anti-cancer compounds, antibiotic and antiviral were estimated 2.4 billion, and annual profit from the drugs is around 50 million to 100 million per year for this. So what we do in our research group. So we are the new research group. So we call it Center of Drug Discovery and Development. One of my focus is marine natural products. So we also working also with herbal medicine. I will mention you before. So we collected marine macroorganism in several areas in Indonesia. Right now we have around, I think we already have thousands collection of the species from around Indonesia, from Sabang to Biak. The last collection that we did, that we got from our collaborator in Research Center Oceanography, Pak Tri Aryono Hadi. Then after we collected marine organisms, so we never forget about the marine microorganism, because that's very important, because in some papers mentioned that microorganism also produce the same bioactive compounds with the macroorganism. One of our success story about the isolation of bioactive compound from Indonesian marine invertebrates, that is Sinularia. So we isolated around 8 compounds and 5 of them, so you can see in the with the red color, 5 of them are the new compounds. And from the egg compound, we call it the gorgosterol. This is gorgosterol. This gorgosterol is new chemotype FX antagonist that's potentially used for the treatment cholestasis. So if cholestasis, if we have got the liver, so we can use this compound, or maybe we need to do clinical trial or in vitro study later for using this compound to do the cholestasis treatments. And after that, we in the same organism we isolated egg compounds. So most of them is non-polar compounds. First of the type of the chambranoid, so we call it the leptocladolide B, scarbolide B, this non-compounds, and the new compounds chloroscarbolide A, chloroscarbolide E, and prescarbolide. And this glycolipid compound, we call it the cerebrosida, is non-compound, and the new compound is, we call it sinularacida. And this is very interesting because we have 3 acetylated here, and this is very rare in natural products. So the other new compound, we call it sinulalfoxide and sinulalsulfone. So all of this compound we have been conducted with biological activity using inverse protein inhibitions. Only 2 of them have the activity, moderate activity, such as chloroscarbolide A and sinulalfoxide. So this is one our interesting also in sinularia. For example, from this one organism, we isolated 17 compounds, and most of them new compounds. I think most of them around 10 compound is the new compounds from the sinularia. So the next success story is about the licheta. So licheta is a very common sponge in Indonesia. We have also several collection in this licheta in our research group. So licheta have the major compounds. We call it lichetamol A and lichetamol B. So this is the non-compounds, but we looking also interesting for the new biological activities. So new biological activities, we use the TRPA1 channel and potent and TRPA-MX, that will be for the neuroactive. And most of them also have the activity for this TRPA1 channel and TRPA-MX channels. And the next compounds, this with the new paper from us. So this Yersinia, we spawned from Yersinia. We collected in Seribu Islands in 2000s. I remember 2019. Last year we collected this spawns. Then we got in collaboration with also with the Pharmamar, we collaboration with this collected this sample. Then we isolate 2 new compounds, and one of the compound we call it seribunamide. So that's our great honor for the Seribu Island. That's why we call it seribunamide. Then, but unfortunately, we test this compound with cytotoxic activity and several cancer cell lines, but not the activity. But right now, we still develop how can this compound utilize for some such as biological activity. So we still developing these compounds, and not only for the tested with the new activity, we trying also to do the semi-synthesis study of these 2 compounds, because the peptide is very interesting compound, then they should have the biological activities. Then the others ones is we got from the Ascidian Polycarpa aurata. So this Polycarpa aurata is really most common Ascidian in Indonesia. If you can go to snorkeling and diving in 1 meter or 2 meter in coral reef, you can find the Polycarpa aurata. Then this we I collect this this organism around 7 years ago or 6 years ago during my PhD. Then we isolated the compound polyaurine A and polyaurine B, and this is very interesting because these 2 compounds not cytotoxics or don't have toxicity. So because they don't have toxicity, we trying use the these two compound with the in vitro study with schistosoma mansoni, and you see that the polyaurine are impaired X production in vitro of schistosoma mansoni. And one of our papers, the others, is about marine cyanobacteria. So we isolated lagunamate T, we call it lagunamate T. And this is very interesting because these compounds have the activity in nanomolar with a 549 cancer cell lines. And that's very interesting because not very common for the compounds have the activity in nanomolar. For example, that I mentioned aplidin before, that with SARS-CoV-2, this aplidin also have the inhibition of the SARS-CoV-2 virus in nanomolar also. That's why the pharma are also interesting to develop this aplidin for the COVID-19. So what should be concerned to the impact of decreasing biodiversity of novel drug discovery? So yeah, in terms of chemists and pharmacy, if we don't have the plants, or we don't have the virus, so we cannot get new compounds, or we cannot. It's little bit chance for us to develop new drugs discovery, because very important for us is biodiversity. Without biodiversity, we cannot get the new compounds, or we cannot get natural product, we cannot get drug discovery. Then in marine area, that's also really interesting because if we lose the coral reef, we can lose the sponge, we can lose the soft coral, we can lose the ascidian, even we can also lose the microorganisms, because most of them living in the coral reef. So that's very important that we maintain the biodiversity in coral reef. So because the products, they are products, the new compounds are the products, natural products together in coral reef. That's really important for us. So in our research projects, in our research group, we have several projects right now. So we have discovery of novel anti-cancer substance from Indonesian marine organism in collaboration with Pharmamar. Then we have national priority research program, or we call it Indonesia PRN, Prioritas Research Nasional. So we have development of herbal drugs and phytopharmaceutical from Indonesian native plants. That's one of my collaborator, Dr. Siti Irma, will be being a coordinator. Then we have also get the INSINAS, Institute Research National, so the development of functional food based on active collagen extract from Holuteria scabra. And the coordinator from my colleague, Dr. Mas Gita Saputra. And we have also national research priority also in collaborations with Ministry of Industry, the main coordinator. So with development of anti-aging and antioxidant from essential oils in Indonesia. So the most collaborator is Mbak Mega Ferdina Warsito. So this is one of this is the project in our lab right now. So we have right now we have good facilities in our group in Center for Drug Discovery and Development. So you can see that we have five rotary evaporators. So we have also the lab for the microorganisms. So we have biosafety cabinet, we have laminary airflow also. We have the TECANs here, for the microplat reader. So we have also this for distillation of water, milieu water, and we have for the immunomodulator also one our focus. We have Luminex here. I think Indonesia we only have 3 institutes have this equipment right now. So this our lab chemistry labs, and we have several HPLC right now. Maybe we have 4 or 5. I think we have in our research group. Now we have the freeze dryer, and we have right now this is our culture cell lab. So we have 2 biosafety cabinet level 2, and we have the incubator CR2 also in our research group. Then this one of project, one of LCMS in our group. So we have the Agilent 640,000 series triple quadruple ACMS system. So thank you. This is the picture of my college in our research group. So we have young researchers here. Maybe we are around 35 or 40, and this may be the younger generation in LIPI working together. And we have in different research area combined with oceanography. And for me, for example, I moved from oceanography to the biotechnology, and together with our friends in biotechnology, and this is the young people working with us. And thank you very much.
Thank you very much, Dr. Masteria, for very interesting presentation. Ladies and gentlemen, I would like to remind you that you can pose a question, and please type your question on the Q&A chat column. And after the presentation of Professor Taiful Mahmud, we can discuss about your question. And I also would like to remind the participant to fill the attendance form that provided by the committee on the chat room. So please access the link, and we will compile all the question, and we will have a discussion session after all the presenters. So thank you very much, Dr. Masteria, and please, for all participants, keep your question for now, and we move to the next presenter. I would like to invite and introduce Dr. Dede Heri Yulianto. He is a researcher at the Research Center for Biomaterials, Indonesian Institute of Sciences, LIPI, and he graduated from Hime University for master and doctoral degree in agricultural sciences, and his particular research interest is in environmental chemistry and microbiology, applied microbiology, polymer science, and bioprocess and bioremediation. For now, he will talk about the bioprospecting Indonesian white-throat fungi for novel bioproducts and enzyme production. So for Dr. Dede Heri Yulianto, the floor is yours. Thank you very much.
Can you see my slide?
Yes, okay, good, Dr. Dede.
Okay, thank you very much, Dr. Khoil Himi, as a moderator. And also I would like to thanks to the committee to inviting me as a presenter in this international webinar, and also for Dr. Atit as a head of Director of Research Center for Biology, and Dr. Lilis as a head of Research Center for Biotechnology, and also Dr. Iman Hidayat, and also all the committee. And my presentation is related to bioprospecting Indonesian white-throat fungi for novel bioproducts and enzyme production. So I will discuss about how we isolate and bioprospecting of this Indonesian white-throat fungi, and utilize the fungi and also lactase enzyme in the green industrial food process. So this is the outline of my presentation. One is about the Indonesian biodiversity, particularly fungi and white-throat fungi.
And second is biological prospecting, or we can call it as a bioprospecting. The third is a national practice on bioprospecting. And mostly I discuss about my research and how is the threat and challenge and recommendation about the biodiversity. So Indonesia is the richest biodiversity country. This is the world's richest nation in the term of its biodiversity. And Indonesia also is a 17 mega diverse country, made up about 70,000 islands, total which there are unique ecosystems containing a large number of diverse species. It is also 7% of the world's flowering species, 12% about the world's mammal species, and also 16% about the reptiles in the world, and also 70% about the birds in the world. So how about the fungi? So actually, the number of fungi in the world is estimated around 1.5 million until 3 million. This is the new reported by the researchers. And in Indonesia, we researchers in the IPSAP document said that the number of fungi is estimated around 86,000 species, or about 6% of the world. And how about the number of fungi has been described or has been identified? It is around 120,000 species, and the number of fungi is described in Indonesia until 2017 in the status KHT Indonesia 2019, called as a 2,273 species, or it is only 1.9% from the in the world data. So this is I can say that this is a very small data of our described fungi. Therefore, effort to describe more biodiversity of our fungi is very important. How about the meso data? This is also still poor data or still limited data we can find in Indonesian publication or in our country data. So mushroom is only one group in fungi. As we can see here, the fungi based on the Basidiomycota and also Ascomycota, and mushroom is only one group here, including the jelly fungi, brackets, and other kind of the fungi. And specifically in this presentation, I will discuss about the white-throat fungi. Why white-throat fungi is important? So white-throat fungi grows in the wood, utilize the lignin, mostly lignin fraction from the wood. If we see in the wood component, we call it as a lignocellulose material. Lignocellulosic material is an important component in the plant cell wall, and mostly consists of cellulose, lignin, and also hemicellulose. And this fungi can utilize the lignin, mostly lignin fraction, to produce some ligninolytic enzymes such as lacase, lignin peroxidase, and also manganese peroxidase. But they also may produce some cellulase and styrase enzyme in the sequential process. So before we go to another topic, I want to introduce you about the lignocellulose material. Indonesia is a rich biodiversity country also of the biomass. So many biomass source from the Indonesia, maybe other country don't have. So from this biomass, if we look into the small molecules, the complex molecule of lignocellulosic materials consists of lignin about 17 until 25%, and also cellulose 40 until 50%, hemicellulose 20 to 30%, and extractive 5 to 10%. So from the cellulose material, we also can extract the microfibril cellulose and also cellulose molecular and also a very small sugar component from this material, also from the cellulose material, hemicellulose material, that can be utilized for example, bioethanol production and other green industry. For example, from lignin also we can produce many chemicals, green chemicals, lignin derived chemicals, and also from extractive we can maybe resulting some active compound for pharmaceutical or medical purposes. So water fungi mostly use this lignin component in terms of their growth, and they eat this lignin fraction and produce the lactase enzyme. So how the lactase enzyme is a potential, we can discuss in this presentation. And we can see that from the fungi, we can divide it into the mushroom, mycelia, and also metabolite, can produce several novel applications, such as the edible mushroom, functional food, and also very important in food industrial purposes. In terms of mycelia, it can be used as a bioremediation purposes. This is also a very new novel new products or novel product, bioproduct, such as we can call it as a microleather and also microbrick, biofuels, and also in pulp and paper industry. As a metabolite, many mushroom or fungi can produce enzyme, but in this seminar, I will discuss about the lactase enzyme. And also maybe some activated compound also can produced by the metabolite of the fungi. And how is the bioprospecting? Bioprospecting is the schematic search, should be a schematic search for genes and natural compound, design and world organism in wildlife with a potential for product development and biological observation or biophysical, biochemical, and genetic methods without disrupting to nature. So some area can be work in this bioprospecting, such as the bionic prospecting, including design, sensor technologies, architecture, bioengineering, and also biomodeling. Chemical bioprospecting, like drug and pharmaceuticals, pesticides, cosmetic, food, and also other industrial variable products. And the third is gene prospecting, including genetic engineering, crop development, fermentation, and also cell culture. Usually, the bioprospecting have 5 or 4 phases. In the first phase is collecting sample from the nature, and the second is isolation and screening the potential of the gene or enzyme, and the third is molecular identification phase, and the fourth product development and testing and disseminating and commercialization. This phase is generally used in the bioprospecting phase. And how we already conducted, we have isolate and collect some white-hot fungi from all Indonesian place, from the Aceh to the Papua, and we collect, isolate, and identify, and we try to analyze the potential of these fungi in a production of novel bioproduct and enzyme. I just just want to report about our activity. In 2020, we contribute to the Ewin exploration in Taman Nasional Laiwangi, Wangameti, and also in Gandang Dewata Mountain. And in 2018 and 2019, in 2017 and 2018, 2019, we also contribute to the Indonesian Biodiversity Strategic Action Plan Program, Deputy of Life Sciences, the Center for Biology. We collect some sample from the Taman Eden in North Sumatra, in the Toba Samosir Regency. And also our staff member also obtained or getting the funding from the MAB UNESCO Program Research Fund. We isolate and collect and assess the potential of the wild fungi from the Taman Nasional Berbak Sembilang, or other program also from the Botanical Garden in 2017. In Batuaden Botanical Garden, Batam Botanical Garden, and also Kuningan Botanical Garden. So, Indonesian's Biodiversity Strategic Action Plan in 2018-2019, we conducted isolation from Toba Samosir Island in Taman Eden. This is the location. We only use in this area because this is the like the forest, wild forest. So we try to only this area, and in 2 days we get about 175 mushroom samples. So this is a very unique and diverse mushroom found in this Taman Eden area. And in 2019, our team conducted isolation, screening, and assessment of wild fungi in Berbak Sembilang Biosphere Reserve for their potency in wastewater treatment. In Jambi or South Sumatra, Palembang, this is the map of the Berbak Sembilang National Park Biosphere Reserve, and we only collect the sample in this area and also this area. So as we know that Indonesia is home to the largest peat swamp and mangrove forest, about 49% in Asia, which is the largest, and it is located in Berbak National Park. And one of this Berbak and Sembilang Biosphere Reserve is one of the newest designed biosphere reserve by UNESCO in 2018 within the main and biosphere program. And it is located in Southeast Asia, coast of Sumatra, Jambi, and South Sumatra province. The total area is around 3.8 million hectares, dominated by swamp forest, water swamp forest, and mangrove around 350,000 hectares. The area is an important role for biodiversity conservation, as it has a diverse and wide range of rare and endangered species, as well as undistributed peat swamp forest ecosystem. However, this biosphere reserve is threatened by the extensive agricultural development, illegal logging, and grinding and burning of many adjacent swamp forests. Therefore, very important to collect the biodiversity from here and to know to understanding their potential. So from this site, we collect around 1.8 sample from the Berbak and from the Sembilang also. So we bring to the lab, and this is from Bogor Botanical Garden. We also collect around 46 mushroom samples from the forest, mangrove, and also from the garden area. And we try to identify in the based on the morphology and also GNA sequence. And we bring to the lab and screening all these fungus because we want to get the fungus produce lacase enzyme or lacase producing fungi. So we use the lignin, alkali lignin media. This is the selective agar medium for fungus production of the lacase. And after we get the selected fungus, we purify on the mal-stock agar media to get the single colony, and we screening in the several synthetic dyes. And from this screening, we grow the fungus in the center of the petri dish containing the dyes, and we observe 7 days for the growth and also the decolorization rate, and we select. In general, from the Taman Eden, we got the 3.5% potential fungus for lacase producing fungi, and more still, the work is still so many for us to identify the potential from this unidentified fungi or mushroom. From the Bebak Sembilang, we obtained around 20% fungus, potential fungus for lacase producer, and this lacase is a potential for the decolorization agents, pulp and paper application, and etc. So from the Taman Eden, we also get a fungus with the potential for lacase producer, and their growth rate and their decolorization rate is very higher, and this is a potential to be used in the application of the lacase production. This is also the screening method. We screen in the LBBR, and we found that fungi selected based on their ability to degrade or to decolorize LBBR in the petri dish. And we then identified the fungal species using the mushroom itself and also the mycelia, and we then confirmation about the name and the body mushroom, fungal body also. And we found and we check in the status of publication. Many of our findings is new record for tropical fungus from Indonesia for dye decolorization, and also new records for lacase producing water fungi. This is a very potential because Indonesia is so many place in Indonesia and tapping by the biodiversity identification. And what is the application of the mushroom itself? From the mushroom, mostly as an edible fungal mushroom, and this is the global market for edible mushroom, around 42 billion per year. And in the world has been confirmed that about 3,050 species has been confirmed as a fungal food species, with the 18 orders and 114 genera. It is about only 6% from the total mushroom. And from this genera, this is the many is from the Boletales group and also Russulales group. This is the many fungi can be used as a edible mushroom, also found in Indonesia, and this is a very promising business to increase our market in Indonesia. And in terms of mycelia, the fungi can be used for environment purposes, and we have published several journals utilize these fungi to degrade crude oil. And as we know that crude oil become a very compound can contaminating the soil or seawater and resulting the toxic component in the water environment and also in the seawater, and also in the soil. And component of the oil is mostly aliphatic fraction and aromatic fraction, resin and asphaltene fraction. So fungi differs in the ability of degrading this crude oil because of this component. Some fungi difficult to degrade this aliphatic fraction, sometimes easily to degrade the aromatic fraction, and etc. So diversity of the fungi is influence the ability of this fungi to degrade the crude oil. And we found that fungi can degrade the oil. This is the aliphatic fraction, aromatic fraction, resin, and also asphaltene fraction. And we have reported this in this journal. And also one of Indonesian important fungi, Trametes hirsuta D7, collecting from the peat swamp forest in Riau. Also, we already reported in high impact factor journal to degrade the polyaromatic hydrocarbons, penanthrene, anthracene, and also benzoaphyrene. And mostly lacase from this fungus has play a role in the degradation of this polyaromatic hydrocarbon, resulting non-toxic compound. And recently, we also used this mycelia from several places in Taman Eden as a biodegradation agents of the textile and batik wastewater. And this is, we can see here, this is the batik wastewater can degraded by the fungi, the mycelia, to become the colorless and resulting non-toxic component after assay in the human dermal fibroblast cells. And this data is now under review in Journal of Hazardous Material, and we hope this will accept it in this month or next month. Other report also published about the fungal degradation in for using the expanded polystyrene, styrofoam, and microplastic, and also antibiotics. Is a very potential for mycelia for the clean environment. And how about the novel bioproduct or novel biomaterials? Recently, we have a collaboration research with the Mycotech. Mycotech is one of company in Indonesia utilize the mushroom as a bioproduct, such as the leather. And this leather is coming from or made up from mushroom. So it is important to check the potential of the mushroom in order to get the best raw material for the leather. So in our research center LIPI, we have the testing machine for detect the tensile machine, and we screening several mushroom to tensile properties using the UTM, and to obtain the potential or the best mushroom with the higher tensile properties and also elongation at break properties. So this is based on the ISTM D2201. Mycotech has analyzes that the tensile property of this leather is from 6 to 8 megapascal, and also elongation at peak from 38 until 47%. And also mycelia can be used as a novel bioproduct. This is also mycotech. They try to make the fungal mycelia to become a brick, and this brick is strong properties compared than the conventional bricks. This is contain the lignocellulosic materials and the mycelia from the fungus, and grows in the certain incubation condition, and then we get the microbricks. And also mycelia for the biofuels, sustainable biofuels. So in bioethanol production, usually the biomass is harvesting and delivered to the biorefinery factory, and then biomass is a pretreatment by the chemical, physical, or biological. And then the move to enzymatic breakdown of cellulose, we call the hydrolysis, and fermentation using the microbes, usually Saccharomyces. And also we produce the ethanol. Recently, researcher from LIPI, Andriani et al., we reported in Bioresource Technology report in 2000, just accepted this in this month, reported the peripotential Indonesian fungus Trametes hirsuta for sequential production of lignocellulosic enzyme comprise lignin, manganese peroxidase, lignin peroxidase, and also cellulase and silanase enzyme in sequential. So this fungus utilize utilize lignin by the ligninolytic enzyme, lacase, manganese peroxidase, and also lignin peroxidase. And then lignin or degraded in this term until 8 days. After that, the cellulase and silanase enzyme is produced by this fungus and degraded the cellulose and xylose to become small molecular of sugar. In this new findings, we can sort the mechanism of the bioethanol production by combining the pretreatment to remove the lignin in the lignocellulosic materials and the hydrolysis processes. So it is a very important findings for Indonesian biodiversity of water fungi. And we move to the lacase enzyme. So lacase from the water fungi can be utilized as an exploration of biodiversity itself, and also using the enzyme immobilization. Sometimes to make a large-scale production of lacase, we use recombinant production. Or we can also use the protein engineering and also metagenomic research. So
sorry, you have 3 minutes left. So
okay.
Could you speed up the presentation? Thank you.
Okay, thank you very much. So this is the lacase for industry, commercial use for bioremediation, textile, and pulp and paper, and yeah, this is our research group. We use a microbe and we screen, isolate, and the potential mussel become a source of biomaterials. We also work with the enzyme and also biomaterial, and we also making the microbial composite like the micro leather and so on, microbrick, and also we cloning the lacase into the recombinant production, and we produce the enzyme materials, biomaterials in the grafting, synthesizing, and immobilization. So this is our roadmap. We now apply some lacase for bioremediation of textile dyes, and we can replace this step in conventional technology using the lacase technology. And lacase also transforming the food industry by improving the flavor and also addition sugar and so on. And in pulp and paper industry, lakes used in many steps, from the bio-pulping process here and bio-bleaching process, bio-grafting process, and also bio-de-inking process to remove the small part of the black part in the paper, and also used in the effluent treatment to water treatment process, wastewater treatment process, and also utilize lignin as the side products of this industry to become a green chemicals. And lakes also very potential for novel biomedical and also personal care products as an antimicrobial and also skincare. This is very interesting findings by the researchers, because some plant Toxicodendron flume is a result produce usual. Usually this can be poison ivy dermatitis, and the lakes can oxidize this component to become non-toxic compound. So if we use as a skincare or lotion, we can avoid from this poison ivy dermatitis skin. And also lakes can oxidize iodine to become the disinfectant, and this is already reported by some researcher. Lakes also can be used as a hair dyes or textile dyes fibers, resulting more color and more biological color from the lakes enzyme. And also for the personal care has been established in toothpaste, deodorant, mouthwash, and also detergent, soap, and also diapers. And very new research also reported that lakes as an anti-cancer. So global market of lakes is around $3.1 billion in 2019. But we are passing the three biodiversity laws, as already mentioned by our minister, Professor Bambang, and also our chairman LIPI. This biodiversity laws is treat our biodiversity. So by the human activities or natural disturbance like forest fire, logging, and deforestation. And how the challenge is enormous number of untapped and unidentified biodiversity is still large, and we need integrated research and number of human resources like students, researcher, and also research facility. And the second, how we integrated between the conservation and utilization. After we getting the benefits from the bioprospection, this budget can be used as a conservation. So the conservation and the exploitation or utilization, it should be balanced. And we also promote our facilities in LIPI, Integrated Laboratory of Bioproducts. We're having a very advanced equipment and open for all Indonesian researchers can be used here. Please join with us to improve our biodiversity data. We acknowledge our LIPI chairman and our deputy, and also our Bu Atit, as a research center for biomaterial, research center for biology from the IDSAP program and also priority program of LIPI, and all our members, all our collaborators from the Taman Eden, Bebak 9, and also exploration team. And yeah, this is last. NASA said that NASA already investigated how mycelia could help build habitats in Mars and the Moon. So before that, we hope that mycelia also help our Earth in this life, our life. That's all. Thank you very much for your kind attention.
Thank you very much, Dr. Dede Herianto. Again, I remind all participants to post your question on the Q&A chat room. So now we move to the third speakers. I invite and introduce Professor Taifo Mahmoud. So he is a prominent scientist that currently working on the Department of Pharmaceutical Sciences, Oregon State University. So his interest is about pharmaceutical science and also regarding with bioprospection. So he will talk about bioprospecting and genome mining in natural products discovery. So Professor Mahmoud, the floor is yours. Thank you.
Thank you, Dr. Himi, for the kind introduction. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. First of all, I would like to thank the organizing committee for inviting me to give a talk in this wonderful symposium. If I can share my screen first. All right, can you see my screen?
Yes.
Yes, you can see.
Yes, thank you. So, so I was very happy that Dr. Masteria just now have given a very nice introduction about natural products, drug discovery, and then also about biodiversity. And Dr. Dede also talked about biodiversity and the potential of microorganisms, particularly fungi, for biomaterials and also for enzymes that are very useful for biotechnological purposes. Okay, today I would like to share with you some of the work that we have been working or doing in my lab at Oregon State University, particularly on bioprospecting and genome mining in natural product discovery. So perhaps I don't have to explain too much anymore about natural products, but just want to remind you again about natural products, as we know that natural product has been used as medicine for centuries, and even today, natural products continue to be an important source of new drug leads. If you look at the all small molecule approved drugs from 1981 to 2014, about 65% of them are either natural products derivatives, synthetic compound, but with natural product pharmacophore or natural product mimics. So only 35% are pure synthetic compounds without natural products involved in the design of the molecule. This is particularly true with antibiotics. So many antibiotics that are used in clinics are natural products or derived from natural products. Many of them are produced by fungi and also produced by bacteria, particularly soil bacteria. But if you look at the timeline of antibiotic discovery, most antibiotics that we are currently use, such as penicillin and streptomycin, tetracycline, chloramphenicol, erythromycin, vancomycin, all these antibiotics were discovered in the 1940s, 1950s, or 1960s. By the way, this vancomycin here, which is one of the most potent antibiotics that can kill MRSA, for example, was isolated from bacteria that was discovered in Indonesia. And so, as I mentioned, many of these antibiotics were discovered during this day. This is what is called as the golden age of antibiotics. After that period, the number of natural products, antibiotics that were approved for clinical use has declined significantly. Many new antibiotics that were approved more recently are actually only derivatives of known natural products. So in fact, many of those antibiotics have become less effective due to resistant problem. The R&D pipelines for new anti-infectives also is drying up. Cases of antibiotic resistance have steadily increased, but at the same time, the number of antibiotics being approved in the US has significantly decreased. So what is the problem? Apparently, one of the problems is that the traditional approach to drug discovery is no longer sufficient to provide novel drug for us. And of course, there are many compounds that we discover. They are known compounds. They are not new compounds. So natural product drug discovery scientists need to think about more innovative ways of solving this problem. One of them is to find new sources of natural products, such as looking at organisms from extreme or unique ecosystem, looking at symbionts or microbiome, etc. So this is part of the bioprospecting effort. Secondly, we can improve the output of natural product discovery by using new tools and technologies, such as more sensitive instruments, like new screening platforms, or using big data and system approaches such as genomic, proteomic, transcriptomic, and metabolomics. And the third way is to use new approaches in drug discovery research. So basically incorporating new approaches in our research, such as using genome mining, biosynthetic pathway engineering, synthetic biology, etc. So when we talk about finding new sources of natural product, of course, as described very nicely by the two speakers earlier, Indonesia has a lot of advantages because it is rich in flora, fauna, and microorganisms and their biodiversity. Those organisms represent a wealth of untapped resources for pharmaceuticals, agriculturals, and environmental application. But now, of course, they are threatened by climate change, rapid economic growth, for example, and also human activities. So this is something that's become direly important to do research in this area. So as part of our effort to investigate the potential of Indonesian biodiversity as a source of new medicine, we have collaborated with Dr. Dwi Andre Santosa previously from Indonesian Center for Biodiversity and Biotechnology, or ICBB, and also he is a professor at IPB, to investigate microorganisms from Indonesian unique ecosystem called blackwater ecosystems. These ecosystems are poorly studied ecosystems with extreme physical-chemical conditions, but with great species richness. So we receive about 1,000 bacterial strains, 70% of them are actinomycetes, but we just screen a portion of it and have isolated more than 200 natural products. And among them, about 15% of them were new compounds. That means 85% were known compounds. Although, well, we have published many of them in peer-reviewed journals. But although we have some good success with this project, I should admit that this is a laborious and time-consuming effort. So it is quite difficult really to find new natural product by screening and doing biological testing alone. More recently, we have been collaborating also with a number of young and highly energetic scientists from Indonesia. One of them is Dr. Risa Noviani from University of Tanjung Pura, who is focusing on bioactive compounds from marine symbionts. We pursue this project by using a combination of bioprospecting, genome mining, and metabolomic methods. This is a relatively new project, and from 250 bacterial isolates obtained from spawns, well, it's unidentified spawns and coral collected off the coast of West Kalimantan. And we found 12 strains have been identified to be members of Streptomyces species. All of them showed their ability to produce active compounds, again, gram-positive and gram-negative bacteria, and some of them were active against the opportunistic gram-negative bacteria, but bacterial pathogen Pseudomonas aeruginosa. And then we also did metabolomic profiling of extract from the most active strains and found the presence of some known compounds, but also there are a number of unidentified natural products. So they are highly promising source of bioactive natural products. This is still an ongoing project, but I just want to introduce to you that we are doing this kind of stuff, but some preliminary data from this project have been published recently. Another young scientist from Indonesia who has also visited my laboratory was Dr. Yohana Nurulita from University of Riau. Her research focuses on fungi from Indonesian peat swamp soils as antibacterial compound producers. So she is also using a combination of bioprospecting, genome mining, and metabolomics to find new active natural products. For those of you who are not familiar with the term of genome mining, here briefly I want to explain what genome mining is. So the typical technique or methodology for natural product discovery is by analyzing the chemistry of the compound produced by the organism. But as I described just now, it's really laborious and time-consuming. So more recently, with the advance of sequencing technology and metabolomic and so on, it is now possible to do it in a different way. In this case, first thing is to look at the genomics of the organism. Looking at the potential of the organism to produce certain or new natural products. So basically we're looking at the blueprints, the genetic blueprints of secondary metabolism in those organisms. And of course that's required bioinformatic technology or analysis to identify biosynthetic gene clusters that are involved or responsible to make natural products. And then sometimes we require some molecular biology technique to induce the production of natural product by activating the biosynthetic gene cluster. And then of course in the case of fungi or eukaryotic system, then we can do some epigenetic experiment to induce the production of certain natural products, and then spend time to analyze the compound if they are they are prospecting or they have a good potential. So one of the more recent work that we have done with genome mining is by looking at the genome of a bacteria, soil bacteria called Streptomyces pecten. So we obtained the draft genome sequencing from Streptomyces pecten using a combination of Illumina and nanopore sequencing technology, and we got about 35 contigs. And the estimate size of the genome is about 7.8 megabase pair. And then using anti-smash analysis program, we were able to identify 33 putative biosynthetic gene cluster. This means that this particular bacteria can, or at least has, potential to produce 33 different classes of natural products. And 9 of them or putative polyketide synthase gene cluster. And then from our study, we know that the bacteria can produce pertamycin, which is the anti-tumor antibiotics, a very interesting structure. And then NFET133, which is immunosuppressive, anti-diabetic, and anti-trypanosomal drugs, and pyrisidine and conlubatin. Well, of course, if you are only interested in looking for new natural products directly, perhaps you should ignore these compounds, but then pay more attention to the other 29 potential classes of secondary metabolites. But in our case, we are also interested in studying or understanding how these complex molecules are made in nature. So for many years now, we have been studying the biosynthesis of pertamycin, and more recently, we have been able to demonstrate through recombinant protein analysis that this 3-aminoacetophenone moiety here is derived from 3-aminobenzoic acid, which is attached to the acyl carrier protein from a polyketide synthase system. And then this molecule being extended, and then glycosylation occur on this molecule to give this intermediate. So apparently this sugar moiety is the precursor of this 5-member ring of pertamycin. So glycosylation of natural product is very common, but glycosylation of a compound that is still attached to a protein, it is unprecedented. So we have published this result last year, and if you are interested, you can take a look that we were able to demonstrate this stepwise reaction by looking at the LCMS or mass spectrometry of this protein attached molecule. And also we have studied the downstream pathway to pertamycin, including a number of very interesting enzymes and intermediates that involve in the biosynthesis of pertamycin. So pertamycin is, as I mentioned, is anti-tumor antibiotics, but it is broadly or highly toxic. It can kill bacteria, can kill fungi, malarial parasite, also can kill human cells. So it is desirable to make analogs of pertamycin and see if they can have different pharmacological effect. So based on our knowledge of the biosynthesis of pertamycin, we can generate new analogs of pertamycin through biosynthetic pathway engineering. So just imagine that in the bacterial cell, there are pathways or roads to make natural products. Those pathways may be blocked or detoured. By doing this, the bacteria may be making different compounds that they do not normally make. Here we were able to generate a number of new analogs of pertamycin. The red arrows here show the modification that we made compared to the parent compound. We could also introduce fluorine and introduce some other functional group by doing mutasynthesis by feeding the mutant with fluorinated substrate to produce, for example, fluorinated analogs. And interestingly, some of these new compounds, they have much better pharmacological profile than the parent compound. For example, some of them still have very strong anti-malarial activity, but they lose the antibacterial activity and also has very low cytotoxicity. And we have also generated many mutants actually of Streptomyces spectrum by knocking out the biosynthetic gene cluster systematically one by one. As I mentioned just now, there are 33 biosynthetic gene clusters in this bacteria, and at least 9 of them are polyketide synthase. And it has been known that the secondary metabolism is regulated in a very complex way. There are many regulatory systems in bacteria that regulate the production of natural products. Those include global regulation or pathway-specific regulation, and sometimes by knocking out the production of a class of natural products, that would affect the production of other natural products. As you can see here, if you compare this bottom one, by knocking out additional gene cluster, we knock out this cluster 1.23, which is involved in making conglobatan. As you can see, no more conglobatan is produced. But then you can see a lot of other metabolites being produced. Those are even, you don't really see them usually in the parent parent strain or in the wild type. So this natural product just appeared after certain biosynthetic gene cluster being knocked out. The same thing with this. So we knock out the other biosynthetic gene cluster, and suddenly pyridines production almost gone, but then you saw a lot of new compounds being produced. So they are indeed, some of them are new compounds that we haven't seen before. So this is something that is very interesting in terms of using bioprospecting, but also using genome mining to find new natural products. To do genome mining study, you don't really have to be able to sequence your bacteria. You can also mine the genome database available in public domains. We have done this again and again, especially when we like to focus on a certain gene or proteins or enzyme that is involved in natural product biosynthesis. As an example here, I show you our genome database mining study of a family of enzyme called the sugar phosphate cyclases. These sugar phosphate cyclases are involved in primary and secondary metabolism. The most well-known and well-studied is the dihydroquinone synthase, which is involved in the biosynthesis of aromatic amino acid like phenylalanine or tryptophan, also involved in the synthesis of ubiquinone, folic acid, etc. However, there are a number of other enzymes homologous to dihydroquinone synthase. So the gray here is dihydroquinone synthase, but there are other enzymes homologous to dihydroquinone synthase that are involved in secondary metabolism or involved in natural product biosynthesis. We have screened more than 35,000 unique protein sequences from this family and classified them based on their catalytic activity. So far, 6 families of enzymes similar to dihydroquinone synthase have been identified. We have been studying on 4 of them mostly, including this new group of enzyme that we still don't know its catalytic function. 2 of them, EEVS and DDGS here, are unique because they use the same substrate but make different products. So both EEVS and DDGS use cereoheptylose 7-phosphate as substrate, but EEVS produce 2-epi, 5-epi bioluolone, and DDGS produce desmethyl 4-deoxygadusol, using completely different set of chemical reaction. By the way, this enzyme, the crystal structure there looks very similar, and then actually EEVS catalyze the cyclization of this compound to this by using 5 different step reaction. 5 different reactions all occur in the active site of this enzyme. So this is very interesting enzyme. But the thing that many people don't realize is how widely they are distributed in nature. We found that EEVS is distributed in many gram-positive and gram-negative bacteria, and also in vertebrates. And DDGS is distributed in fungi bacteria and marine cyanobacteria, algae, and invertebrates. And EBS involved in the biosynthesis of diverse natural products, whereas DDGS is mainly involved in the biosynthesis of mycosporin and mycosporin-like amino acid, which are known as natural sunscreen. And the presence of EBS in mammals or vertebrates is rather puzzling because, as you can see, most of these are microorganisms. And in fact, vertebrates even, they do not have dihydroquinone synthase, the homolog, the primary enzyme that is similar to EBS and DDGS, and how vertebrates can gain this enzyme or genes. So we then look at the genome sequences of various vertebrates and found the EBS gene in fish, amphibian, birds, and reptiles. It is always together with another genes that we call MTOX because it's encoded a bifunctional enzyme that has methyltransferase and oxidase activity. Human and mammals, they do not have the genes. Perhaps they lost them during evolution. And we also found that 2 species of algae have exactly the same set of genes. So we then characterized the function of the gene biochemically, and we found that these 2 genes are responsible for the biosynthesis of a small molecule called gadusol. Gadusol was first discovered in eggs of Atlantic cod. This is a cod fish. It has a unique UV absorbance at 294 nanometers, so it has a UVB protection activity. It is also a strong antioxidant similar to vitamin C. It is found in many fishes in the eyes, embryos, and skin mucus. And it was suggested that gadusol is acquired through diets. But our study here showed that fish can produce this sunscreen compound by themselves. So we also look at the ancestor of vertebrate like tunicate and lancelet, and apparently they do not have the genes. But the green algae has the gene, so presumably this gene has been transferred horizontally to the bony fish about 500 million years ago. And those genes have been carried over until a modern animal, except mammals. Maybe during evolution, a mammal lost the gene. And interestingly, the coelacanth, this is the fish deep down in the ocean that can be found in Madagascar or even in Sulawesi. This fish, ancient fish or living fossil, they do not have the genes. So perhaps because they stayed too long in the cave down there, they lost the gene. So gadusol and mycosporin-like amino acids play a critical role in marine ecology. They protect marine animals and other organisms from UV exposure and other strong environmental conditions. Gadusol protects them from UVB, and mycosporin-like amino acid protect them from UVA exposure. Some of them can be produced some of the organisms can produce MAAs and gadusol de novo, and some of them can or have to acquire the sunscreen compound from their diets. So again, I highlight here that fish can make gadusol de novo or by themselves, but they also acquire mycosporin-like amino acid from their diet. So they are protected from both UVA and UVB. So this, we have a review article in Natural Product Report a couple of years ago. So what we can do in the lab was to clone the genes from fish and transfer them to yeast or Saccharomyces. And we were able to produce gadusol in yeast. This, the one that we isolated from yeast is really identical to the one that we isolated from fish and also from our enzymatic reaction. So this has made it possible for us to produce this natural sunscreen in large scale by fermentation. Another thing that we were able to do was to combine genes from different sources as part of our synthetic biology work through genetic mix and match. We took a gene from a soil bacterium that belonged to the mycin pathway. We took a gene from zebrafish, from gadusol pathway, and 2 genes from this plant pathogen, Rhodococcus fascian, and then put them together in a host called Streptomyces silica, and we were able to produce new sunscreen compound called gadosporin. So this can be considered as unnatural natural products. So in the US, natural products cannot be patented or protected because they are not really our invention, but compounds such as this that are produced through biotechnology innovation can be protected. So as summary, I have mentioned that natural products continue to be an important source of new drug leads, but unfortunately, traditional approach is no longer sufficient for us to provide novel drug. So it's appear to be that new sources, new tools, and new approaches are necessary. And then of course, bioprospecting and genome mining may lead to new natural products. And I have shown you that biosynthetic engineering also is a powerful tool for generating new compounds. And then also genetic mix and match may lead to unnatural natural products. So with that, I would like to thank the people who did the work, my past and current group members, graduate students, postdocs, and our collaborators. And here is a conflict of interest statement and all the funding agency that support the research. Thank you very much for your kind attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Mahmud. And thank you very much because now in the United States is midnight, isn't it, Professor?
That's correct, yes.
So thank you very much for your time and to join the important session. And now I open to all participants for the Q&A. At first, I will resume the question for each speaker. And after that, I give a chance for the participant to raise a hand, and the committee will allow you to speak directly to the speaker. So please be patient with us. We move to the question to Dr. Masteria. So actually a lot of questions. But I resume it like how to balance between biodiversity exploration and conservation. And in advanced stage, when the bioactive compounds are successfully identified and isolated, what is the next step for the collaboration with pharmaceutical industry? So this is the question that I resume for Dr. Masteria. Please, Dr. Masteria, you can answer
very
directly.
much. Yeah, thank you very much. So the first questions, what's the interesting of the compounds? Can you repeat again the questions?
The question is how to balance between biodiversity exploration and conservation. Some participants thinking about the dangers of like over exploration that can endanger the diversity. So like I rephrase the question, how to balance between biodiversity exploration and conservation. And the second one is in advanced stage, when the bioactive compound are identified and successfully isolated, what is the next step for the collaboration with pharmaceutical industry? What is the strategic step for?
Thank you very much, Dr. Hemis. Yeah, to balance with the conservations and to research. For examples, when we're working in the marine organisms, so we never collected all of the organisms. So we only cuts or small pieces of the samples. For examples, we just only needed on maybe maximum 500 grams of the samples. So then we leave it along all of the species, then they can grow it again. So then if we interesting these compounds from sponge, maybe we got this, we also collecting the microorganism from sponge that we believe it that's, so microorganism also the producers of the same compounds with the sponge. So that's what we did in marine environment. So we never collect all of the material. We only collect a small piece of the compounds. So this is also advantage for the advanced equipments that we have, like we have the mass spectrometry also, so we can identify that compound is new or non-compound. So if in new compound, so we can collect it again with a little bit more, or if we some of the compounds only we can isolate it with what we have it, so we continue the process. So we never collect all of the species because maintain of biodiversity. Then if what the next step after we got the biological active compounds. So yeah, if we got the biological active compounds, so we need to. That's what in my presentations. So for the drug discovery, we have need several tests. So several steps that we need to follow. So for example, we need the in vitro study first or clinical studies. So then if we got the good chance of mechanism of action, so we got the clinical good data, so maybe we can talk with the industry. So maybe in Europe and US, industry can involve in preclinicals, but sometimes in Indonesia, industry they don't want to invest the money until the products good get approval from BPOM, or they just want to, okay, we just involve, we can, for example, we developing but phytopharmaca in Indonesia. So the first steps, industry doesn't want to involve it because they don't want to spend money for research. After we got the good clinical studies research, industry want invest the money. That's the problem in Indonesia because they don't interested in the basic research for drug discovery. So maybe that's my answer, Dr. Himi.
Thank you very much. And some short answer about relating the current work that your team are working on about the immunomodulator for the COVID-19. So about the the jamu or pharmaceutical resources like phytopharmaca. So how you control the supply, like you know, it's very difficult to have standard quality of the natural ingredients, especially that related to the natural drug compounds. So how you working on the if okay, maybe you find like a bioactive compound isolated from like turmeric or something, but how to control like the supply, the quality material that can provide the same and consistent result that you want to achieve, you and your team want to achieve.
Yeah, thank you, Dr. Remy. Yes, that's a very interesting question. How can we maintain the supplies? For example, we have now in Indonesia the national research priority for the we call it the PRN, HT and phytopharmaca. So that's our national research priority, included also how to standardize the materials, how to get the good materials such as Simplicia. That's very interesting. Then in our consortium, so we collaborated right now with Ministry of we get the Balitro for the Ministry of I don't know, Farmer. So they're working about the Simplicia for the phytopharmaca. Then we're working also with the WMKM. So we call it. Then we are working also right now in our national research priority with Kemendes. So Kementerian Desa. So we working with together with the farmer, how to maintain the supply. So we teach the farmer how to get a good standardization of, for example, ginger. So we get the standardization, for example, what else? Philanthus nuri. So that's why we maintain the supplies, they maintain the good quality. So we involve all of the consortium for that. Then, yeah, then we working together also with the industry for that.
Thank you very much. And now we move to the question for Dr. Dede. So may I resume that the participant asking about the potential of edible mushrooms or white rot fungi for bioremediation application, for instance, like heavy contaminated lands, and also how about the potential application of lacase, the versatile enzymes found in plants, fungi, and bacteria on the management of industrial waste, like batik industries or like dirty rivers or something like that. So, Dr. Dede, please answer the question.
Okay, thank you very much, Dr. Himi. And I want to answer the question. So we already conducted the research on biodecolorization of textile dyes and also wastewater from batik industrial and also textile industrial. And we obtained the positive result for this application. Then that our lacase from the Indonesian fungal lacase has a potential to degrade some textile dyes, wastewater treatment, and also not only degraded the compound in the textile wastewater, but also detoxify the wastewater from the industrial or the home industry. And we now submitting this publication in the in the journal, and we hope that it can be accepted in this month, or we hope. And the lacase is can be used directly from for example, powder enzyme. Actually, we already able to produce lacase in the small scale, but using the recombinant lacase production is very important to large scale production of lacase. But the lacase can be used directly to the wastewater treatment, but it is directly loose after utilization. In the best application is using the immobilization. So we are also develop some immobilized matrix for this lacase, and we obtain some matrix is very potential to be used as a matrix for the immobilization with a very low leaching from the lacase itself, and enhance the stability of this lacase to be used in the like the environment, real environment. For example, high temperature until 40 or 50 degrees Celsius, and also from the chemicals. So, and by using this immobilization technique, we can also reuse the fungal lacase in the matrix to be for the another cycle application. So the lacase itself can be used directly and also can be used by the immobilization of this lacase into the matrix, like that.
Okay, thank you very much, Dr. Dede. And now we move to the question to Professor. So, Professor Taifu, so it's a very simple, but I think very interesting question from all participants. How to initiate the collaboration with your laboratory and that's one of the questions from the participant before we continue to the more technical question. Thank you.
Oh, that's easy. Just contact me directly by email. So
okay. very, and as you know, like some issue in Indonesia is like the funding sometimes, Professor, is more focused on the like applied science. And when we hear about like your presentation, so it's more like we are working in very basic science, but have a very big potential to achieve the result. If based on your experience, like for starting the particular project, like in genome mining and bioprospecting, how long should be the ideal condition for multi-years funding to achieve the good result?
Right. Yeah, I think it's all you know, this influence or control by funding agency, right? In the US, it's different from in Indonesia. I know I have been working with many scientists in Indonesia. I know how the funding situation is, you know, how usually you get 3 years funding and then you have to publish first year, second years, and even third years. You have to have a patent application, perhaps.
Yes,
So
yes,
I think
you're right.
I think that's well, that at least not the case here in the US. Basically, you just propose your projects and then you get funding. Depending on the funding agency, NSF usually give for 3 years, but NIH you can get it for 5 years. And then, of course, in your application you have specific aims that you want to achieve. And then that aims, of course, need to be realistic, because other scientists, your peers, will review your application. You cannot just put something or put a lot of promises, and your with the funding amount of money you ask and the amount and the number of people you propose to employ. And then you have to be realistic. And so, of course, everybody know, and like Dr. Masteria said just now, in drug discovery, for example, it's impossible to get a drug, within 5 years or even 10 years. It's a long process.
I see.
And then, of course, with that very clear understanding. So that's the reason why funding agency here never really push you to have a patent application. Because to get a patent application, it needs to be really big invention. You don't want to just file for patent application and nobody even care. So, and it takes money and takes work to write patent application. And in our case, because we are in academia, of course, the university will pay for the fee. And then the university will look at your invention, and they will, we have a special office here just dealing with that, right? Several people in there, they are expert, and they look at your invention, they can tell whether this can go or this cannot go. If they don't see any potential, they are not going to waste their time and spend money to file for application, right? And then most of the work that most of patent application that we had actually came from the basic science research that we didn't even think about or we didn't even plan. You just do your thing as an academician or as a scientist. You do the work that you think is useful, not only for the application of it, but also for the purpose of scientific knowledge. Because a lot of applied science, they come from this very basic scientific knowledge, right? And then sometime, well, this is also the trend in the US. Many university administrators, like the president of the university, they forget about the purpose of being an academic institution. They want to make money too. That's why they push you to do a lot of applied science, so that hopefully you can sell the license and so on. But in academia, you need to understand one of your mission really to produce knowledge. Well, research academia, of course, you have to produce knowledge. And for me personally, if my discovery can get into a textbook one day, that's really a big achievement already. That means what I discover really useful for for the next generation. But well, of course, fortunately, some of our discovery has some potential, and then we end up getting patent application, and some of them also actually as a basis of the new startup company that we founded like 2-3 years ago to develop natural sunscreen compound, and those kind of discovery was not even in the original proposal. So
I see.
So I guess, yeah, I hope that's answered the question.
Thank you very much. Like a very mind-opening answer. Like we may, like internally, can formulate that kind of policy to have peace on mind for the researcher to explore from the very basic research and science. So for this session, the committee contacted me that there is 2 participants that would like to ask directly to all speakers for this session. So I open the chance for Professor Gono Semiadi and Mrs. Windriyanti. So please the committee to arrange the question. Thank you.
Hello, can you hear me?
Yes, Prof. Gono, we hear you very well.
Thank you very much, Prof. Mahmud. I have a question regarding the synthetic biology, and it is not related to the science, but it is more to the policy. To your view, a product coming from the synthetic biology, would that be considered as a product from the non-organism or from the live organisms? And can you please more elaborate about the statute, about the regulation that anything from synthetic biology that in the United States cannot be patented? That, if I'm not mistaken, that you mentioned like that. Can you please more clarify on that issue? Thank you.
Thank you, Professor Gono, for the questions. Can I directly answer?
Yes, Professor Mahmud, you have access directly.
So what I mentioned just now, so the US PTO, US Patent Office, I think about 10 years ago, they had a new regulation or policy that you cannot or you no longer able to patent natural products. Meaning if you go out there and then you isolate plant, you collect plants and isolate new compounds, and then you want to patent the structure of the molecule, you are no longer able to do that. Also from bacteria, any natural products cannot be patented now. So, well, the way how people try to overcome that problem is to make derivatives. So if you have a good natural products, then you modify it and put a methyl group or put an acetyl group that is not natural products, then I guess you can patent that. So in the case of synthetic biology, it's obviously the compounds that are produced are not natural products, meaning it's still produced by the living organism, but it does not occur naturally because you use biotechnology method to modify them. In our case, we took 1 gene from a soil bacteria and then 1 gene from fish and 2 genes from a plant pathogens. So we have 4 different genes from 3 different sources and put them together. And then we were able to make a new compound. Of course, again, that's coming back to Dr. Himi's question just now. The reason why we are able to do that, because we studied the basic science here. We know exactly the function of each of those genes, right? And then because of that knowledge, then we can kind of design a new molecule by mixing and matching, different genes from different organisms. So that definitely an invention. That's an invention that you can protect because we created, the molecule from nothing really. It's not like the organism already know how to make it. I hope that answered my question, Professor Gono.
Thank you for the question. Professor Gono, is it enough or you still want
Yes,
to
it's clear
elaborate?
enough. Thank you very much. Thank you.
Thank you very much. And we move to the second participant. Please, committee, arrange the question. Ibu Windrianti, you can ask. You have your chance now.
You're still mute.
Ibu Wiwin, please unmute your microphone. We cannot hear any question from your side. Thank you. Okay. It seems we cannot hear from Ibu Wiwin. So is there any participant that want to raise the hand to ask directly to the presenters? I open the session. Two participants raise hand. Agustina Senjayani and Wiwin Windrianti. Ibu Wiwin first. Ibu Wiwin, can you talk to us for now? If you cannot access the microphone, then I will pass to Miss Agustina Senjayani. Please, you can talk to us.
Thank you.
Okay. Agustina Senjayani, yes, thank you. So please.
Thank you very much for this opportunity. I would like to ask to Dr. Dede, as you've mentioned previously that you and your team have found a very potential fungi to treat water, wastewater, especially in textile sludge. What about the implementation of this finding? We have so many problems in water pollution today. What have been done and what we need to do more to implement this finding? That is my question. And the second maybe about biomaterial, about the self-life of the endurance and the price maybe when we use biomaterial, what about its self-life and its relation to its price? I mean, is it economically or not? Thank you.
Thank you, Ms. Senjayani. So, Dr. Dede, please, you can answer directly.
Thank you, Bu Agustina. And first question, yes, we have proof that latex can degrade the wastewater dye from wastewater, batik wastewater, and also not only degrade it but also detoxify. And how about the implementations? So this is a good question because in the real wastewater treatment, we have to still need other combination between the physical and also biological. and then we should try in which part this technology can be used in the textile or batik industrial wastewater. And we hope that in the next 2 years, we can apply as a mass production for this technology, I hope. And about the biomaterials, novel biomaterials, yes, of course, the life time of this product should be considered as the quality of the biomaterials and also the price. Some price is still increased, not higher compared than conventional product from other material. For example, our researcher from Research Center for Biomaterial has now already produced like a wooden bicycle, Dr. Wahyu Dwianto. And if we compare the price, yeah, still, because this is a very unique production, very unique product, and we cannot compare with the conventional bicycle because this is coming from the biomaterials. And also, if we compare the micro leather from the conventional leather, yeah, of course, the tensile strength still under if we compare from the conventional, but it can be used as a part of the new product. We can find other good product coming from this micro leather. I hope this can answer your question. Thank you.
Thank you very much, Pak Dede. Unfortunately, the time is very limited, and I already allowed 15 minutes extension from the committee. So thank you very much for the committee to allow the extension. So yes, all ladies and gentlemen. So if we can conclude this very important talk of our session, like we know, like very fast and wide application of bioprospection is very beneficial for us, like for the industries of agriculture, aquaculture, bioremediation, even pharmaceutical. So, and as Professor Mahmoud said, the important support about for this particular field is necessary, even though starting from very basic research and for multi-year support, we can generate important and strategic result. So thank you very much for all the speakers for their interesting sharings and presentation. So time and session, I return to the committee. Thank you very much.
Thank you very much.
Thank you.
Thank you, Dr. Hilmi, for leading the session, and of course, to our great speakers of the session too. Ladies and gentlemen, for continuing to the next session, we will have an interactive quiz again. There will be merchandises from LIPI, and so be ready. And just like previous session, I would like to pass the session to Mr. Akmal. Mr. Akmal, time is yours.
Okay, thank you, Ms. Renata. Time for quiz now. Hello, ladies and gentlemen. It's time to break the second session. Again, we will relax for a few minutes to play the quiz. The committee will display the procedures for playing the quiz. Again, all participants, please click the quiz link in the Zoom chat feature. Once again, all attendees, please click the quiz link in the Zoom chat to go to the game arena. Please pay attention. I will read the rules of the game. Once again, please pay attention. I shall read the rules of the game. Procedures for playing quiz. I will read again. First, This quiz is only followed by WEBINUSA attendees on the Zoom channel. Second, the committee and participants who have won the first quiz session are prohibited from taking the quiz. Again, the committee and participants who have won the first quiz session are prohibited from taking the quiz. Third, before the quiz begins, all participants must provide the identity of the quiz players according to the rules. If there are attendees who do not comply with the rules, they will be disqualified. Fourth, the format for the identity of the participation in the quiz is as follows. Name, 1 or 2 syllables registered in the webinar registration, dash, origin of the institution. The name is up to 25 characters long. First name or second name could be abbreviated. Look at the examples. M. Nikmatullah, dash, LIPI, comma, Akmal Q, dash, LIPI, comma, Husni M, dash, UGM, and Tiara S, dash, IPB. And the last, the quiz will be guided by the MC, and the time for the quiz to end is up to the MC's decision. From all participants who take the quiz, we will choose 5 winners. After getting the winners, the MC will close the quiz by giving a signal. And the following are the books giveaway for all the winners. You can see on the screen. The participant can see on the screen. May you be among the lucky ones to get the prize for the book. Okay, now I want to check the number of participants who have answered. The code for the game, 9343333. 16, 18 participants now. Please join the researcher, the teacher, the students. Please join with us. Click the link that will be shared in the chat. Once again, for the attendees who have not answered, please join by clicking the quiz link in the Zoom chat. We will wait for a few minutes. $102 now. Come on, let's join us. $175 now. Maybe if the participants is up to 200. Okay. 185 now. $193 now. 7 again. Okay. 3 participants again. Okay, let's begin. Ready, set, go. Okay, the quiz game has started. For the quiz participants, answer 10 questions quickly and accurately. Not only facts, but also time. First place now. Waiting for the best one. You still have saya pikir ini lebih seru dari yang pertama. Nyalip terus terjadi dibanding yang pertama. First question is from someone from Universitas Berdaulat. Congratulations to the top 5 winners. Each winner will get LIPI merchandise and one book of LIPI Press. Sekali lagi kami ingatkan, untuk nama player yang tidak sesuai akan didiskualifikasi. Oke. Dan kami akan memilih 5 teratas yang sesuai dengan aturan. The organizers please take screenshots as proof of the names of the winners. We shall contact the winners. Once again, congratulations to the winners. Okay, for the next session, Ms. Renata, time is yours. Thank you.
Okay, thank you, Mr. Akmal. Actually, I tried it also, Mr. Akmal, but however, I didn't make it. So congratulations to the lucky participants who has the top 5. Ladies and gentlemen, I would like to inform you that we will start the next session, session 3 presentation with the topic of mainstreaming bioeconomy, bio-based economy of sustainable biodiversity and bioresource for better future at 3:30 p.m. As the moderator for the third session will be Ms. Anggia Prasetyo Putri from LIPI. So stay tuned, we will back at 3:30 p.m. Now we will play a movie created by LIPI. Please enjoy.
Pada bulan Desember 2019, negara Tiongkok mengumumkan kepada dunia bahwa terdapat virus yang menginfeksi warganya. Bulan berikutnya, virus menyebar ke negara-negara lain dengan kasus bertambah berkali-kali lipat per hari. Virus ini adalah SARS-CoV-2 yang menyebabkan penyakit COVID-19, dan orang-orang menyebutnya coronavirus. Sejak wabah COVID-19 menyerang, segala produk dengan klaim pencegahan dan penyembuhan mendadak laris manis di pasaran. Tak terkecuali ragam rempah Nusantara seperti jahe, kunyit, dan temulawak. Mereka dipercaya bisa meningkatkan daya tahan tubuh sehingga bisa menangkal transmisi virus corona. Dalam dunia botani, tumbuhan rimpang atau rizoma adalah tumbuhan yang tumbuhnya menjalar di bawah permukaan tanah dan dapat menghasilkan tunas dan akar baru dari ruas-ruasnya. Suku temu-temuan, Zingiberaceae, dan paku-pakuan, Pteridophyta, merupakan contoh yang biasa dipakai untuk kelompok tumbuhan yang memiliki organ ini. Rimpang menyimpan banyak minyak atsiri dan alkaloid yang berkhasiat untuk pengobatan. Rizoma yang membesar dan menjadi penyimpanan cadangan makanan, biasanya dalam bentuk pati, dinamakan tuber atau disebut umbi batang. Tumbuh-tumbuhan rimpang di Indonesia dikenal sebagai salah satu sumber bahan pengobatan tradisional. Di antaranya untuk mengatasi sakit perut, kembung, mulas, diare, kehilangan nafsu makan, dan gangguan pencernaan lainnya. Tumbuhan rimpang yang bermanfaat di antaranya temu giring, Curcuma heineana, kunyit, Curcuma longa, temu ireng, Curcuma aeruginosa, temulawak, Curcuma santoriza, rimpang jahe gajah, Zingiber officinale farosco, jahe merah, Zingiber officinale farubrum, jahe emprit, Zingiber officinale faramarum, lempuyang gajah, Zingiber zerumbet, bangle, Zingiber casumanar, dan lengkuas putih, Alpinia agalanga. Kandungan senyawa metabolit sekunder pada tumbuhan ini terutama golongan flavonoid, fenolik, terpenoid, dan minyak atsiri. Senyawa metabolit sekunder yang dihasilkan tersebut dapat menghambat pertumbuhan dan membunuh patogen pada manusia. Senyawa aktif pada tumbuhan rimpang, diantaranya flavonoid pada lempuyang gajah, fenolik pada rimpang jahe gajah, jahe merah dan jahe emprit, dan kurkumin pada kunyit, temulawak, temu giring, temu ireng, lengkuas putih. Terpenoid berupa sesquiterpen pada minyak atsirinya, zingiberen, bisabolen, farnesen, sesquivelandren, dan kurkumen pada rimpang jahe gajah, jahe merah, dan jahe emprit. Senyawa aktif fenolik gingerol, juga zingeron dan gingerdiol, bersifat anti jamur dan antibakteri Staphylococcus aureus dan Escherichia coli secara in vitro. Senyawa terpenoid zerumbon bersifat aktif antikolinesterase secara in vitro untuk pengobatan penyakit Alzheimer. Senyawa fenolik, kurkumin, bermanfaat untuk kesehatan sebagai antioksidan dan antiinflamasi, radang sendi, dan hiperlipidemia. Senyawa tersebut juga diketahui bersifat antivirus dengan secara in vitro. Juga komponen fenolik lainnya bersifat anti kanker gastrointestinal baik secara in vitro
in vivo, dan beberapa penelitian klinis, juga komponen fenolik lainnya, kurkumin, zingerol, dan flavonoid, kaempferol, berpotensi menghambat virus SARS-CoV-2 penyebab penyakit COVID-19. Kurkumin dan flavonoid juga bersifat meningkatkan imunitas atau memperbaiki sistem kekebalan tubuh, imunomodulator. Manfaat dari tumbuhan rimpang yang beragam karena kandungan senyawa aktifnya, baik sebagai anti jamur, antibakteri, antivirus, dan juga bersinergi dengan efek meningkatkan imunitas. Hal ini menjadikan tumbuhan rimpang dapat sebagai alternatif pengobatan modern pengobatan tradisional.
Apa itu vegetasi laut? Salah satu penyebab pemanasan global adalah emisi gas karbon dioksida, yang sebagian besar bersumber pada aktivitas berlebihan manusia. Di atmosfer, gas ini menangkap energi panas dan memantulkan kembali ke bumi. Semakin banyak karbon dioksida atau CO2 di atmosfer, artinya semakin banyak energi panas yang dipantulkan ke bumi. Hasilnya adalah suhu di bumi semakin panas. Sejatinya, gas yang juga larut di dalam air ini adalah bahan baku utama fotosintesis tumbuhan. Di dalam proses fotosintesis, tumbuhan menyerap gas karbon dioksida atau CO2 dan melepaskan oksigen atau O2. Karena itu, eksistensi hutan sebagai penjaga iklim dunia mendapat predikat sebagai paru-paru dunia. Begitulah harapan bagi perubahan iklim dan pemanasan global. Namun, apakah gas karbon yang dihasilkan dari aktivitas manusia seimbang dengan oksigen yang dilepaskan oleh vegetasi hutan? Mungkin tak banyak dari kita yang mengetahui bahwa ada entitas lain di dunia ini yang dapat menyerap karbon dan menghasilkan oksigen atau O2 lebih efektif dibanding hutan manapun di dunia ini. Entitas ini adalah komunitas tumbuhan yang menempati daerah pesisir, mulai dari bagian pasang surut hingga daerah daratan yang masih terpapar pengaruh laut. Ini adalah hutan mangrove, dan ini adalah padang lamun. Keduanya merupakan tumbuhan yang hidup di pesisir dan laut, dan sebagai kesatuan, mereka dikenal sebagai vegetasi laut. Jumlah keduanya mungkin tidak dapat dibandingkan dengan jumlah vegetasi di darat. Lalu, bagaimana mereka bisa menyelamatkan kita dari pemanasan global? Setiap tumbuhan membutuhkan karbon dioksida atau CO2 untuk tumbuh. Mereka menyerapnya dari udara. Karbon ini akan disimpan pada akar, batang, dan daun. Jika bagian dari tumbuhan tersebut gugur dan jatuh ke tanah, maka akan diurai menjadi kompos. Pada vegetasi darat, proses dekomposisi ini akan melepas kembali karbon yang disimpan tumbuhan kembali ke udara. Sementara dekomposisi pada vegetasi laut memiliki mekanisme yang berbeda. Proses dekomposisi pada vegetasi laut terjadi di dalam sedimen yang tergenang air. Karbon yang dihasilkan akan terperangkap pada sedimen dan jaringan akar mengunci gas karbon atau CO2 di bawah air, serta mencegahnya kembali ke udara. Hutan tropis mampu menyerap karbon dioksida sekitar 9,17 ton per tahun per hektar lahan. Namun, sebagian karbon itu akan kembali ke udara melalui proses dekomposisi. Di Indonesia, hutan mangrove menyerap rata-rata 52,82 ton karbon dioksida per tahun per hektar lahan. Sementara, padang lamun mampu menyerap rata-rata 24,13 ton karbon dioksida per tahun per hektar lahan. Itu berarti, hutan mangrove dan padang lamun 2-5 kali lebih efisien menangkap emisi karbon dioksida dari udara. Sementara karbon yang terserap sebagian besarnya akan mengendap di sedimen dalam bentuk material organik selama ribuan tahun. Hutan mangrove dapat menyimpan hingga 1.500 ton karbon per hektar. Sementara padang lamun mampu menyimpan karbon pada sedimennya hingga 293 ton karbon per hektar. Jika ditotal, jumlah ini lebih banyak 6-7 kali dari jumlah sedimen karbon pada hutan tropis, yaitu maksimal 215 ton per hektar. Dari data ini, kita bisa katakan bahwa vegetasi laut adalah gudang karbon organik yang dimiliki bumi. Menjaga kelestarian vegetasi laut berarti kita sedang menyimpan karbon organik dalam tanah agar tak menguap kembali ke udara. Ini berarti kita menjaga kelestarian sumber makanan, kualitas udara, dan air bersih, serta kita menjadi bagian kepedulian dari mitigasi kebencanaan seperti perubahan iklim dan pemanasan global. Dan yang terpenting dari semua fakta ini adalah proses tersebut dapat berlangsung selama ribuan tahun, asalkan kita menjaga eksistensi mereka agar masih tetap ada dan lestari. Mari kita memahami agar lebih peduli.
Lihat kebunku penuh dengan bunga. Kita pasti pernah mendengar lirik lagu berjudul Kebunku. Akan tetapi, pernahkah kalian mendengar cerita mengenai kebun raya yang ada di Indonesia? Sebenarnya, apa itu kebun raya? Kebun raya dalam bahasa Inggrisnya botanical garden adalah suatu area lahan yang ditanami dengan berbagai jenis koleksi tumbuhan dengan tujuan utama untuk keperluan penelitian ilmiah dan konservasi ex situ atau di luar habitat aslinya. Kenapa sih kebun raya itu penting? Tuntutan pembangunan akan selalu berdampak pada perubahan lingkungan. Kebutuhan ruang hijau terbuka merupakan salah satu peran penting kebun raya sebagai paru-paru kota. Kebun raya juga berfungsi sebagai pusat penelitian dan konservasi tumbuhan langka yang hampir punah di Indonesia. Oleh sebab itu, kebun raya disebut sebagai benteng terakhir penyelamatan flora Nusantara. Selain untuk penelitian dan konservasi, kebun raya juga kerap kali digunakan sebagai sarana pendidikan, objek pariwisata, dan jasa lingkungan bagi pengunjung. Di Indonesia, pengembangan dan pengelolaan kebun raya sepenuhnya berada di bawah otoritas ilmiah Lembaga Ilmu Pengetahuan Indonesia atau LIPI. Salah satunya merupakan kebun raya tertua di Indonesia yang saat ini telah berusia 200 tahun, yaitu Kebun Raya Bogor yang didirikan pada tahun 1817 oleh C.G.C. Rainward. Lebih dari 40 jenis tumbuhan dari benua Amerika, Eropa, Afrika, dan Asia didatangkan dan dibudidayakan di Kebun Raya Bogor. Di antaranya kelapa sawit, kopi, karet, dan coklat yang kemudian diteliti dan disebarkan ke seluruh wilayah Nusantara, bahkan Asia. Selain Kebun Raya Bogor, masih ada kebun raya yang lain di Indonesia yang berada di bawah naungan LIPI, yaitu Kebun Raya Cibodas, Kebun Raya Purwodadi, dan Kebun Raya Bali. Keindahan mekarnya bunga sakura yang berasal dari negeri Jepang dapat kita nikmati di Kebun Raya Cibodas, Cianjur, Jawa Barat. Kebun Raya Purwodadi menyajikan keindahan tumbuhan iklim kering dengan pemandangan daun berguguran yang eksotis. Dan Kebun Raya Eka Karya Bali yang memiliki lanskap indah dengan koleksi begonia terlengkap di Asia Tenggara juga dapat menjadi salah satu destinasi wisata yang tidak boleh dilewatkan apabila berkunjung ke Pulau Dewata. Kebun Raya Bogor juga menjadi pendamping untuk pembangunan kebun raya daerah yang tersebar di seluruh Indonesia agar dapat mengembangkan koleksi tanaman dan tumbuhan lokal dengan menyesuaikan iklim dan kondisi lingkungan daerahnya masing-masing. Saat ini telah ada 32 kebun raya daerah yang tersebar di seluruh Indonesia. Penambahan kebun raya di Indonesia diharapkan dapat menambah kesadaran kita sebagai generasi muda untuk lebih mengenal flora Nusantara dan juga menjaga serta melestarikan alam dan lingkungan. Yuk kita ke kebun raya.
Sejak tahun 2015, Lembaga Ilmu Pengetahuan Indonesia atau disingkat dengan LIPI membangun sebuah kawasan terintegrasi dan zero waste sebagai etalase hasil penelitian, khususnya di bidang ilmu pengetahuan hayati. Kawasan yang terletak di Cibinong Science Center Botanic Garden, Kabupaten Bogor ini merupakan kawasan yang dibangun dari program BioVillage, salah satu program unggulan LIPI dengan fungsi sebagai kawasan konservasi, penghijauan kota, sains dan teknologi, serta wisata lingkungan.
Banyak teknologi dan hasil penelitian di LIPI yang tidak dapat dimanfaatkan oleh masyarakat. Oleh karena itu, program BioVillage ini salah satu upaya dari LIPI untuk dapat lebih mendekatkan hasil-hasil penelitian LIPI menjadi dekat dengan masyarakat, sehingga masyarakat merasa bahwa teknologi itu memang perlu dimanfaatkan dan dapat memecahkan masalah masyarakat. Jadi dengan cara senang, dengan cara bahagia, bisa sekaligus rekreasi atau wisata di wilayah BioVillage, tapi bisa juga mendapatkan wawasan dan teknologi yang bisa mereka dapatkan untuk memecahkan masalah mereka.
BioVillage merupakan etalase paket teknologi yang siap diadopsi oleh masyarakat. Paket teknologi ini saling terintegrasi, ramah lingkungan, dan zero waste, yang meliputi teknologi pertanian, peternakan, pengolahan susu, pengolahan limbah pertanian, dan lain-lain.
Total area biovillage di kawasan Cibinong Science Center dan Botanic Garden ini adalah sekitar 189 hektar. Meliputi perkantoran, yaitu Pusat Penelitian Bioteknologi, Pusat Penelitian Biologi, Pusat Penelitian Biomaterial, Pusat Penelitian Limnologi, dan ada pula Pusat Pendidikan dan Pelatihan Peneliti, serta Pusat Inovasi. Kemudian, kebun konservasi yang terdiri dari ekopark dan kebun koleksi plasma nutfah buah Nusantara. Di dalam kebun koleksi plasma nutfah buah Nusantara, kita dapat menemukan berbagai jenis buah Nusantara, antara lain alpukat, pisang, belimbing, sukun, rambutan, manggis, dan beberapa di antaranya yang sudah mulai punah, antara lain ada gandaria, ada kemang, dan menteng. Serta kebun percobaan, demonstrasi plot, dan showcase hasil penelitian yang dibagi dalam 3 zona berdasarkan lokasinya.
Sejatinya program unggulan BioVillage ini telah diinisiasi sejak tahun 2015 lalu.
Zona 1 mewakili pertanian dan peternakan terpadu, kemudian kebun plasma nutfah buah, taman kupu-kupu, dan pupuk organik hayati.
Zona 1 merupakan showcase untuk pertanian terpadu yang terdiri dari pemuliaan dan pengelolaan sapi untuk menghasilkan bibit sapi unggul, pengelolaan sapi perah, unit pengolahan pakan awetan, unit pengolahan susu, serta pengolahan limbah kotoran ternak untuk menghasilkan biogas dan kompos. Selain itu terdapat koleksi plasma nutfah, rumput untuk pakan, dan buah-buahan Nusantara yang dibudidayakan secara tumpang sari. Terdapat juga koleksi sumber karbohidrat untuk mendukung ketahanan pangan. Kegiatan lainnya adalah budidaya sayuran dan padi organik dengan menggunakan pupuk organik berbasis mikroba unggul. Pupuk organik ini diaplikasikan di seluruh areal kebun kawasan BioVillage sebagai pengganti pupuk kimia. Limbah padi yang berupa jerami dimanfaatkan sebagai bahan baku media budidaya jamur pangan. Pada akhir produksi, media jamur bekas digunakan kembali dalam pembuatan kompos. Dari koleksi-koleksi tersebut, dibudidayakan pula lebah madu lokal yang bermanfaat untuk menghasilkan propolis. Selain berfungsi untuk kesehatan, lebah madu ini penting untuk membantu proses penyerbukan tanaman. Di dalam zona ini juga terdapat taman koleksi kupu-kupu dan oposum layang yang bermanfaat sebagai area edukasi untuk memperkaya pengetahuan.
Kemudian zona 2 merupakan kawasan untuk produksi.
Zona selanjutnya adalah zona 2 yang merupakan zona produksi untuk menampilkan teknologi perbanyakan bibit tanaman jati, tanaman penghijauan, serta kehutanan, padi, ubi kayu, dan rumput untuk pakan ternak.
Dan di zona 3 merupakan zona untuk konservasi dan pengolahan limbah.
Kawasan konservasi Ex Situ Eco Park merupakan pengembangan dari Kebun Raya Bogor untuk menampilkan tumbuhan yang mewakili ekoregian Indonesia. Selain itu, di zona ini ditampilkan percontohan agroforestry jati yang ditumpang sarikan dengan tanaman pertanian seperti padi dan garut. Terdapat pula area kebun avokat unggul yang dapat berbunga sepanjang tahun serta sebagian kebun koleksi pisang. Salah satu kegiatan penting lainnya yang berada dalam zona 3 adalah yang terkait dengan biocyclofarming, yaitu pengelolaan dan pengolahan sampah yang berasal dari kegiatan-kegiatan program BioVillage dan di kawasan Cibinong Science Center pada umumnya. Sampah-sampah yang telah dikumpulkan dibagi dalam kelompok organik, kertas, dan plastik. Sampah organik yang meliputi serasah daun, sisa makanan, limbah pertanian, kotoran sapi, diolah menjadi kompos dan pupuk organik cair. Limbah lainnya berupa kertas dan plastik diolah menjadi produk bermanfaat yang bernilai ekonomi. Misalnya pengembangan kafe organik dan toko serta tempat diklat untuk publik.
Konsep BioVillage LIPI ini diharapkan dapat direplikasi di daerah. Oleh karena itu, kami sangat terbuka apabila masyarakat ingin mempelajari lebih lanjut mengenai konsep dan pakai teknologi yang ditampilkan di kawasan BioVillage ini.
Pernahkah kalian menyadari berapa banyak jumlah plastik sekali pakai yang kita gunakan setiap harinya? Mulai dari tas belanja plastik, sedotan plastik, kemasan makanan dan minuman, serta masih banyak lagi produk berbahan dasar plastik yang lainnya. Lalu, kemanakah semua sampah plastik yang kita gunakan itu berakhir? Plastik, material berkarakter ringan dan tahan lama ini pasti akrab dijumpai dalam keseharian kita. Selain praktis, plastik sangat digemari karena harganya yang relatif murah dan mudah didapatkan. Namun sifatnya yang tidak mudah rusak membuat plastik sulit terurai sehingga tidak ramah lingkungan. Sebagian plastik berasal dari material alami, seperti minyak bumi dan karet yang dimodifikasi secara kimia. Dengan mengurangi pemakaian tas plastik, berarti kita juga mengurangi konsumsi plastik. Membutuhkan waktu puluhan hingga ratusan tahun. Bisofenol A atau BPA dan phthalate atau DEHP merupakan zat yang terkandung di dalam plastik. Berbagai penelitian menyatakan bahwa kedua zat kimia ini berdampak buruk terhadap kesehatan karena berpotensi merusak hormon dan sistem reproduksi. Bahkan bersifat karsinogenik apabila terakumulasi secara terus-menerus di dalam tubuh. Meningkatnya produksi plastik global, jumlah plastik yang terbuang ke laut juga akan jauh lebih besar. Hewan laut yang menjadi bagian dari rantai makanan akan menelan plastik termasuk partikel racun di dalamnya, yang kemudian pada akhirnya kembali ke atas piring yang kita makan. Kita memang tidak dapat menghentikan masifnya produksi plastik, karena dibutuhkan peran serta dari semua pihak, termasuk pemerintah selaku pembuat kebijakan. Lalu, apa yang dapat kita lakukan? Semua orang dapat menerapkan prinsip 3R dalam keseharian. Cara termudah adalah dengan mengurangi penggunaan produk plastik sekali pakai, misalnya mengganti kantong plastik dengan tas belanja. Jumlah pemakaian dan pencemaran lingkungan akibat limbah plastik nyatanya kian hari makin bertambah banyak. Bisakah kalian membayangkan bagaimana nasib bumi beberapa tahun mendatang apabila kondisi ini terus-menerus terjadi? Pilihannya ada pada kita. Satu perubahan kecil dapat berdampak besar. Maka, mulailah dari diri kita sendiri, mulai saat ini.
Ladies and gentlemen, welcome back to the international webinar on Indonesian Biodiversity, Wednesday, September 16, 2020. First, let me read the rules of the webinar. Please the committee to display the rules. First, attendees must ensure their device meets the system requirements for Zoom and have stable internet or sufficient prepaid data package for smooth connection. Attendees are required to join Zoom webinar. Unfortunately, there is some connection error. However, step up the following agenda, our last session of presentation with the topic of mainstreaming bioeconomy, bio-based economy of sustainable biodiversity and bioresource for better future will be moderated by Ms. Anggia. And for the session, I will kindly invite Ms. Anggia Prasetyo Putri as the moderator. A little bit about Ms. Anggia. She is researcher in Research Center for Biotechnology LIPI. She graduated from Institute for Molecular Bioscience, the University of Queensland, Australia, for her doctoral degree. In 2019, she received Candidate Development Award Travel Grant from UQ Graduate School to attend summer school in Liverpool, UK. Ms. Anggia, please, the floor is yours.
Okay, thank you very much, Mbak Renata. Can everyone hear me okay? Good afternoon, everyone, and welcome to session 3 of this webinar. And so we've learned about different aspects of biodiversity today, from the conservation point of view to the different bioprospecting approaches that can be taken to optimally tap the potential of our biodiversity. And so now in this session, as has been mentioned before, we will move on to a different perspective, which is the economic point of view. So we will have speakers this session who will enlighten us all on the topic of bioeconomy, specifically on ensuring sustainable biodiversity and bioresources for ensuring our better future. So without further ado, I will first introduce everyone to our first speaker. So our first speaker would be Dr. Jacob Phelps. He is a senior lecturer at Lancaster Environmental Center, Lancaster University. Oh, excuse me, my mistake. So apologies. So our first speaker would be Arif Ansari Yusuf from Department of Economics, Pelayanan University. So Professor Arif has extensive research interests, especially in economic development, modeling, and interlinkage between economy and the environment. And he also has extensive professional experiences and is currently the professor of economics, but he has also has been a visiting professor at the Department of International Development at King's College London, and also a honorary senior lecturer at Crawford School of Public Policy of the Australian National University. So Professor Arif, time is yours. Please, you may begin your presentation. Thank you.
Okay, thank you. Can you hear my voice?
Yes.
Let me share my screen. Maybe I would like to say hi to Pak Rudi. Pak Rudi, baik-baik saja, sehat? My close colleague, and then also my new friends, Jacob. So let me begin. So I title my presentations on economic valuations, particularly on Indonesian biodiversity, and so the subtitle is the long way forward. So this is the outline. So it's a bit of overview of my perspective on what is the status of an economic assessment or valuation of biodiversity in Indonesia. So I will start with briefly with what is it, how is it measured, and then maybe because I know that this is not specialist, so I think it's a good chance for me to share from the angle of the economics, what is the economic value we are talking about here, what is the economic valuation, what is it for, and then maybe a little bit of seeing literatures, globally in Indonesia, what do we know so far, and then about the because there are two things. There is ecosystem service and biodiversity. I will explain to you this is not the same thing, and maybe my assessment of the status of Indonesia, and then how can we progress. So first of all, well, the definition of biodiversity itself sometimes confusing for some people, especially if it's multidisciplinary, because when we talk about the biodiversity, it is very specific. So we stress the importance of the diversity itself. So we are talking about the diversity, not the resources itself, but how diverse it is, how rich it is in terms of the variability of the system. Either it is the diversity within species, between species, or even ecosystem within ecosystem. So this is, we are talking about diversity if we want to be accurate. And I know this is very difficult concept sometimes, and sometimes it is quite abstract. And this is how normally how biodiversity measures in the literature. I'm not pretending I know this, because this is mostly specialized for some biologists. But what I know is this is measuring the diversity of each of the level of the diversity. And sometimes it's not easy when we want to convert this into a valuation story. So this will be the biggest challenge that I will mention a bit later. So, however, we all know that in terms of diversity itself, Indonesia is the richest country, not among the richest. I think the richest country in the world in terms of the biodiversity. This is at least what the CBD in 2001 at least says. Okay, now let me begin with what is exactly economic value. I'm going to stress about the difference between price versus values. So, as we all know, I think most of the economies in the world now, I think 99%, use market system in the way they organize the economy. So since the fall of the communism, at least 99% of market mechanism which rely on market prices is used in most of the country that we know, yeah, except a few like North Korea, somewhere else. Okay. Yet, however, when we look at the, that market has the pure market mechanism. Okay, has a very minimum role for government, and this system is used in 99% of the countries in the world. Yet there is this what is called Wagner's law. Wagner's law says when the country is getting richer, I mean the richer your country you are, the more you rely on government. So this is kind of anomaly. Why 99% of the world is relying on market price and market system? Why the more you reach, the more you educated, the more you know better, actually, you ask government to play more in your economy. So this must be something else then, yeah? And it's fact, it's true, because maybe I would like to, I hope that you can hear this.
Too much and for too long, we seem to have surrendered personal excellence and community value in the mere accumulation of material things. Our gross national product now is over $800 billion a year. But that gross national product, if we judge the United States of America by that gross national product counts air pollution and cigarette advertising and ambulances to clear our highways of carnage. It counts special locks for our doors and the jails for the people who break them. It counts the destruction of the redwoods and the loss of our natural wonders in chaotic sprawl. It counts napalm, and it counts nuclear warheads and armored cars for the police to fight the riots in our cities. It counts Whitman's rifles and Speck's knives and the television programs which glorify violence in order to sell toys to our children. Yet the gross national product does not allow for the health of our children, the quality of their education, or the joy of their play. It does not include the beauty of our poetry, or the strength of our marriages, the intelligence of our public debate, or the integrity of our public officials. It measures neither our wit nor our courage, neither our wisdom nor our learning, neither our compassion nor our devotion to our country. It measures everything in short, except that which makes life worthwhile. And it can tell us everything about America.
Okay, so, well, the price is different than value. So basically, price, what I mean by price is the market price. The value is something else, and it's not exactly the same thing. I normally, before I begin my class in environmental economics, normally I did a little survey to all the students and asked them, can you mention 5 things that you think the most valuable for you in your life? And I write them on the board, all of them, and it always turned out that most of the things that these students put value, high value on it, we cannot get it from the department store. So most of the things that people value actually is not marketable. It's not market. We cannot buy it just nearby. It's something that market cannot provide. Okay. So for example here, what is the price, market price, yeah, of the service that forest provide to protect us from flash flood? Basically none, zero. Yeah. However, what is the value? Of course, yeah, many life loss avoided. That's the value if you have flash flood. It is happened in Garut in 2016, for example, 34 died. The closest example of what you are here is what is the price of upstream ecosystem around Bogor area, yeah? To regulate water in Jakarta. What is the price, the market price of this? Nothing, zero. But what is value? Trillion rupiahs of economic loss that could have been avoided. So price is different than value, but market price itself alone cannot detect this price. So how can we rely on economic system that disregard most things that we value? Yeah? So this must be something wrong. So maybe that's the reason why there's Wagner's law. Why the richer country you are, the more educated you are, the more you know better, then you ask government to place more role in the economy. And that's the same thing for environment. So basically, economic valuation is the use of economic analysis to disclose the invisible, the things of which its value is invisible to us, the thing that doesn't have market price. For what? to help us understand trade-off better. Yeah, because trade-off is everywhere, resource is constrained. So sometimes there is a misunderstanding. So economic valuation is not just simply to put dollar value to anything that we respect, to life, to whatever, right? No, it's not that. And also many misunderstanding. It's not also to neo-liberalize ecosystem. No, this is just to help understand trade-off better, because most of the thing that we value like that doesn't have market price. Okay, otherwise then it's very difficult. And how do we do exactly economic valuation in practice? Well, it's not that complicated actually in principle, because we just simply try to find out either from primary survey or from secondary data to find out if there is some kind of indicator of happiness of people, and that happiness is a function of income that they earn, this is in dollar, and some kind of other non-marketed commodities like environment at certain level, let's say N0. And if we want to improve that N, that nature, for example, from N0 to N1, then you have to take away some money that you have. Okay, you got the new N1, but you take away some money that you have, and your happiness has to be the same between these two conditions. Then this WTP is actually the value, the economic value that we put on this improvement from state of the environment N0 to N1, as simple as that. So all this economic valuation technique is basically to find this WTP. So, however, it's not that simple actually, because there are so many details, yeah, in terms of the ethics, in terms of the ideology, in terms of the school of thought, yeah, in terms of how this should be measured. So basically, economic aspect is more relying on the preference-based approach. So this is basically the preference-based approach. So we use this preference-based approach. So in that case, then we think that the economic value, but we try to be broadly defined the economic value. So we have use value, we have non-use value, and then we use value, we have direct use value too, and then indirect use value, we have option value, direct use value, yeah? And then also existent or altruism value. And then this is how this is the various economic valuation method that we normally have. This is the reserves of the tools that economics use to do this valuation. I'm not going to go into the detail because of the limited time, but this is actually what we have. So we are here in the economics. Okay. So what about biodiversity? Well, this economic valuation, needless to say, is a human-centric concept. So it's an anthropogenic concept. It means that to be properly valued, biodiversity first needs to be translated into services of ecosystem, services of the ecosystem that has value to us. Okay, not marketed to us, but that has value to us. For example, so you have to connect it to the socio-economic system in the red zone here. So you have human well-being, you have to have the benefit, yeah, and this is the biodiversity. So biodiversity does not stand alone and itself give value to human. It has to go through this ecological process. First, for example, in terms of nutrient recycling, and then produce biodiversity, the enhancement of the efficiency of the ecological process. Then that will goes to socioeconomic system. Or, for example, increase the diversity in the pollinator species. So increase the pollination service of the environment, that goes to the values. This is what we values. And also diversity to create more habitat for insect. And then this is species richness. Now this is more direct, for example, species richness, people like bird, game watching, or identifying different species of butterflies. Yeah. So this goes to this ecosystem service. So this is the value that matters to human when they have of use. Okay, so you have to translate that first into these services, not only biodiversity itself. Okay. However, we have to economists not also acknowledge that part of the use that we define, there's something called non-use values. So this is a very, so don't worry. So don't worry that when we talk about biodiversity, it means something that is useful for us, but that use is very broadly defined. So we have direct use. For example, biodiversity can improve recreational experience. Okay, so that's direct use, but also indirect use. This is already non-marketed. This is already non-marketed. We don't have market price for this one. This one maybe have market price, but starting from this bold box one, normally we don't have market price. So what market price value? Normally only this one. Starting from this to this to this, no one. So biodiversity ensure more resilient in ecosystem, for example, to provide benefit to ecosystem service, forest function, flood protection, landslide protection. So it's not non-market. Option, species or gene that are poorly understood today may provide valuable information in the future. So this is also biodiversity function. Existence, some people think something that exists are valuable to them. Even though never see the thing, but just to give them comfort that knowing that they exist. Okay, so they have value to it. They're willing to pay something like that. Altruism, knowing that I don't know, I never seen the thing, but I know that I can give it to my children. Okay, so that also has value. People are willing to pay. So this is very broadly defined. Well, in a way, this economic valuation is not just simply putting numbers and disclose the numbers and then announce the number to media, to public, or to economic journal. No, it is a tool, and that tool has to be useful. If there's no use of it, we don't really need to discuss. So this valuation is a tool that more often useful, because basically, in principle, it discloses the invisibility of the nature's values, which is important in the resource concerns work. So this EV basically inform regulation, sometimes it help enforce justice. For example, this is the example. So that inform justice. So when the Gulf of Mexico oil spill in 2010, and then the trustee of the Gulf Natural Resource in US and the NOAA in US actually assign some team to do economic valuation so they can use it as evidence at court. So in economic valuation help justice, enforce justice, I mean. So, and then help cost benefit analysis. So basically make the world a better place, I think. So this is something important and useful. And this is an example of the most widely cited the ecosystem service value of the world by my colleague here from the NU, Bob Costanza, in 2014, he updated the value. So you can see here, but maybe I just show you the Indonesian part because I invited him to come to Jakarta and then to make the calculation of this table to Indonesia, and then he kindly enough to do this for Indonesia, and then he saw that, for example, in the baseline in 2011, all of this natural environment give ecosystem services. This is services, not necessarily biodiversity. Biodiversity may be there implicitly, yet it's actually the service is 3 times the GDP of that particular year. So when you have GDP around 540 billion dollar in 2011, the ecosystem service is basically 1,600 billion dollar. So 3 times of the GDP. So basically all this ecosystem service is way more than what is measured by the market. Okay? So just a proof. How about the valuation of biodiversity? So, well, this is from what I learned in the literature. So economic, if we want to be very limited or specific in terms of the valuation of the diversity itself, then this is the status in the literatures. Actually, not that many, but they are, right? They are. So from various region, from various type of habitat, with various type of techniques, and also the various level of the biodiversity. So they exist, but not, of course, not as many as the valuation that actually measure the ecosystem services, in terms of the services, not the diversity. This is particular on the diversity. So they exist, and I just give you a little bit of head-ups atau example of what do economists do actually when try to evaluate. So basically, this is one of the primary survey. For example, so people are asked if we have a project of reintroducing the beaver in Africa. This is Africa, yeah, and then cost considerable amount of money. Will they pay if the tax increase by this much per year, for example? And then from this information of thousands of respondents, then we can calculate with a statistical system what is the value of this introducing the beaver, for example. This is one of the example. Another one, and this is called contingent valuation, but there are other one, for example, using the technical choice modeling. In the choice modeling, people are given options, yeah, option of several level of diversity, for example. This is, do you pick policy level 1, policy level 2, or do nothing? And then you have also the price that you have to pay with that options, and then you have to trade off between this species familiarity of wildlife, okay, rare or not rare, so basically diversity. Yeah? So, and then what the respondent pick will be different, yeah, according to characteristic of that respondent. And then from that information, from that thousand of information, then we can do statistical analysis to calculate how much actually people are willing to pay for protecting for this diversity. So this is just an example. Okay, my last 2 slides. How about Indonesia? So basically, in Indonesia, as far as my experience, yeah, in reading the literatures, and then also I have quite years of being active in the environmental economics research, at least supervision, yeah, with my organization in they are quite big organization. Yeah? So actually, there are quite many studies on economic valuation of ecosystem services on Indonesia, not in Indonesia, on Indonesia. However, my note is that not many good ones, actually. Good one mean published in good journal, yeah, done by international academics. So we need more good primary studies done by national academics. So I think this is important because we need that capacity building if we want to translate better these valuations into policy. Yeah. So capacity building, especially quality-wise, yeah. There are many environmental economic valuation on Indonesia by Indonesian, but the good one is not many. So need capacity building. If we are more specific in terms of diversity, then we have a bit of problem because difficult to find specific valuation on Indonesian biodiversity, like very sharp, narrowly defined diversity. So I think I only found one study cited in the literature by Barkowski, yeah, in 2015. And then in Indonesia, I found for my experience in engage with government or policymaker, cost benefit analysis is actually, not to mention economic valuation, it's not really a normal thing to do in Indonesian policy or business decisions. So if you don't really do much cost benefit analysis, then economic valuation have not that much role. Yeah. And well, I have to admit there are some progress in legislations. Yeah, I think economic valuation, especially green economy and similar kind of things like that, already been explicit in certain legislation. And also there are efforts because I'm actually quite involved with corruption commissions, yeah, to try to insert this economic valuation into damage assessment of corruption, so people can be charged if they do corruption and then damage the environment. It's quite active about this, and then also in the Ministry of Justice also they have been quite active about this damage assessment. So I think Indonesia damage assessment analysis by the court or by legal system has already started to adopt certain economic valuation. However, it's not really in the direct policy regulation or business decisions. Yeah, maybe Pak Rudy maybe can explain a bit later. And then recently there are omnibus law that I think is a setback for this, yeah, because the environment, not only from the valuation but the environmental aspect also is a bit of setback. So how should we go from here? Well, I think what still we need to do is we should train more and better academics on economic valuation, and then try to publish them in good academic journals. So that's a part of the capacity building, because of the peer review process will make you better in terms of how you do this. Yeah? But however, we have to link this research into most urgent policy agenda. So we always put our economic valuation work as close as possible to the policy arena. Yeah. For example, pick something urgent. Pick something people are debating or discussing. For example, what about economic valuation related to moving capital city? Has anyone done thing or something like this? Done something like this? I'm not so sure. I don't think so. So do economic valuation related to the moving capital city? That's a big issue. Yeah, there's something. So try to find something like that, some relevant something like that. And then also, because as you can see from my explanation before, the use value has so many elements, and then each element of the use maybe need one specific studies. Yeah? You cannot do economic valuation, primary economic valuation, I mean, yeah, to find the economic value of one thing, all the use with one research paper. No way. I saw that common in Indonesia. We want to estimate the value of certain resources, the total economic value of certain resources with one paper. Okay, that's something we have to avoid, because then you lost the accuracy. You lost the accuracy, then you lost the relevance. Yeah. So that's why we need to be selective in what value of the biodiversity to be researched. Things of urgency. Okay, I think Indonesia has many issues that is specific to our context. Yeah. For example, instead of estimating something abstract like existence value, why don't we identify the direct use first in terms of the commercialization potentials? Yeah, I'm sure it's not been, like investigated thoroughly. Yeah, or something that has high social value. For example, something related to medicine. Yeah, something related to tourism, or even something tourism to mining, because that's the sector of the economy. I just want to share my colleague, Ayen Choa from University of Tadulako, is my colleague at ALMI, that was featured recently in the BBC about her research on the rare plants that actually can produce nickel. This is very, like rare, and then we are actually rich in this type of plant. So maybe something like this as an example. We do research on this, yeah, research on the diversity, on the plants itself, and also on the economic valuation of this very, exciting new things, yeah. So I think that's how I want to share about the economic valuation of biodiversity. I hope can can induce and encourage some more discussion later. Thank you. I would like to stop here.
Okay. Thank you very much, Professor Ari, for that very insightful presentation. I think it will provide a really good background and has given a nice overview of what's involved with biodiversity evaluation. And I'm sure there will be questions that people will be asking. And for that, I would like to remind everyone, should you have any questions in this session, please utilize the Q&A and chat forums to write your questions there. And at the end of the session, then we'll have a discussion with all of the presenters. So we will move on now to our next speaker. And so we will have now Dr. Jacob Phelps, a senior lecturer at Lancaster University. And he has quite a extensive research interest on addressing the governance of key environmental challenges in different aspects, including illegal wildlife trade, Indonesian peatland wildfires, and also from the policy, legal, and governance dimensions as well. And I think he's also has worked with Indonesian researchers and government on this area as well. So I'm sure this will be a very insightful presentation as well regarding wildlife trade and ecosystem services. So Dr. Jacob Phelps, time is yours. You may begin your presentation. Thank you.
Thank you very much, Dr. Anggia, and hello, Prof. Arif and Dr. Arifin and all the colleagues joining us. Let me just share my... Has that come through?
Yes, we can see.
Great, thank you very much. And thank you for the invitation. I'm going to be talking then, continuing this discussion about kind of bioeconomy and thinking about the economic values of biodiversity. I'd like to propose that we need to think about the value of biodiversity moving forward, as we've been discussing, its great potential, including its economic potential. But not only do we need to look forward, we also need to look back at the cases, as Prof. Arif mentioned, where quite often we harm the environment, particularly biodiversity, in really extreme ways, and whether economic tools can also help us there in terms of building a more sustainable economy. Because in today's conference, we're talking about value of biodiversity in a number of different, right? We want it in different aspects. We want to recognize its enormity. We want to recognize that it provides direct benefits, but also indirect benefits. And we want to think about different ways in which we can use economic tools to help us understand that. But a lot of this is, again, looking forward. It's thinking about these tremendous biodiversity and natural capital stocks and ecosystem services that we have in seas and on land, and particularly biodiversity, species, and its different levels of organization. And all of these have tremendous values, both economic
Looks like we may have some technical issues, so apologies for this. And I'm sure the committee is working very hard to ensure that we can get Dr. Jacob Phelps back for his presentation. So could I please ask you all to be a bit patient with this? Thank you. So again, I'd like to remind everyone, if you have any questions, please do so through the Q&A and chat forums. And since this is also live on YouTube, those of you who are paying attention from the YouTube channel, you can also put in your questions via the chat forum as well at YouTube channel. So and also a few notes from the committee. Everyone who has attended this webinar today and has filled in the attendance form, the e-certificates will be sent to you via email that you have filled out approximately 14 days from today's date. So make sure you do fill in the attendance form. And yes. So, and remember, we still have one more speaker after Dr. Jacob Phelps, so please stay tuned. So I think maybe while we're still waiting, I do have a question to Professor Arif, if I may. So you were talking about that biodiversity has to be translated into services that has value to us humans. So would you think that would then be quite a subjective approach as to which biodiversity is given more value than others? And I'm sure there will be quite a few considerations in terms of deciding which part of a forest, for example, that has more value than others, or what you were saying about utilizing natural resources without considering for the damages, and that of course will then be detrimental to the value of that biodiversity. So could you please comment on that? Thank you.
Well, yes, I think there are two considerations. Yeah, one is the urgency of the problem itself, and the urgency can coming from two parts. Yeah, the first is the stress to the environment itself, yeah? And also the stress to the economic system itself. So if it's very useful, if biodiversity is useful, then you have to think of that as priority. So that's from the urgency of the problem itself. And the second one is the valuation itself, good one, yeah, good economic valuation study itself is not need resources. Yeah, need resources. Need resources. What I mean is, good valuation study, for example, has to have a minimum standard of people as a respondent to be sample. Yeah. So in that case, then those we cannot just do as if that valuation is something easy to do. So in that case, then we have to select two. Yeah, the issue, yeah, the most appropriate issue. So I think Jacob already mentioned before that one of the consideration is the urgency of the stress. Which one is the most stress to the environment? But also economic. But what I add in the context of my presentation is that we may have certain kind of biodiversity use, yeah, that if we can take advantage of it in terms of conserving, yeah, it actually can generate economic alternative benefit, yeah, to us that we never think before. For example, nickel, yeah, as coming from plants, yeah, or even something else, or even tourism that I don't know. No one know what is the global preference of tourism. Yeah, actually maybe it can be go hand in hand with preserving some forests that has rich biodiversity. So that's what I'm talking about. So we need to think in the two considerations. First is the urgency of the problem that coming from two, environment and the economic aspect. And the second one is the resources that we have to do these economic valuations. Maybe that's my question, my answers.
I see. Thank you very much for that. And now it looks like we have Jacob back. So I think we will continue with his presentation. Hi, Jacob.
Hi, my sincere apologies.
It's okay.
The British infrastructure is just as bad as it is everywhere else. Don't let anyone tell you otherwise.
Okay, sure. So please resume your talk. I'm sure everyone's looking forward to it. Thank you.
Thank you, and apologies again. What I was highlighting, and I think Prof. Arif was just talking about this, is that there's huge gains that we can make, and the bioeconomy is really focused on those huge gains. You know, that might be, he was just mentioning ecotourism or bioprospecting we heard about earlier today, or novel mechanisms for synthesizing compounds, like you were talking about nickel, or new wild crops, or integration of genes from wild crop relatives into commercial crops. So there's huge potential, and that's important. But what we focus on perhaps less is that we also need to, in a responsible bioeconomy, recognize losses. And so the big question that I have in my current work is, doesn't biodiversity still have value, economic value, but also non-economic, non-financial values to us when it's harmed? We care about biodiversity when it's out there, but when it's injured, surely it still has importance to us. And it has to, because we know that even though we have a lot of protected areas and a lot of biodiversity, we also know that a lot of those areas are still threatened. We know that we have huge areas of degraded land. There's an estimate released last year from the IPBES report that something like 2/5 of the world population is directly affected by ecosystem services on degraded lands. And of course, huge amounts of pollution, including the hot topic of plastic waste. All of these issues are part of our normal economic, our current, mainstream business as usual economic model. A bioeconomy is interested in alternative, innovative, more sustainable ways of building our economy that don't go along this route. But surely that responsible bioeconomy must also be concerned with the losses caused by our regular economic model. So I'd just like to give an example from wildlife trade, which is one of the main topics that I'm interested in my research. And if we think about, for example, the pangolin. So this is a nocturnal scaly mammal, right? And the Sunda pangolin, Manis javanica, native across much of Indonesia, and it's traded for its meat, but also traded internationally for its scales, which are incorrectly believed to be medicinal. And there's a huge trade, and this is a critically endangered species. Now, there are potential bioeconomy gains. Maybe someone will pay us to protect this species. Maybe in some sort of ecosystem accounting, we're going to try and of course, Prof. Arif was talking about the different methods that we might use to try and do some accounting of the ecosystem services that it provides, such as perhaps pest control. Maybe also there are benefits to us for Indonesia being recognized globally, not only for its unique biodiversity, but for protecting that globally significant biodiversity. So there's a positive reputational image. Maybe there's also ecotourism here of people wanting to visit regions that have rare and unusual and exotic animals. Those are the gains. But of course, if we're going to extend this economics argument, we know that any accounting ledger doesn't just have gains, it also accounts for its losses. And so if we think about this species not as just something that might have economic potential, but also as a species that's heavily traded for the illegal wildlife trade. Here we see, this is a photo of a confiscation several years ago, 5 tons of frozen pangolin for the illegal wildlife trade. But in these types of cases, if the animals are alive, we might have to care and rehabilitate them. If they're dead like this, we need to figure out, what do you do with 5 tons of pangolin that has to be destroyed? But also those 5 tons of dead pangolin, that has reduced the wild populations of a critically endangered protected species. So there's losses here. Also, we've derived benefits, ecosystem services from those species that have now been lost, including one of the interesting things about pangolins, and colleagues at LIPI know this, Prof. Gono in particular, very acutely. We don't know very much about this species. It's a very understudied, secretive species. There's a lot of scientific discovery yet out there, as there are with so many unusual species in Indonesia. We have lost that opportunity to do that research, to do that investigation. And also, let's say there's reputational harm. Can Indonesia protect its species? What sort of signal does this send as Indonesian citizens into the world at large? So if we're trying to think about an economy, it's not just about what we can get from biodiversity, it's also accounting for what we might lose. Now, this isn't just about doing math, right? We don't just care about coming up with numbers and trying to figure out values. These numbers have to have a real life. So where do we see these gains and losses? Where are they manifest? Well, in the context of bioeconomy gains, we might have new markets, maybe new markets for a new pharmaceutical drug. Or we might have incentive payments, again, someone willing to pay us to do conservation. Or new biodiversity-based products. Or in other cases, perhaps we're going to be doing using new techniques so that we can use biodiversity, for example, better fishing techniques that are more sustainable, that allow us to meet our human needs in ways that don't harm the environment quite so much. So these are the types of examples where that math, those economic gains are actually manifest into institutions. But where are the losses represented? Well, there are at least broadly 3 different categories that I can think of. One is, of course, environmental taxes. Let's say if you pollute, we tax you for that pollution in order to try and change your behavior to pollute less. Or maybe we charge you for using plastic bags. That's a campaign here in the UK. But we can also use, and Prof. Arif mentioned this, we can also use law, right? We also have criminal sanctions. If you illegally trade wildlife, we bring criminal charges against you and try and fine you for causing that environmental harm in an attempt to try and correct that distortion that happened when you've caused environmental harm. But there are also, and this is what I'd like to focus on specifically, is civil sanctions, where we hold companies, individuals, agencies responsible for the environmental harm that they cause. And I'll explain what I mean by responsible. But if we take a step back, we often use criminal approaches. So this is a case, right, that I mentioned. There was 5 tons of illegal pangolin that was traded. There were 89 animals that were traded in this is a particular criminal case that happened in Medan in 2015. Someone was trading pangolin illegally, and you see there was a really large scale case. The response that we used was by pursuing criminal processes, and the sanction was imprisonment and a fine. And that fine was a 50,000 rupiah, right? So about $3,500 US. Now, that's fine, and that's correcting, and that's a benefit. But the question is, does it represent the losses that we actually experienced in that case? Why, when biodiversity is alive, do we talk about the bioeconomy of something of huge value, lots of opportunities, great optimism? And then all of a sudden, when that biodiversity dies, is illegally traded or illegally mined or deforested or burnt, does it have such low economic value and yet such high public cost? Because we, as taxpayers, as citizens, as conservationists, as researchers, end up being the ones responsible for trying to protect that biodiversity. So what happens when this biodiversity, why does its value change so much? Well, of course, and one of the reasons that I'm working in Indonesia is that Indonesia has more actively than most countries in the world actively challenged that. And it dates back to this case, and this was a criminal case, but also a civil case. So not only perdata, but also not only pidana, but also perdata sort of legal approach. And that was the PT Kalista Alam case in Aceh, which you may well have heard about. This was a case of illegal burning and forest clearing by an oil palm plantation in prime habitat, including for the Sumatran orangutan. So forest was harmed, but also biodiversity was harmed. And in this case, there were criminal charges. Someone went to jail, attempt to go to jail, criminal fines, but also there was almost $20 million found in harm to biodiversity and ecosystem services, and demands that that should be restored. A recognition that there was a cost here. There was not just there was a loss here. And I'm arguing that it's that $20 million figure that is an attempt to recognize harm in the bioeconomy. And that an economy that is actually concerned with biodiversity is actually really interested in also coming up with and understanding the value of this type of harm that occurs. Now, coming up with that figure of 20 million can be really difficult. And Prof. Arif has already talked about some different methods that we might use to come up with that value, right? We might use a default value. We might come up with a value from the literature that already exists out there and try and use it to say, okay, well, biodiversity is worth $1,000 per hectare. Let's apply that in any case where biodiversity is harmed. But as Prof. Arif mentioned, there's not a lot of good studies, especially for biodiversity available. It can be really hard to find those studies that already exist. Now, we might be able to use market price. We might be able to say, what was the value of those orangutans that were killed in the PT Kalista Alam case? Or what was the value of the market price of those 5 tons of pangolin in the case that I showed you earlier? We can do that, right? We can go to the markets, we can see what it costs, we can talk about how much tax is lost, and that's possible. But as Prof. Arif also said, we need to make sure that we understand the difference between price and value. And in the case of a critically endangered species that we don't have a lot of research on, market price doesn't really show us its value. So maybe we want to go out instead and we want to think about all of the ecosystem services associated with either Bete Kalistalam case or with the pangolin case. And we can do that. We could come up with a list of all of the different values that exist in those cases and try and come up with economic numbers for them. That's possible, but as we also saw in the previous speaker's slides, it's also very complex. There's a lot of science to be done there. So it's an approach, but it's a demanding approach. So there's another approach to think about, which is one that I'm increasingly interested in, and that's the cost of remedies. So if we have a case where there was illegal burning and the forest burnt down, or illegal wildlife was traded, what are the actions that we need to take to fix that? Do we need to reintroduce pangolin? Do we need to restore habitat? Do we need to monitor species? So instead of saying, what is the value of nature, the economic value, let's instead say, hey, LIPI scientists, what are the actions that need to be taken to fix this? You're the researchers, you're the communities who are aware of these species and systems. What do you think we should do? And once we know what the actions are, then we can do economics on figuring out what those actions are. But also, as Prof. Arif also said, value is not just about numbers, economic numbers. It's also about other types of non-financial value. So maybe there are other actions that need to be taken, things like apologies or education or even a public prayer that help us to feel better, to be remedied when harm has occurred. And I'm arguing that a bioeconomy is concerned with all of these elements, not just economic value of intact systems. I'd like to give you an example, recent example of illegal fishing in France. This is a case where there was illegal fishing inside of a protected area, and the people who were responsible for that illegal fishing were charged. But they didn't go out and charge them the cost of the fish that they caught. They didn't say, you caught $10,000 of fish, so we're charging you $10,000. They also didn't say, all of those fish provided all of these ecosystem services and make a list. No, what they said was, you fished illegally in a protected area. The cost of monitoring a protected area is approximately this amount. And they came up with a cost per meter square. And they said, we think you affected approximately this many hectares, this many meters. And so what we really need is more money to better monitor that protected area so that this type of harm never happens again. And so that those fish can replenish, those stocks can be replenished over time. The other thing that they said was they recognized that harm is not just about market prices. They said, wait a minute, when you did that illegal fishing, you also reduced the park's environmental protection mission. You also harmed its reputation and its brand. And those have value. And we can come up with economic numbers, with financial figures that reflect that value. And interestingly enough, they also charged money for that harm to the restaurants that were illegally buying that fish. So this example in France brings us back to the pangolin example that I presented earlier. The question here then is, can civil law help us to recognize the value of harm in a bioeconomy? In that case, remember that I said there were 89 animals that were still alive, 89 live pangolin? Well, someone has to care for those. Someone has to release those back into the wild. That has cost. So what about, remember that I said there were 5 tons? That's approximately 213 individual pangolins was by my calculation that were actually killed. So the population is decreased by over 200 individuals. So what actions are needed to increase the population by 200 individuals back to where it was before harm? What about the scientific losses? What about reputational harm? Well, my argument is that we can use economic and non-economic tools in order to think about appropriate remedies. So in some cases, it might be payments. So we have to pay a rescue center to care for those pangolins, to figure out where to release them, to do the release. But then we need to take actions or maybe make payments so that we can protect habitat or whatever the scientists tell us we need to do to increase the population by 200 animals. Maybe we need to protect, maybe we need to extend protected area. Maybe we need to better monitor, like in the case of the French case that I just showed you. Maybe we need to pay BKSDA in that area so that they can track the recovery over time, because otherwise, who else is going to do that? Maybe a public apology is in order. Maybe the harm that was done to the reputation of the protected area also needs to be recognized. So my point is that there's a lot of different remedies that could be considered, and that these are important not only to fixing environmental harm, but to thinking seriously about what a bioeconomy looks like, because the bioeconomy doesn't only include gains, it also must include for recognizing of losses. And so I think that that's my main takeaway message, is that we want to be very optimistic and positive about the bioeconomy, but we also have to be ready to address hard questions about the very common occurrences when we harm nature. And that's one of the things that I just want to make a quick plug for this project that I'm collaborating on, which is of NGOs and colleagues in LIPI and BKSDA. And these are economists and social scientists and lawyers as well, who are thinking about the value, the economic and non-financial value that happens, particularly from TSL, from illegal wildlife trade. And then also thinking about how can these values be reflected through different economic and potentially different legal instruments. And I invite you to learn more about our project, as well as to consider getting in touch with me if you're interested in this type of work. So I'll end there. Thank you very much.
Okay, thank you very much, Jacob, for that really insightful presentation as well on the different ways to not just, give value to the gains of biodiversity by utilizing it, and also, but also to the losses that can obviously occur from utilizing them as well. And I think that we have already a number of questions in the chat, and but I think before that we should move on to our next speaker. So thank you, Jacob, for your time. So welcome, Bapak Dr. Arifin Rudianto. Welcome, Pak. So we have our speaker. He is the Deputy of Maritime Affairs and Natural Resources, Ministry of National Development Planning. Welcome, Pak. His education history, he went to ITB University for his undergrad and completed his master's in the UK and his PhD in maritime policy in Wollongong, Australia. So we're both Australian alumni, Pak. So at this moment, he has been the Deputy of Maritime Affairs and Natural Resources for the past 3 years. So I'm sure that he will be able to provide us the government's or the national development's perspective on bio-based economy valuation. So time is yours, Pak Arifin Rudianto. Thank you.
Thank you, Moderator, Dr. Andia, distinguished panel, my friend, Professor Arif Ansori, and also Dr. Yakob. First of all, thanks to LIPI for the invitation to join this important webinar to share about information and knowledge on Indonesian biodiversity, conservation, bioprospection, and actual biodiversity. In this case, in this opportunity, I would like to share about biodiversity in national development. It is well known that biodiversity is the variation among the living organisms from different sources, which describe the richness and variety of the life on Earth. And biodiversity consists of 3 levels. Those are ecosystem, species, and genetic. As we have heard that the open-ended working group on the post-2020 Global Biodiversity Framework set the framework vision for 2050. It's a world of living in harmony with nature, where by 2050, biodiversity is valued, conserved, restored, and widely used. Maintaining ecosystem services, sustaining a healthy planet, and delivering benefit essential for all of you. The GBA owns 2020 has 4 long-term goals related to the 2050 vision for biodiversity, and each of those goals has an associated target with 20 target in 2030. In extent, the 2030 mission for this framework was divided into 3 groups of action-oriented targets. Those are reducing threat to biodiversity, meeting people's needs through sustainable use and benefit sharing, and tools and solutions for implementation and mainstreaming. Then, biosphere provides the basis for Earth's natural life support system. In Indonesia, many economic activities depend on biological resources, and there are still many people who depend on the existence of biological resources and biodiversity. Loss of biodiversity has potentially devastating effect on economy, such as water scarcity, increasing crop production, natural disaster, and so on. Thus, sustainable development are the blueprint to achieve a better and more sustainable future for all. Indonesia, as a mega biodiversity country, covers 74 types of ecosystem. In species diversity, flora of Indonesia is about 50% of the total number of flora in the world. 80,000 species of spore plants and around 30 until 40,000 species of seed plant flora. For Indonesia fauna, is about 8,000 species of vertebrate and 1,900 species of butterflies, equal with 10% of the world butterfly species. Furthermore, Indonesia has the highest endemicity of bird, mammal, and reptile fauna species in the world. In genetic level, Indonesian genetic diversity developed from plants, animal, and microbes through conventional or biotechnological cultivation into drug, cosmetic, other plant, and animal variants. The biodiversity has been and continue to be studied, protected, and used sustainably to support economic growth. To ensure the sustainability, Indonesian government constructed the IBSA, Indonesian Biodiversity Strategy and Action Plan 2015-2020. In Indonesia 2045 vision, industrial sector is directed to be a driver of economic growth. One of the efforts is through the development of natural resource processing industry toward acceleration of industrialization and application of technological advance. Therefore, the industrial development gradually implemented in 5 subsectors. Those are food, textile, automotive, electronics, chemical, and pharmaceutical. Indonesia 2020-2024 vision to create a developed Indonesia that is sovereign, independent, with characteristics based on Gotong Royong, our mutual cooperation. The President vision, and directive that comprise human resource development, infrastructure development, simplification of regulation, simplification of bureaucracy, and economic transformation have been translated into 7 development agendas of RPJMN 2020-2024. Talking about the biodiversity, the Midterm Development Plan 2024 consider natural resource carrying capacity as a development constraint for Indonesia midterm development. The document contains policy and strategy of biodiversity management, including bioresources for protection and sustainable use, where sustainable use aspect is interpreted in industrial value added, and it described in priority number 1 for productivity, competitiveness, and reducing poverty. And also described in priority number 3 to ensure the sustainability of ecosystem and cities listed in priority number 6. In detail, our national priority of midterm development 2020-2024 that have strong relation to bio-based economy are in strengthening economic resilience for quality growth. In priority number 1, with priority programs such as water supply, renewable energy, food for consumption, and increase value added. Second, in developing region to reduce inequality and ensure equality. Third, to improving quality human resource and competitiveness in order to reduce poverty and increasing productivity and competitiveness. And the last is priority number 6, building the environment, enhancing disaster resilience and climate change to improve environmental quality, increase climate change and disaster resilience, and create low carbon development. Bio-based economy in national development contribute to support economic, especially reducing poverty and increasing foreign exchange. To support food security and health through develop genetic and germ-bound resources. Biodiversity also as a component of IG target and particular SDGs. Biodiversity also provide benefit both tangible and intangible value, like already mentioned by Dr. Fab and also Prof. Arik. The economic value of biodiversity that presented is only part of the empirical studies and not the total economic value of biodiversity. We concern that Indonesia biodiversity still has a lot of potential economic value and it is still need to be reviewed. In advance, Indonesia have various bioresources products. Bioresources products that have been developed are food, such as staple food and supplement food. In pharmacy industry, herbal medicine was recorded around 7,500 until 9,600 types of plants developed from around 30 to 40,000 species estimated exist in Indonesia. Only around 30 have become commercial products. In renewable energy resources such as bioethanol and blue energy, to produce wood and also non-wood material, provide marine resources, for example, wood, bioactive, medicine, and also livelihood, and provide ecosystem services like water, ecotourism, carbon sequestration, pollinator, and so on. And finally, thank you very much for your question.
Okay, thank you very much, Bapak Dr. Arifin Rudianto, for that really good presentation and overview of what bio-based economy in Indonesia is like and its connection to national development and how everything ties together in terms of making sure the biodiversity is being sustained and also improving national economic development. So now that we have around 30 minutes for question and answer session, which is really good because we do have quite a few questions already in the Q&A forum. And I would like to start with, okay, so there's one question for Dr. Jacob from Indaryani. The question is, how is bioeconomic dealing with poverty in which in practice, there's a tendency to damage the environment, and what variables should be taken when we want to assess the valuation of a certain environment, such as the forest? Thank you.
I think that actually the environmental harm and poverty are greatly linked, because it's often rural and often poor communities who are most directly affected by environmental harm, either because they're directly reliant on provisioning services, on harvesting plants and animals as part of their immediate nutritional need and livelihoods, and or as part of their livelihood, and also because they're often at the front line of the environmental degradation, the erosion, the haze, and so on. And so I think the links are very clear. Making those links, just because the links are clear doesn't mean that they become actualized. And I think that that's one of the things that I'm really interested in. Is it's very nice to come up with values for these things. It's important, but what do we actually, what is the action that follows from those values? So in the bioeconomy, of course, again, a lot of it's very positive, and we can think about how, bioeconomy might benefit rural livelihoods. But when it comes to how environmental harm in the bioeconomy might be accounted for in ways that affects poverty, I think that can be really challenging, but again, it's really important. And it's really a matter of identifying who are the victims, whose livelihoods are affected, whose cultural resources are affected. And then once those are recognized, coming up with decisions about what needs to be done in order to fix that harm. And again, that might be non-financial, but it might be financial. It might be, for example, payments to rural communities whose livelihoods are directly impacted by certain cases of illegal action. One which I've not explored in detail, but kind of is continually in my mind, is particularly around illegal fishing, for example. Where we know that there's large-scale commercial illegal fishing. This is literally stealing fish out of people's nets and out of people's mouths. And surely that has huge implications for poverty and for livelihoods, as well as for real economic signals that we would be able potentially to use economic tools, but also the court system to help to remedy.
I see. Yeah, thank you very much. That's really, yes, there's a lot of factors to consider, and I'm sure there will be other people who would like to know more about that aspect as well. We have three people who have raised their hands, three participants. I'd like to call Nurjayanti. You are welcome to ask your question. Ibu Nurjayanti. Hello. No? Yes, Bu Nurjayanti, you may ask your question. Okay, maybe we'll move on first to our next participant who has raised their hands, and Ibu Ika Widowati, you may. Okay, 1 second. Okay. Yes, Bu Ika Widowati, you may ask your question, please. Okay. All right. So I think, I will just move on again to the questions that have been put forward in our Q&A forum. So, 1 second. Okay. I think there's one question on what is the kind of the best policy and strategy of biodiversity in the pandemic situation. I guess maybe this has more to do with the sense of urgency that we have in the pandemic situation and how that relates to the best policy and strategy in terms of using biodiversity. I think this is an open question to any of the panelists. Please, if you'd like to answer, maybe we go to Prof. Arif first for that. Thank you. Oh, I'm sorry, apologies. So the question was directed to Bapak Dr. Arifin Rudianto from Bappenas. So what would be the best policy and strategy of biodiversity in the pandemic situation? So would you please answer that? Thank you very much.
Thank you, Dr. Andia. On the question, the best policy during the pandemic for biodiversity is a blessing disguise. During the pandemic situation, the demand for biodiversity has increased, especially for the traditional medicine. And in this case, we have a program for the next fiscal year to redesign about our medicine system, in which we will do a new strategy to develop what we call JUMP, based on Indonesian biodiversity, to increase the value added and also to keep the environment. If the people knows about the value of the biodiversity, then they will keep the resources. So we will do from upstream until downstream for the biodiversity, from the production process and then value-added process, and also the marketing, and also the most important is to set up the regulatory framework for the jamu as a medicine based on our traditional wisdom, and then also develop infrastructure to support this value-added process. And then develop another system like digital marketing for the local industry. And so this the best strategy during the pandemic, how to increase the value added of the biodiversity.
See. Thank you, Pak. So we have a next question from Bapak Agung Yuswana. I think this is addressed to all three speakers. So the question is, various arthropods and vertebrates in Indonesia. Some of these species are of high economic value because of their specific characteristics, and the animals have been protected by the state as well. But there is still a lot of trade of these arthropods and vertebrates in Indonesia. And protected animals are being sold in animal markets in several provinces. And the high price of these animals are what motivated people to commit these crimes. How would then the speaker suggest on how to solve the problem effectively and efficiently? So I think we'd go to Professor Arif first to answer that, please. Thank you.
Okay, well, it's not an easy question, yeah. However, there is a tendency of people pretend to know the answer. I'm not going to pretend to know the answer, but what I know is this kind of situation need very subtle attention, and this has to be approached not only from economics perspective, from commercial perspective. This is a multidisciplinary problem. Yeah, maybe there is some financial reward and this incentive element to this, but also I know that there are some element of sociological factors and education factors and local knowledge that may help together, you know, to solve this problem. I think that's what maybe what missing, but I can understand the tendency. If you learn a lot in your discipline, normally you think silo that this is the way you solve it. But I learned that it never works like that. So my answer to that is let's work together, multidisciplinary approach from sociology, economic, to see what is the most effective way to solve this problem. I mean, that's my take to this question. Thank you.
I see. Yes, indeed, we do need cooperation from all stakeholders to do so. So we'll move on to Dr. Jacob Phelps. Would you please give your thoughts on that question? Thank you.
I think Prof. Arif's answer is a really excellent one. And I think specifically to my experience when I was a student interviewing people who were illegally trading wild orchids in Thailand. And my immediate response was, oh, well, it's probably just that we need more enforcement, or we need to give people another livelihood so that they have an alternative. But when I started interviewing them, what I found was that my assumptions, to a certain extent, were often wrong. For example, people were not just trading wild plants because of economic need. People were trading wild plants illegally because they liked wild plants. And so you're referring, Arif, to sociological factors. In some cases, it wasn't just economic need. It was just like you and I, we make career choices. They were also making career choices, right? And of course, there were also economic elements in it. So I think that multidisciplinary aspect is really important. And there's also sometimes a challenging one in the Indonesian system, because in Indonesia, if you do your undergraduate in economics and you want to become an economics professor, you must then do your, as I understand it, your master's in economics, your PhD in economics, and then join in economics. So there's sometimes a very linear educational approach. And yet many of the challenges that we're dealing with, we just talked about pandemic and wildlife trade and economy. They're so complex, and often our systems are, in the UK as well, really struggling. Our institutions are slow to move to prepare us to address these challenges. One of the things that I'm really struggling with is now I'm working a lot with economists and lawyers. I'm not an economist or a lawyer. I'm an environmentalist. But the questions that I ask are very different than those of my colleagues, and the tools that they use are also very different. So trying to find interdisciplinary ways is important, but it takes twice as long. And it also takes a lot more confidence of personal relationship with our colleagues to be able to even understand the problems, because we have to be very patient with each other to understand terminology and concepts, which is sometimes a little bit frustrating.
Yes, true. So I think, I guess we're all also looking forward to what Dr. Arifin Rudianto has to say as the government point of view on this illegal trading, and how to, what's the policy that's dealing with that as well. So could you please give us some of your thoughts, Bapak Arifin?
Thank you. In addition to Professor Arif Ansori and Dr. Yakubai answer, I would like to use answer from law enforcement point of view. Currently, we are depend on government official or civil servant to do the law enforcement. And it's not enough, because the ratio between number of government official and the area that should be covered is very, very big. So we try to develop a community-based conservation management, in which the communities surrounding the national park will also do the enforcement, especially to deal with this illegal trading on some species. To encourage the people involved in this law enforcement, we also develop many programs. For example, social forestry, and also another program in which the local community can get benefit from the area. So they will keep the area, including the biodiversity inside, to be keep and safe. So the answer is, we try to develop community-based management for conservation.
Okay, thank you, Pat. Yes, I'm sure the government's trying its very best to ensure the balance of these, economic development, and also making sure a sustainable future for our children too. So let's move on to the next question. This is for Professor Arif Ansari Yusuf. From an economic point of view, how do we explain to all of the people that economic activity in these cases, when we use our biodiversity, is the right way and make the value for Indonesia, not the losses? So I suppose how we maximize the values and minimize the losses in this regard. Thank you, sir. Would you like me to repeat the question, or?
Yes, please.
Yes, so from an economic point of view, how do we explain to all of the people that economic activity in this case is where we use our biodiversity is the right way and make the value for Indonesia and not the losses. So to optimize the values.
Okay, I will borrow the language that lucky of me happened to say to me directly when I met him directly, which is Lord Nicholas Stern. I met him directly in Oxford, and he said this. So this is not my words. He said that that when our message is correct, right, is right, but people still doesn't understand the message and they still keep doing whatever they're doing normally, it means it's not the message that is wrong, but how we deliver the message that is wrong. So he suggests something like this. Don't pretend because you are an academics, yeah? You are a professor, you are a lecturer, and then you speak with common sense to them and it will easily be transferred. No. He said that sometimes our mistake is we pretend that people think as the same as we are. So my tip, his tips actually, and I agree with that, is that don't only touch the rational when we want to communicate something in terms of the importance of biodiversity, but also try to touch their emotions, because that's what matters. For example, think of something very, very, very direct to them. For example, loss of family members because of natural disaster due to the loss of forest functions. That's an example. So touch their emotions. So maybe that's the thing that we forget, yeah, because not everyone are academics and read journals. So think of something else. Maybe we have to ask also communication specialists, PR specialists, of how we do this together. Maybe that's my answer. Thank you.
Okay, yes, I see. I guess communication is key in this sense, right? So I think I would now move on to one of the questions we get from our YouTube live. So we have Martina Langi from University of Samratulangi, Manado. This is a question addressed to Dr. Jacob Phelps. In your opinion, what would be the most suitable political agenda for saving biodiversity in Indonesia? Thank you. From what you have worked with so far. Thank you.
Well, I'll use Dr. Prof. Arif's previous comment, which is that I won't pretend that I know the answer. And of course, that's a pretty broad-reaching question. But one of the issues that I'm very passionate about, and again, as I mentioned in my talk that I'm working on in Indonesia, is that we need proactive policies that are providing for livelihoods and meeting environmental and economic needs. But I think we also need to look for accountability. And I know that this is an issue that, whether it comes in the sector of corruption or environmental harm, that Indonesia has struggled with and is also taking very seriously. And part of the reason that I focused on Indonesia is that Indonesia, among tropical developing countries, is one of the few and really a global leader in terms of trying to hold primarily, corporate sector as well as large-scale perpetrators of environmental harm accountable. Of course, there's a huge amount of work to be done, but a great deal is being done to use existing laws, criminal and civil, to create accountability. And I think that accountability is really important, particularly when it comes to large-scale environmental harm to promoting sustainability, because it creates a cycle of understanding, right? In that, by that I mean that right now, and to date, there's been a large understanding that in Indonesia and in many other countries, you can harm the environment for economic benefit with relatively little repercussions in many cases. And that's a frustration we face in my home country of Costa Rica as well. And when the rich and powerful get away with these things, in particular, it sends a message to, it sends a certain sense of hopelessness, I think, to us who are making individual decisions in our everyday life. And so I'm really interested in legal mechanisms that draw on economic tools that seek to create accountability at those highest levels. Because I think that not only sends a message to that one individual or that one corporation, it sends messages to entire sectors. And it sends messages to little people like us who don't have a lot of power, but are making decisions and recognizing that actually this is not just a case of everyone out for themselves, that there is a process that is holding people, particularly in these big cases, really responsible for the harm that's being done to our collective resources. So for me, that would be a big policy agenda item, accountability through the legal process.
I see. So I think that would be a big homework for the government of Indonesia as well. And so that would probably link to our next question from Santi from University of Pajajaran Bandung to Bapak Dr. Arifin Rudianto, Pak. So the question is, who is in charge to define national commodity to develop? Do we still have a midterm and long-term national development plan to maintain the sustainability of long-term research and development in Indonesia despite of short-term political policy drivers? Thank you, Pak.
Okay, thank you. In terms of research, I think the government has just developed Badan Riset Inovasi Nasional. And this body is responsible to set the priority of commodity to be studied, and also the value-added process for this commodity. So we still have a long-term plan, also mid-term development plan, and national plan. We already identified many species that has a high value for medicine, for cosmetic, also for traditional activities. And to do this, we agreed from many stakeholders, the academician, also the business entity, and the government, about which species is a top priority to be touched and also for the process. For this, for example, there are many species that already studied and has a potential value for medicine with discipline in phytopharmacology. I think LIPI is very pioneer in this. But the problem is how to make this in mass production. The link between research and industry. This more as about regulatory framework, because involve also the patent right about the value, economic value, who will buy the process. So most is about regulatory framework rather than scientific aspect. This is our homework. The answer of the question is the Ministry of Research and Technology has the responsibility to set the commodity and will be coordinated by coordinating ministry, because if we will do value-added process, then involving Ministry of Industry, Ministry of Trade, and so on. This becomes the responsibility of coordinating ministry. But Bappenas will guard the process from upstream until downstream.
I see. Thank you, Bapak, for that very elaborate answer. Well, there's one question as well addressed to all of the panelists, and I think this is a great concern to a lot of the countries as well. As the question asked by Irika Devi Anggraini. So in these recent days, many countries have struggled from biodiversity loss caused by forest fires. In some aspect, this is kind of inevitable as the result of climate change, as we've seen in the US, in Indonesia, and in Australia recently as well. And we also, in Indonesia, we have struggled from illegal civil activities as the cause of forest fires. So the question is, how do we reconcile this challenge as in the future, this is likely to happen again continuously? And that would mean a great loss to bioeconomic resources every year. So what would be the most likely approach to reconcile and address this challenge? Maybe we'll go first to Professor Arif for this question, and then we'll go on to Jacob and Bapak Dr. Arifin. Thank you.
Sorry again, the question is, I didn't really get it.
So how do we reconcile the challenge of continuous forest fires in different countries?
Forest fires.
Yeah, thank you.
Well, I always give, actually in many presentations about economic evaluations, I always mention this, that a recent studies conducted, I think it published in a top journal, I don't remember, the avoided death, yeah, the avoided death because of the last big forest fire is equivalent to Indian Ocean tsunami in Aceh. I think I think if I remember, if I'm not mistaken, is half of the death avoidable death because of the haze in Kalimantan and Sumatra is close to almost close to Aceh tsunami. So this is if we talk about avoided death, it means that the death that we can prevent if the haze didn't happen. So this is due to or attributed to the forest fire alone. And that's the economy, of course, and there's a value, this unprecedented value, the human loss of that magnitude. So we don't really need to count the dollar value of this debt. We don't need to. As far as we acknowledge that there are family member loss because of forest fire, then we understand that the cost is, is very huge, very paramount. Yeah? So I think we just need to think more and then read more and communicate more. That's the most important thing. Thank you.
Yeah, sure. I agree. Thank you for that answer. So we go on to Dr. Jacob now. Would you please give your thoughts on that as well? Thank you.
Well, so on one hand, there's issues, of course, of awareness and how people perceive the problem. And of course, for those of us internationally or people living in Jakarta, it's going to be fundamentally different from an oil palm plantation owner or, a farmer who's hired to work on someone else's land. And I think that, these fires are happening because of behavioral choices all across that, the types of products we buy, the types of policies we support far away, but equally the types of policies that are supported at the national and local level, and then the decisions that individual companies and individual farmers are making. So I think part of the challenge is, of course, one of awareness and communication, as Prof. Arif was referring to earlier. We did a study where we were looking at how different people perceived the problem of peatland fires across those different types of contexts and scales a couple of years ago. And unsurprisingly, people perceived the burdens, the costs of those fires in very different ways, depending on who you are, if you're in the sector, or if you're a farmer, or if you're just a regular citizen who's not in farming or whatever. And they also, of course, people are doing fires because there's benefits to them, right? They're not burning for fun. They're burning because it provides, economic benefits to them in different ways. And those benefits were also perceived in different ways. And what we found was that actually, as well as and then what do you think the solutions are? People also really clustered in very, very different ways. But there were a couple of issues that people agreed on. And this comes back to Prof. Arif's first example around public health. Even though some people really cared about biodiversity harm and others didn't. But the one thing that everybody really focused on in our sample of over 200 people across these contexts was public health. Because, and as I think Prof. Arif also mentioned earlier, that emotive element about what does this actually mean to my life, to my neighbors' lives. So I think that it's about identifying common messages that we know are going to resound with different people in different contexts. But even then, awareness, I'm aware that flying is bad for the environment, but I still do it. And I use that example to show that awareness is not the only part of it. We need to understand and care about the issue, but we also need to have incentive structures around us that favor pro-environmental behavior. In the case of fires, this isn't just a case of an individual farmer or an individual company on one night deciding to set a fire or not to set a fire. They exist in a broader structural set of kind of a governance context that creates incentives or disincentives for them. And right now, the reality is the chances of getting caught are low. The economic benefits are generally high, and the perceived repercussions social and environmental are often disregarded. So I think it's about revisiting some of these, sure, using enforcement, and that's of course been an important and continues to be an important approach for the government, understandably, and it should be. But I think it's about thinking again about what are the incentive structures that are allowing people to continue seeing benefits of the same behavior. Because they're not stupid, they're doing it for a very clear reason. And I think sometimes we're failing to ask the question, why are people burning? Instead of how do we stop them from burning? You have to understand the motivation before we can start to come up with a cause. And a motivation that just says, oh, because it's profitable, that's the start, but we need to understand a little bit more in depth about why. Is it because people can't access tractors? Is it because there's corruption? Is it because the people, the processing plants are not checking chain of custody to make sure where this came from? Is it because enforcement's too weak? And I think that a more structured approach maybe can give us a little bit more of an understanding about the benefits and burdens that different people experience. And then, of course, how we can intervene.
Yes, sure. So we get to the actual point of the problem, not just the symptoms and what appear outside. I see. So, Bapak Dr. Arifin Rudianto, would you also kindly give your thoughts on this question, I guess, as a government perspective on this?
Thank you. In terms of forest fire, our strategy is focused on the prevention action rather than reactive action. And this also involving all stakeholders, not only government, but also non-state actors, the private sector, the academician, local organization, and also the community itself. And also involving all levels of government, central, provincial, district, even village government. And to do this, we use so many approaches. For example, we applicate the technology to identify the hotspot from the DMKG. From this information, then we inform all level, especially the district government, and the district informed the village government that this area is in danger. So they have to prepare to do something when the fire is in a little area rather than after become large area. And also we use many approach to deal with the cause of the forest fire, like Pak already mentioned, but the farmer who have agricultural practice to burn the land. No, we have more environmentally friendly technology to do this. So we do some approach about this. And then also to develop awareness among the people. For example, in village area, we also train many young people to become Panggala Ahli, youth people who care with forest fire and has experience and capability to overcome the problem when still in small scale. So focus is on in the prevention and involving also regulatory framework with more penalty for the, especially the private companies who obey the regulation, and also reward for the everybody, individual or also company, who do the best thing to do the prevention of forest fire. So there is a reward and also punishment, but also we prepare and do some approach, innovation approach in the field.
Okay, thank you, Pat. Sounds like a lot on your plate as well on that regard. So hopefully the best for Indonesia and other places as well. So I think in the interest of time, we have come to the end of this session. And I guess this has been quite a fruitful discussion where we learned so much about biodiversity valuation and valuing gains and losses and the importance of communication across multi-sectoral and multidisciplinary approach to trying to deal with the issues with biodiversity use and ensuring its sustainability. And yes, we have also seen what's happening in Indonesia and what approaches have been done and what are going to be done to ensure that we can benefit optimally from having our great wealth of biodiversity and making sure it's still here for decades to come. So I would like to thank you, all of our speakers, Prof. Arif, Dr. Jacob, and Bapak Dr. Arifin Rianto. And thank you for all of the participants for giving questions and paying attention to this session and also all of the sessions we've had since this morning. And so as the moderator of this session, again, I say thank you, and hopefully everyone has had a wonderful time, and hope to see you all in the next webinar. Now I would like to return this session to our MC. So thank you very much, everyone.
Thank you.
Thank you so much to Dr. Anggia as the moderator for the last session and to our wonderful speakers. Ladies and gentlemen, proceed to the next session. Closing remarks by Director, Research Center for Biology LIPI. Dr. Atit Kanti, time is yours.
Okay, thank you very much, Renata. Good afternoon, distinguished guests, ladies and gentlemen. Thank you for still joining a very long webinar today. The International One-Day Webinar on the Indonesian Biodiversity has achieved good success. I would like to thank all the speakers for delivering very good information and for spending a few hours during very busy times. Our panelists for having advanced our thinking on the interconnected session in these webinars. I would like also to thank to LIPI management and the committee team for the excellent coordination of these webinars. I know this is a lot of work to get so many people gathering together in virtual meeting to discuss the mainstreaming of biodiversity, conservations, bioprospections, and bioeconomy. For sustainable livelihood, participants from various backgrounds have participated actively, especially for decisions. Very great and very good active discussions during these webinars. Actually, biodiversity, conservation, bioprospection, and bioeconomy are interconnected and mutually supporting pillars for our sustainable futures. Biodiversity conservation will support the sustainable practices on bioprospections and bioeconomics. Finally, the insightful talk and discussion from this webinar hopefully can provide recommendations for the Indonesian governments, especially Dr. Arifin is here from the Bappenas, and in formulating national policy that harmonize the importance of biodiversity as well as the livelihood of Indonesian peoples. And that concludes my statement in these occasions. I hereby officially close the International One-Day Webinars on Indonesian biodiversity, mainstreaming biodiversity conservations, bioprospection, and bioeconomy for sustainable livelihood. Warm regard for our deputy, life sciences that cannot be able to attend this webinar today. Thank you very much again for your cooperation, and we wish we can work together closely for our biodiversity, conservation, and its sustainable use. Good afternoon, everybody. Please stay safe and healthy. Thank you very much.
Thank you. Thank you so much, Dr. Atit, for officially closing this today webinar. Ladies and gentlemen, finally we come to an end to an international webinar on Indonesian biodiversity, mainstreaming biodiversity conservation, bioprospection, and bioeconomy for sustainable livelihood. Wednesday, September 16, 2020. We would like to thank you for spending your time with us today and all the active participations. Thank you for the speakers, moderators, the committee, and last but not least, our distinguished participants, ladies and gentlemen. And we would like also to remind you that due to the pandemic of COVID-19, please practice the health protocols properly. Keep the physical distancing, wash your hands regularly, and please wearing mask everywhere you go. Let's take care of each other by staying at home if it's possible. Stay safe and healthy. Finally, we would like to apologize for any, and of course, the next series of WEBINUSA. Good afternoon, see you and I see you.
Thank you.
And we have
Perkenalkan, silakan untuk diaktifkan videonya, baik kawan-kawan behind the scene. Terima kasih Pak Arif Rudianto sudah berkenan hadir. Jacob Phelps, thank you so much. Pak Profesor Arif, Bu Atit, Bu Kapus, Bu Lilis, Pak Iman, terima kasih sekali sudah hadir. Ini. Baik, mungkin bisa di-on-kan kawan-kawan.
Baik, Bapak Ibu, kita akan foto sesi pertama. Yang pertama, mohon untuk di slide pertama untuk mengaktifkan videonya. Mohon maaf, Ibu Dr. Puspita mungkin bisa mengaktifkan.
Sebentar ya.
Oh iya, baik Bu, maaf. Kami tunggu Ibu. Persiapan juga untuk Bapak Ibu yang ada di slide kedua untuk mengaktifkan videonya.
Langsung aja Mas, nggak apa-apa.
Baik, Bapak Ibu, kita akan foto bersama di slide pertama. Hitungan ketiga ya. 1, 2, 3. Cheese. Sekali lagi, 1, 2, 3. Say cheese. Kemudian untuk slide kedua, Bapak Ibu, kita foto bersama di slide kedua. Baik, dalam hitungan ketiga kita foto. 1, 2, 3. Sekali lagi, 1, 2, 3. Terima kasih Bapak Ibu atas kerjasamanya. Mohon maaf atas segala kekurangan hari ini.
Terima kasih.
Terima kasih banyak semuanya.
Terima kasih.
Terima kasih Bapak Ibu.
Enjoy.
Ya, makasih Pak Ayu. Hebat Mbak Ayu.
Terima kasih. Sekia.
Yes, sudah beres ya. Selamat hari yang nyenyak. Makasih banyak semuanya.
Masih live YouTube.
Nggak apa-apa. Makasih Bu Lili sudah hadir dari pagi Bu Lili sama Pak Iman.
Oh, menarik ya.
Menarik ya, bagus-bagus itunya.
Bagus.